Yep, 1, 2. 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 Yep. Yep, 1, 2. 3, 4, 1, 2. Yep. Yep, 1, 2. Yep, 1, 2. Yep, 1, 2. Yeah. Yep, 1, 2, 3, 4. Yep, 1, 2.
Is the lightning good now? No, yes, maybe. Mute me. I'm done.
Yep, 1, 2. Yep. Yep, 1, 2. Yep, 1, 2. Yep, 1, 2. Yep, 1, 2, 3, 4. Yep, 1, 2. Yep, 1, 2. 2, 2, 2. 1, 2. 3, 4, 1, 2. Yep, 1, 2. Yeah. Yep. Yep. 1, 2, 2, 2, 2.
Can you see? Why don't you go up? Good morning, everyone. Welcome to the 20th H3Africa Consortium meeting. Thanks for your patience. We have our honored guests present now. So I'd like to open this morning by welcoming our sitting H3Africa Steering Committee Chair, Zanay Lombard. She's Associate Professor of Human Genetics at WITS and NHLS. Her focus is around human genetics, also bioinformatics-driven approaches to disease gene discovery. She also represents the DDD Africa project, and she's also affiliated to WITS Health Consortium. So please allow me to welcome Zanay to open this morning's proceedings as well. Thank you so much.
Good morning, everybody. Thank you so much for joining us this early on a Monday morning. I'm very excited to see everybody here and really very excited to welcome you to hear more about what H3Africa has achieved over the past 10 years, but also to look forward to the future and think about what our next steps are and where we're going. So I really like this picture to just illustrate what the footprint is that H3Africa has left over the past couple of years across the African continent as far as building capacity for genomics. As you can see, there's a wide spread of projects and sites that are supported. And this is really illustrated that there's 51 projects across 30 countries where H3Africa is currently doing genomic research, building capacity, and really bringing the genomics revolution to Africa. So this is just a summary showing you some of the achievements of H3Africa, which we're really proud of within a 10-year time span, which is really not a very long time. I feel the consortium has achieved so much and has made such an impact. What we can see from these figures is this rise in one of the outputs that we monitor, which is publications from African scientists in genomics. Just really this wonderful upward trajectory that we're seeing that is to some extent boosted by H3Africa, but also with some other activities that's happening on the continent. So something to really be proud of. And we're excited to showcase some of the success stories, some of the outcomes from the H3Africa projects this morning, and hope you will be able to engage with that. And one of the other really important successes for us is the young researchers, emerging researchers, students and fellows that have come up through this program and who will also be sharing their stories and their research with us today. But I think really what we want to do with this meeting today is to think about the future and what the next steps are. So as great as that upward trajectory is, I think we need to contextualize it that there's so much more to do for us and there's so much more that we want to achieve on the African continent. Professor Charles Rotimi, who's one of the founding fathers of H3Africa, I think has always given us the message that we need to dream big and we have to have a dream and a vision for Africa and for genomics. And myself and my co-chair, Gita, along with the steering committee, recently wrote this commentary to really think about and position ourselves onto what we want for genomics in Africa. And we really want Africa to be the future for genomic research. We want this to be a home where excellent world-class research is being done, but also where genomic medicine is being envisioned for the health of people who live on the African continent and also for people across the globe. So I want to encourage all of you not only to bask in the glory of what H3Africa has achieved in the last 10 years, but also to purposefully today engage with our guests here, with our visitors here, but also with our scientists to discuss and think about what the future is and how we each can play a role in making sure that Africa is the future of genomic research on the continent. Thank you so much. I hope you really enjoy the talks today, the engagement today. I'm going to hand over to our independent expert committees to Charmaine and to Professor Bloom to speak to us and to welcome you all and open the proceedings. Thank you so much.
Zene, thank you very much for that wonderful introduction. And thank you everybody for coming. It's my great pleasure with Charmaine to represent the Independent Expert Advisory Committee and welcome everybody to this, I think, really important, wonderful meeting. I thought perhaps I would begin with a kind of evolution of H3Africa as I experienced it. It really began as a concept to have a consortium or to have a major African genomics program in 2003, when some of the senior scientists got together and decided to create the African Society for Human Genetics. And their view was to exchange knowledge and views to enable the science to be developed in the African continent. In 2009, when I first was involved, there were fewer than 3% of all the genomes that were sequenced that came from Africa and African Americans. And there was a meeting at the Wellcome Trust to review their international and population programs. And subsequent to that, I wrote a letter to the director of the Wellcome Trust, Mark Walpert, suggesting they might link two of their existing programs in a sense together. One was the major thrust of the trust to study the human genome, particularly at the Sanger Center. And the second was a program that wasn't doing well to somehow link networks of laboratories in Africa. They took that seriously, and the trust created a meeting in 2009 in Yaoundé. And the first speaker at that meeting was, at that time, an unemployed human geneticist named Francis Collins, who was at that time waiting to be confirmed as director of the NIH. And he gave a marvelous talk, and the first picture in that talk is a picture of Francis in Nigeria doing a surgical procedure as a medical missionary. And at that point, I really knew that the director of NIH for the first time in my lifetime in global health was really committed to global health. It was a really moving moment. The result of that is that members of the board of trustees came back, and the Wellcome Trust committed to creating a program on H3Africa. Didn't have a name at the time. It was to foster human genome research. Francis became director of NIH, and the first major meeting that he held was the first meeting ever that I'm aware of where the institute directors of all of NIH were invited to hear visions of how NIH could contribute to global health. And that said, there were two major committed donors to making a program work. After many, many revisions and suffering, the name H3Africa was agreed upon, at least by most people, and that has wonderfully stuck. So what were the premises when this was being formulated? And I think the first premise was, it is appalling that so little was understood about the most evolved genomes in the human species. We all came from Africa. The linkage disequilibriums are smaller. The potential to learn things that cannot be learned elsewhere are sitting in Africa, and they were scientifically being, to a large extent, ignored by the world scientific community. I think the second point is that the diversity within the African population made it possible again to see things that would not be possible in other more homogeneous populations. And one of the outcomes of the British Biobank, much has been learned, most of the sequences come from there, but there is a lack of diversity not comparable to anything you would find in Africa. The second thing is that there were limitations, to put it nicely, on the level and quality of biomedical science in Africa. And the idea of a program to put human genetics, the cutting edge of all modern biomedical science, as a priority for the H3Africa program was a way to leapfrog all of the steps that had to be gone to elsewhere to move African science directly to the frontier. There was a third concept. There was a debate about creating centers of excellence, which is a great idea, versus networks, which is another great idea. And I argued strenuously that it was a better idea to create networks. And what you just saw from Lane indicating participation in 50 countries and each of the projects requiring collaboration of other institutions is unique and novel to my knowledge. And that has given this program a continental network in everything, as you will hear from BioNet and the collaborative centers in each of the diseases. And finally, the way the funding went was by competition, which I'll speak to briefly at the end. The best ideas don't come from bureaucrats. They don't come from professors emeritus like myself. They come from the scientists themselves. So the focus was to create a program where the competition of ideas would come from the scientists. And that meant creating grant programs, which both of the institutions did, allowing competition, and then hopefully selecting the very best. The accomplishments Lane has already described to a great extent. It's just astonishing how, starting so small in 2012 officially, we have 700 fellows, 700 publications, almost 1,000 fellows, and many, many countries involved in collaborative research, working in various levels at the frontiers of human genomics, but not to forget pathogen genomics, which built on the trypanogen program and built on the Wellcome Trust malaria programs. And it was wonderful to have dinner last night with somebody working on chlamydia, which I hadn't even known was part of the program. So the program has broadened, and the ultimate game is there to look at the relationship of the pathogen genomes and human genomes and try to get ahead of the next set of epidemics and pandemics. The diversity of data that has already been accumulated has established the differences in ancestry, in diversity, genomic diversity, and that enabled the first Africa chip to be made, because the original chips that were being used in the West didn't have enough genomes to be able to give accurate information about the evolution of Africa. And one of the great rewards of this program has been to study the movement of genes for the Great Migration, for example, that would not be possible without things like the Africa trip. Clearly, after 10 years, African scientists have made already major contributions to genomic science and to biomedical science. And Lane asked us to think about how to move forward in the future. And that requires some explanation as to why, as it has been constituted, the program is not going to continue in its previous form. And I would start with the concept behind the creation of the NIH and the National Science Foundation in the US after World War II. And a scientist, a physicist named Vannevar Bush, had a vision that if you want to be a great country and really look after the health and prosperity of your people, it is science that has to lead the way. And he created a concept paper called Science, the Endless Frontier. The frontier has always to be moving, and that means science is always changing, and that requires selection, essentially genetic selection for the best ideas in science. So nobody that I know of, at least in my little world, gets lifetime grants to study what they want to study. We live in a world of competitive science where our ideas are competing with everyone else's. And I think that is painful for all of us who have to write grants every 5 years, but I think it's inevitable for the frontier to continue to move. I think what is not so clear is that is absolutely true of donor agencies and foundations. They have to move their frontier. Their funds are catalytic. And it is unusual to have a foundation or donor continue a single program for more than 5 years. We've been given the luxury of 10 years. And it is now up to us as a group, as a consortium, and of the broader donor community to figure ways to maintain at least both the science and the consortium collaborative context. We know that the NIH has now put forward a new program on big data, and we know that H3Africa scientists are eligible to compete in that program. And we know that the Wellcome Trust is initiating a new program. I understand it less well now, but it will have to do with consortia and population studies, all of which would be needed and very relevant to all of you who have been part of this program. So it is our hope, and you'll hear much discussion of ways that we can build on the legacy of H3Africa to reduce the inequities in not just genomic science, but in health between the populations in communities in Africa and other parts of the world. And the ultimate game is to improve human health wherever we can. And now with that, I am delighted to turn this over to my colleague, Charmaine Royal.
Thank you, Barry. It is such a delight to be here and to see everyone again at the 20th meeting for the H3 Africa initiative. I'm sorry I was not able to make the last meeting in Abuja because I had COVID at the time. And so I'm just happy to be here. Like Barry, I just, the progress of H3 Africa has just been remarkable, and we're here to celebrate it again today. And Barry, you talked about the beginning of the conversations about H3 Africa and this question of whether to do networks or centers of excellence. As we're talking now about the future of H3 Africa, we realize it doesn't have to be either or, it's both and, right? So now we're talking about centers of excellence as a possible next move, next step for H3 Africa. So it's just a matter of time before we get to centers of excellence, and who knows where else. Before we hear from, we're going to hear from some of our projects, the H3 Africa projects. We're going to showcase, we're going to highlight some of those. But before we do, we're going to hear from the Consul General of Cape Town, in Cape Town. So Consul General Todd Haskell assumed the position of Consul General in Cape Town in April 2022. He joined Mission South Africa as a change of affairs at interim in March 2021. Mr. Haskell had previously served as United States Ambassador in the Republic of Congo from July 2017 to January 2021. He is a 35-year career member of the Foreign Service, class of Minister Counselor He served as a Deputy Assistant Secretary in the Bureau of African Affairs from 2015 until his appointment as Ambassador to Congo. Prior to that, he served as the Director of Public Diplomacy and Public Affairs, directing the Department of Public Diplomacy Personnel and Resources for 48 US embassies and consulates in Sub-Saharan Africa. In both of the latter positions, he worked with US embassies in the field and agencies in Washington to implement the Mandela Washington Fellowship, the signature program of the Young African Leaders Initiative. Mr. Haskell previously served as a public affairs counselor in the Dominican Republic from 2010 to 2013, as public affairs officer in Johannesburg, South Africa, 2006 and 2010, and as public affairs counselor in Burkina Faso, 2003 to 2006. With that, I welcome the Consul General.
Hermine, thank you for that very generous and long introduction. That was great. I appreciate it. If it hasn't already happened, I'm the U.S. Consul General here in Cape Town. If nobody has welcomed you to this beautiful city, let me be the first to do that. Welcome to Cape Town. I think you'll enjoy it. I had the chance to look at the agenda that you've all got over the next 2 days, and I'm very impressed. It sounds like you're going to all hear from a lot of really smart, expert people who will be talking about this issue, the future of genomics research. I'm going to tell you, you're not going to hear about that from me because I'm one of those bureaucrats who don't come up with great ideas. But I know enough to listen to scientists when they do. No, it's fine. It's fine. I have no doubt that I'm probably about the least expert person in this room, but I am here because a priority for the United States is pursuing partnership to overcome shared challenges with shared solutions. And I'm here also because the US government has invested in, since 2010, in fact, genomic research on the continent through H3Africa. And we have done this because, thanks to experts like you, thanks to scientists like you, we know that genomic research helps us understand humans at a molecular level, as well as their pathogens and interactions with the environment, allowing us to partner with experts globally to develop tools like diagnostics, like therapeutics, like vaccines. Well, a number of initiatives have supported cutting-edge human genomics research on the continent. I'm proud to say that this initiative, H3Africa, supported by the United States through our National Institutes of Health, has been a leading contributor to this field, providing research, collaboration, infrastructure, training, and most importantly, partnership networks. In fact, since 2010, the US, NIH, and partners in the UK and the African continent have invested over $175 million in these activities. We know that there's still more to be done, including more research capacity and more transnational science to advance public health implementation at our local clinics and hospitals. And looking ahead, the US government continues to believe that it is vital to establish innovative, sustainable financial commitments for genomic research, including both through public-private partnerships to invest long-term and at scale in both people and infrastructure. We believe these investments will advance not only science and public health practice, but also an enabling environment for researchers and healthcare practitioners to continue working in Africa. And that's important to local populations here and on the continent and globally. As the committed funding for H3Africa comes to an end, the United States is committed to addressing global inequity and health-related genomics through programs such as, and certainly not limited to, the Data Science for Health Discovery, the Innovation in Africa Project, or DS1, whose second consortium meeting was also held here in Cape Town back in October. Everybody likes to come to Cape Town for conferences, and I've learned during my year here. The United States government, we stand ready to work with our partners to continue this genomic research, and I look forward to seeing what we can accomplish in the decades ahead. Thank you very much.
Okay. Well, thank you, Mr. Haskell. So we're gonna hear from some of the H3Africa projects and get some updates and hear a bit about the fantastic science that's been happening in H3Africa. We're gonna start with Professor Mogomotsi Matshaba, who is a principal investigator for Collaborative African Genomics Network, that's CAFGEN group. Hold on a second. Okay. And he's clinical associate professor of pediatrics, retrovirology at Baylor College of Medicine, and executive director of Botswana Baylor Children's. Boy, you just got too much here. Children's Clinical Center of Excellence. And he's going to talk about CAFGEN, celebrating a decade of excellence in collaborative genomic research in Africa. Mogomotsi.
Thank you, Charmaine. I'm not alone. I'll be with Dr. Gaunere Tshabile, who's one of our trainees. It's good to see everybody back here. And I hope that by the end of our presentation, we would have shared the little bit that we can share in 15 minutes. It's very difficult to compress 10 years of work into 15 minutes. So we have chosen to go through high-level achievements that we have gone through, and I'll take you through that. So this is our outline, and like we usually do, we look at learning from the past, learning from our culture. And one of those is a long-term vision of whatever you want to do. So if you plan to sit under a tree in your retirement, you need to plant it now. We planted a tree 10 years ago. We are beginning to sit under the shade of that tree. And I think it's important that we celebrate both the challenges as well as the successes. I'm preaching to the converted here. We know our global problems. HIV continues to be a huge problem in our region, TB as well. So because of that, when this opportunity came up to study genetics and genomics and do training in sub-Saharan Africa, we thought, why not combine these two? We looked at it from the view of a child that has to deal with both TB and HIV. As has been said, less than 2.5% of all genetics work in the world was from Africa. So to redress that, we set up a consortia of different, apologies, I think something did not load there. There's supposed to be a map there. Of different institutions. So 2 continents, this is Africa and the US, where the US, we're talking about Baylor College of Medicine. Then 4 countries, Botswana, Uganda, as well as Eswatini, including the US. And then 6 centers. Our center as the coordinating center of these institutions. Again, the infrastructure was there in principle, but what was missing was the technical expertise to answer some of these questions as we moved along. In terms of the science, we tried to look at 2 extremes. These are your rapid progressors. These are children who actually develop symptomatic disease before the age of 3, as well as long-term non-progressors. These are those who do very well without treatment for a long time, and study these phenotypic extremes and try and figure out what could be contributing to that, because it was not just by chance. So, like I said before, what was known at the time was mainly from Europe, because most of the genomes that were analyzed were mainly from there. We knew about CCR5. We knew that it was low in preponderance in Africa. We also knew children are not small adults. So again, meaning that we needed to study this in detail in our network. We have a network of clinics across the continent and 6 countries. This is Baylor College of Medicine working with different African governments. So in Botswana, Uganda, Lesotho, Eswatini, Malawi, as well as Tanzania. We have centers in there. And these centers provide a resource of participants, where in the 3 countries, we're looking at over 14,000 active ART patients who are children. So again, we leverage this resource to actually get to where we are. As part of this, there was community engagement. And as part of that, each community was different. And to try and unify and get the same message out, we developed Genome Adventures, which is a comic book that takes children all the way to university students through genomics. And it has widely been accessed and translated. We've had requests from Middle East, we've had requests from Egypt. So it's been translated way beyond this. It currently is in about 8 languages, English, and then 5 African spoken languages, largely Hausa, Arabic, French, Portuguese, Setswana, because of where we come from, Luganda, Isiswati, we are still working on. So again, sharing the knowledge as much as we can. A lot of engagement has been through social media, and I think we have done relatively well from that perspective. In terms of what we have, biosamples, these are some of them analyzed and some not yet analyzed. Those are the numbers. I'm not going to go through them. We have 425 sequences, 1,000 exomes, some of them using the Africa chip, which we participated in getting it off the ground. And the clinical data I've talked about, and then the equipment as well as the people. So again, all of this in about 10 years of investment. I might say a modest investment given what has been achieved. I'm going to hand over to Haone, who's going to talk to us and take us through the science and what we have achieved. Thank you.
Good morning, everyone. Thank you, Moro. So for the questions that we were addressing, as Moro has described, we were looking at pediatric HIV progression, as well as diagnosis of TB in children in the context of HIV infection. And these are the people who led some of those analysis, where we basically used the DNA that we collected to look at the coding part of the genome, as well as look at off targets. So we took a view that some of the genome that is ignored can be informative, as you'll see with the highlights that we are going to share with you. Some of this work has been published and some of it is ongoing. So I'll start with a description of the TB. So basically here we used RNA from whole blood. We sequenced over 100 transcriptomes, and we took an approach where we're looking at differential gene expression, which is led by Busi, as well as the use of transcripts, which is led by Eric. Busi is from Botswana and Eric is from Uganda. So they're trying to look at ways in which TB diagnosis can be improved, given that it's kind of hard to diagnose TB in children. But some of the things that we are seeing are that some of the signatures that are associated with TB infection seem to be population specific. But we are hoping that we can add to the body of knowledge to be able to discriminate infection in children in the context of HIV. So part of that involved training at DCM, as has been described, as well as training at the African universities that are associated with CAVGEN. And the analysis that I'll be describing were led primarily by the students. What we wanted to first do is understand the variation within our populations, given the fact that not much had been known at the genome level for these populations. And what we found was high levels of, high levels of uncaptured variation, as you can imagine, with underrepresented populations, where basically this work is showing that some of the previously studied African populations are not well representative of all the other African ancestry populations, and therefore it's important to understand those populations when you're working with them. For example, when you look at some of the data that we had where you had common variation in exact for African ancestry population, which tended to be common in the Botswana population, as shown here between Botswana and Uganda. We also showed high levels of IBD sharing in the Botswana population, which seemed to be highly related, but at the same time, the inbreeding levels were not high, suggesting that this is a population that can be harnessed for genomic studies. We showed differences between the Southern African populations and the West African populations within the context of the 1,000 genomes and EXAC. This provided a context for some of the results that we were seeing, where we had promising putative hits, which when you're trying to replicate ended up being common in the population. So it helped to put a context in what we were doing. So Savannah led the off-target work, and his work showed that there seemed to be some immune modulation by virus infection when you compare the viruses that were found in childhood among participants from Uganda and from Botswana. Although there's the caveat that you're probably not sure whether the virus infection is a consequence or is leading, but we feel that the evidence suggests that it's immune modulation when you compare it to the virus infection among the adults. Sam led the HLA work, which has been published. He did some allelotyping that showed some novel HLA-C variants associated with long-term non-progression, suggestive of the utility of doing studies like this, and could help with developing epitopes in vaccines. Finally, I will look at highlight two analyses that are looking at rare variants as well as genome-wide association studies, which are not yet published. We have work led by Marion, which has shown a potential variant that is associated with a long-term non-progression, which, when you look at it, is very rare in non-African populations. But when you look at the transcripts of this variant, is found in the esophagus as well as vaginal tissue. This is a variant that is associated with control of interferon gamma, and its presence and absence can lead to either increased HIV acquisition or decreased HIV acquisition. So suggestive that it can be involved in the control of HIV. Finally, using the H3Africa chip, we have work that is led by Lesedi Williams, which has hits in the HLA as well as in chromosome 7. Basically, when you look at the two populations, the allele frequencies there are kind of different, but when you do a meta-analysis using the Uganda population, you get a genome-wide significant hit there. This is a gene in MAGI2. Interestingly, the work from H3Africa shows that it's downstream of an area that is under selection in the African ancestry populations. The model that we think is demonstrated by this variant is a dominant one, where the presence of the C allele leads to increased odds of disease progression. So those are some of the highlights of the things that we are currently working on, utilizing the things like the H3Africa chip, and we are hopeful that they shall soon be published. One of the things that the trainees are also involved in are looking for sustainable sustaining the work that they are doing is leading in grant applications. For example, they've put in a grant for the DSI Africa opportunity where they are looking to leverage the clinical data that exists within these centers of excellence and put it together with the genomics work and deal with some of the persistent issues that are found across Africa in these countries with respect to things like TB, as well as the things that are resulting from, for example, lifelong use in ART in children. So look into the future in terms of children who have been on ART for life to try to deal with some of these questions and understand them, the development of non-communicable diseases like metabolic syndrome. With that, I thank you, and I'll give it back to you, Moho.
Thank you, Raune. Next slide. And just to end on, we just didn't go through this work that we have shared, but we also used the opportunity to look at other areas of study. For example, in pediatric cancers, where we are part of a network that looks at pediatric cancer in Africa. We are looking at vaccines. We are training in that. We have set up Botswana Genomics and Bioinformatics Society that will be guiding genomics work going forward. This didn't exist before. We've contributed to the COVID response in both countries, actually in all three countries. Our experts were sitting in those. And we are currently looking at a new study, which is Kwashiorkor and genomics in Botswana. On the far right is the CDC director in Uganda, Health Security Agency through Makerere, as well as Baylor, Uganda. So again, this one things that we didn't think about when we started, but we've had input thanks to the funding that we got from H3 Africa. And as we conclude, I think it's only fitting to think that the tree that we planted 10 years ago now has very deep roots, and part of the roots are here. And we hope that we will continue to engage and collaborate and work together to answer more difficult questions. This would not have been possible had it been not with the funding through both Wellcome Trust for Genomics, Comics, as well as NIH for the substantive work, participants, governments, and collaborators that we've worked with. Thank you.
Thank you, Motso and Haon. Great work with CAFGen. And I must say the Genome Adventure is one of the few comic books in genomics anywhere. And so that is just fantastic. And it's not easy to break down genomics for kids. So great work there, great science too. So questions for the CAFGen team? Well, you want to maybe sit over there so people can see it. Okay. Yeah. Any questions?
I'll take the simple ones.
Do we have a question? Do we have a roving mic? All right, so we have a question over here.
Okay. Thank you very much for that nice presentation. My question is about sustainability. You have some more than 6,000 plasma that are probably stored somewhere. and you have some sample that needs to be genotyped. So do you have a plan for those sample, and if you could do more genotyping of your sample?
Respond already?
Mhm.
Okay.
Yes.
I'll take the first step on sustainability. A very good question, Segun. I think as part of the original team that designed this, the wish was always to build almost like a biorepository of samples, data, and all of that going forward. The reason being, from the way we were designed as a center of excellence, as the coordinating center of excellence, we've always stored samples. We have samples going back to 2003, not for genomics, but for future studies that we don't know about yet. So as part of the design of this consortium, that was taken into consideration. Then it happened that along the way, around the time we were starting, there was discussion on generalized treatment of all those children with HIV, meaning that some of these populations will disappear in the future because they will be treated from birth instead of them going on to be long-term non-progressors that we know about. So this became even more important, that we store a lot of samples, of course, ethically, as you expect, and make sure that when those questions come up in the future, whether it be genomics, the omics, and all these other ones, we can go back to a point in time based on the samples that we have. So as part of that, I think it was working to that as a sustainability plan. I don't know if you want to add anything, Paula.
Yeah, I think we're also looking to leverage South-South collaborations as well as the other collaborations we have with expert partners. But we have people at our local universities that we make aware of the samples that we have and the questions that are possible. And we offer genomics capability to address some of their questions, whether it's basic or translation. So we're always looking out for collaborators, both local institutions, South-South, and partners in the North.
All right, great. One more question, Michelle.
Hi, so I think maybe for Kayoni or both. So when you showed that expression in the mucosa of the variant or the NLX, was it the NLX gene? I was just wondering, do you have any idea how that actually happens? How does it functionally sort of capture HIV? Is it a surface protein? Maybe you can elaborate a bit.
Yeah, it's a regulator of interferon stimulation. And there have been studies that have shown that when you have this variant, you are more likely to, in terms of switching on interferon gamma responses versus switching them off. So you're predisposed, whether you're predisposed to Th1 versus Th2 responses is affected by whether you have this variant or not. So we think that that's the pathway that must be involved here.
All right. Okay, one last question here.
Yeah, thank you for the great presentation and the great work. Probably I'll say a question and a comment. It's a very nice work that you've done, especially considering that the countries that are involved have high prevalence of HIV. So my question is, the data that you are generating, I know probably it's not really been harmonized with other data that is being generated from other parts of the world. Considering that other countries are having similar issues, but the prevalence might not be that high, are you considering analyzing these kind of data sets, comparing them, especially at the genomic level, so that you might pick some variants that are not really in the populations that you are studying, which could be useful for therapy, something like a universal one? And then secondly, I want to commend you for the comic book that you've done, because I feel it's a very great tool to get genomics to the younger generation. And more importantly, since these are in the local languages, I got it wrong or something. Is there a way to go to other African countries like my country, Ghana? I would really love people to embrace this kind of getting genomics through the local language to the people. Thank you.
I'll take the compliment, then you'll take the question. Yeah, but thank you for noting that. In terms of the comic books, we have given rights to many different countries. So I think it's just reaching out and getting permission to translate them, and we'll work with you through that because we've gone through the process quite a few times. We did that with the UAE, well, not UAE, but Middle East, the request, as well as from Egypt as well. So we'll be happy to work with Ghana. In terms of the population, so as part of this project, which is the beauty of this, all our data is submitted in the end to the EGAs, meaning that it's accessible to all of those across the world. Obviously through all the mechanisms that have been put in place to protect the participants and the original researchers. And I think we look forward to looking at it from multiple different angles. I think there's still a lot to be mined from this data. And we already called a center of excellence. So I think the next discussion on centers of excellence, we are an easy fit into all of that. I'm looking at Jen and others. But I'll ask you to maybe comment on that.
No, I think you basically covered it. One of the things that we hope that this kind of work will show is the utility of having within the African ancestry populations multiple ethnicities, right? Because some of it is published, but you'll see that we have work where the haplotypes, for example, in the work that Sam was doing, are important in both Uganda and Botswana, although you do have things that are specific for the given countries, right? So it speaks to the utility of the potential to carry this not only in our specific populations.
All right. Well, thank you, Mogo and Gahon. Thanks so much. And as we saw with CAFGEN, for those in the room who may be new to H3Africa, and this might be your first H3Africa meeting, you will see a lot of trainees be part of our presentations, and you'll see them highlighted in the presentations. And that's a hallmark of the H3Africa initiative and the projects is having trainees work alongside the PIs and become leaders themselves on these projects. And we're talking about projects, but also multi-project, across site, across consortium collaborations. And the next speaker, Professor Nikki Mulder, her presentation is going to be about the whole H3Africa. Professor Mulder leads the Computational Biology Division at the University of Cape Town and leads H3A Bionet, which is a large Pan-African bioinformatics network of 30 institutions in 17 African countries. Nikki. And the title of her talk is Facilitating African Genomics.
Hey, so thanks, Charmaine, and welcome to everybody to Cape Town. So as Charmaine mentioned, I'm going to talk about African genomics, and also the same problem with Moko has, having to summarize what we've done in H3 Bionet in 15 minutes. But I'll try to give you the whistle-stop tour. So obviously for genomics, for health research, there are a number of requirements, and I'm talking particularly here about the data requirements. We need to have a data management infrastructure. We need to have mechanisms for tracking participant recruitment, sample data collection, data generation analysis, interpretation, and then of course the genomic skills that cover all of those different pieces. So here we're talking specifically about study metadata, clinical demographic data, genomic data, and then all other data types that sit around that. And there are many different components to the data infrastructure, from the capture to the generation, movement of data, all of which are a challenge in low resource settings. And therefore we need a lot of training. So H3 Binded has been working very hard for 10 years to address all of these challenges. So I'm going to talk about them under these four main headings of infrastructure, generation and interpretation, and then access. So for the data infrastructure, when we started several years ago, we had very little in terms of computing infrastructure on the continent. So we had to put in sort of between small and high-end machines in various different countries. We had some deals with Dell to do this and roll out. So we got up to 50% discount because we were doing this across the continent. And I think we now have these are just randomly placed, but we now have some major centers that are capable of pretty large-scale genomics analysis. And then many others who can do moderate to medium kinds of genomic analysis on site. And so we have 18 computing facilities now that have been established in 11 different countries. And we have, obviously, across those, a number of compute cores, a lot of storage available, and some data transfer infrastructure. In terms of data transfer, when we started, it was really hard. So there was one data set where we took, I think it was 3 months to get the data from the US to the continent. And so we actually did a mapping exercise in our NetMap project of looking at what are the actual speeds. So universities tell you what their speeds are, internet speeds are, but actually it's not that when you come and you actually do a check at different times of the day. So we did a mapping exercise to see what the actual speeds are. And some of our nodes have actually used that as a way to go to the universities and say, this is not good enough. And that has improved the infrastructure. But we've managed to set up infrastructure. So we looked at various different mechanisms for data transfer. And apart from shipping on hard drives, which we still do as the most effective way to move data, we now have various options like Globus Online, for example, and we facilitate transfer across the continent. Another part of data infrastructure is the standards. So making sure that data are collected in a very standardized way. And this has been done with the H3Africa Consortium in the Phenotype Harmonization Working Group, where we've built a number of case report forms, standard case report forms for collecting data, both for pediatrics and for adults, and across multiple different diseases. So there are modules for different diseases, including a COVID one, which was developed over the last couple of years. And these are all available as guidelines, as paper CRFs, or as REDCap templates. And what we hope is that going forward, so the second round of projects actually use many of these. And hopefully going forward for any African genomics, these can be form the basis for data collection to ensure standardized capture of data. Data submissions, it's really important that the data, that the value of H3Africa data is enormous. It's important that that gets out there for the global good. And so we have the archive in Cape Town, which is the data coordinating center for submission to public repositories. So the projects submit the data to the archive, and then we submit that to the public repositories, including EGA, ENA, DBSNP, ClinVar, etc. In terms of the data generation, you've heard previously in the previous talk about the H3Africa array. So we worked on developing this with Illumina, because we knew that the current arrays at the time were not really appropriate for African populations. And so we got a number of SNPs from the PIs who had particular SNPs that they were interested in, and then a number of pharmacogenomic and other clinically relevant SNPs. And this chip led to much excitement. Bill Gates tweeted about it at the time, and it's been used in many publications now. And some of the comments are that the African-centric array really enabled capture of genetic diversity that perhaps would not have been possible with other chips. We provide some resources around the array. So we have this chip info site, where you can, if you want to know if your favorite SNP is on the array and why is it on the array. And so you can search for by SNP, by region, by gene, and identify whether that is on there. And then you can also view that on the genome and see where that SNP is, where the marker is, and what other chips that marker might be on. And then, of course, we have the reference panel imputation service. So if you've run the H3Africa chip, you can then, and also links to download cluster files. But if you run the chip, you can then also run the imputation against the H3Africa reference panel. In terms of data analysis and interpretation, so we've built a number of reproducible workflows across different data types. So from imputation, if you're doing chips, imputation and GWAS analysis, if you're doing NGS, There's the alignment and variant calling, and then 16S for microbiomes. And these are all available to ensure that there's reproducible science, and these are used in our training as well. We've also developed a number of tools. So RefImpute is the tool that's for our imputation service. We are busy working on a reference graph to better represent African populations in the reference human genome. We are working on various other tools. So our node in Dakar has developed an amazing new tool for gene association for family studies. We have the WITS group who's developed the Genesis tool for visualization of data. We have tools for post-GWAS analysis. So looking at if you've got your list of SNPs, how do you interpret that? What is the clinical relevance of those? So through machine learning, through the HUMA project. And then we've also got a group in KwaZulu-Natal and Stellenbosch University who worked on pathogen genomics tools, and they were used predominantly in the South African and African response to COVID. Then the last thing to talk about in terms of the tools that we've developed is around data access. So I've told you that we do the submission to the EGA and other public repositories. But it's important, if you want to make H3Africa data fair, you've got to make it findable in the first place. And if you don't know that it's in the EGA, you don't really know where to find this. So we developed the H3Africa data and biospecimen catalog. And this is for you to search the metadata around H3Africa projects. Once you've logged in and you've got your search results and you've chosen the data sets or the samples that you want access to, you can then submit an online request form that goes directly to the Data and Biospecimen Access Committee. And then they do their deliberations, and then it all goes back into the catalog and gets reported. So we can really easily track who submitted requests for data, when their progress report is due, etc. And it's a way to really see what are the outputs of H3Africa. So that's the search there. And then we're also trying to work a way to see if we can summarize the data nicely. So you can, at the moment, you can search by individual projects or by country. So you can say, I want all samples from Nigeria who have diabetes information. But these summaries are now to show, well, overall, these are the number of females, these are the number of males, these are the number of exomes, these are the number of chip data, and then the different phenotypes that are being collected. Now, not all the phenotypes are in EGA, but then at least you can follow up with the projects if there are additional phenotypes that you want to study. Then we're also making the H3Africa data accessible in aggregate form. So when you download the data from EGA, you have to go and download each individual data set and then do your own analysis. Now, there's much greater value from having that sort of all put together. And so one of the ways we're doing that is the African Genome Variation Database, and that's putting together the genotype data from sequencing and from arrays and creating and sharing out the maps, the frequency information. Now, you know that you can go to Nomad and get that, but if you go to Nomad, it's just African. And we believe that, as you probably will have seen and know by now, that African is it's not one country. There is enormous diversity. There's enormous variation in frequencies that is very clinically relevant. So if you just say African, you're losing a huge amount of information. So what we're trying to do in AGVD is make sure that this data is available at a granular level. So you can see for East, North, South, West, Central Africa, what is the frequency? And then once you've logged in, then you can actually go to a more granular level and get them at maybe the country level, or if the sample size is big enough at the particular ethno-linguistic group level. And so this is being developed. The beta version is out, and so it will be released soon. We also have a beacon of this data. So if you want to, so a beacon is where you can just say, have you seen this allele, an A at this position, yes or no? And then we can see that if that's available in the H3Africa data. And so the H3Africa data is one of the lit beacons we have. And then we're working on a tool to filter by frequency for rare diseases, for example. So if you sequence a patient and you want to filter your variants to narrow down which is the potential cause of variant, you can run your basically your list of variants, and it filters them and gives you a ranked list based on the frequency in the particular population that you're interested in. So that's an API that we're working on. We also have, those are the H3Africa data sets. For other African data, when we try to find, you go to Farm GKB, for example, you try to find information about African variants. Africa is not one country again. So it's really, and yet it's all represented as one country. And it's really hard to find the data because, you know, North Africa would be sometimes lumped with the Middle East. And so it's really just difficult. So what we've done is created the African Genomic Medicine Portal, where we've taken data from Farm GKB, DisGeneNet, and we're busy adding the GWAS catalog and curating the data better from African populations. So we actually taken the publications and finding out where in Africa those publications came from. So you can search by disease, drug, variant, or gene. In this case, we searched by drug. This is efavirenz, and then you get the RSIDs associated with differences in response to efavirenz, which is very different in different populations. And then you can see that we actually show the country in which the study was done. So it's not just Africa; it actually shows you the African country in which it was done. And then this is for disease associations. Again, you can see the country, and we also add the p-value. So we take additional data from the publications. The other portal we've created is for microbiomes. So looking at what studies have been done on African microbiomes in different parts of the continent, but also in the world. There's a summary of all the studies by site, by disease, by technology. And then you can search for these, and for each study, it gives you information about the study, where the study was done, what country it was done in, and where you can find the data. So the other thing we've been trying to do is make H3Africa and H3Bind internationally relevant. So we heard from Mojo that it was used in the COVID response, and this was the same for H3Bind. Many of our PIs were involved in the COVID response, particularly in South Africa. We've leveraged H3Bind in terms of the skills and the infrastructure that's been developed to now run the DSR Africa Open Data Science platform. We're being used extensively by the Africa Biogenome Project. They're leaning on us quite a lot to do analysis and have the data infrastructure. And we've been working with international projects, including the Human Pangenome Reference Consortium, to make our African reference graph more relevant. We are driver project for GA4GH, so we've been working very closely with GA4GH on use cases and using their standards. And then through the Seneca project, we are looking at federated data analysis. And then, as I've mentioned before, another important part is obviously training the genomics workforce, in this case, particularly the data side. We train a number of different audiences. So we know that the workforce is not just about training bioinformaticians. We have to train systems administrators. We have to train users. We have to train clinicians, engineers, scientists, everybody. And everybody needs a different topic area at a different level. So we try to create a training ecosystem that tries to address all of these different components. And we do this by different modes, so face-to-face training, internships, train the trainer to build a sustainable training environment. And then we do particularly objective-driven training through hackathons. And through this, we've also built a number of communities and resources. So we have an amazing training pack, which is everything from how you design the course, every email you need to send, how you do the registrations, and how you do the evaluations. And so I think that's a valuable output of the consortium. So just going back to the original list of all those different requirements for genomics, what do you need to implement genomics? And I think we've managed to tick many of those boxes. So maybe not everything is completely resolved, and we need sustainability for all of these. But I think we've managed to address most of these to a pretty reasonable extent. So what does the future hold? Well, we need to have readiness to respond to new, exciting challenges. We need to sustain and increase the value of what we have. So we need to maintain these African genomics resources that we've built. We need to make sure that they're still there when H3Bind is no longer funded. We want to improve the value and accessibility. H3Africa has up to 100,000 participants. That's 100,000 people for which there's some sort of genomic data and some sort of phenotype data. Imagine if that was all put together in a very standardized way, harmonized, that would be the most incredible resource to the to Africa and to the world. And I think there's the many things that we've done in this that can help to achieve that. And we need to continue to build capacity. Whenever we say this is the last time we're running AGMT, the next day we get an email, when are you running it again? We desperately need the course. And every year we run the introduction to bioinformatics, and the next year we still have another 3,000 applications. The demand is not going away, so we need to continue and sustain that. And then there are many fantastically exciting new initiatives coming along. African Population Cohorts Consortium, DSI Africa, EU Africa, PERMED, GENCO, this network of centers of excellence. And all of these are going to need better data science. We're going to need to think about federated analysis, and we need to think about translational science. So we really need to be poised and take what we've done and get ready for these new exciting challenges. So I'd just like to acknowledge the entire H3Africa Consortium. Everyone plays a role in contributing all of these resources, particularly our work package and project chairs, the team at CBio who are amazing, the funding from the NIH, without which this would not be possible. A wonderful scientific advisory board who's been there to advise us over between 5 and 10 years, and our grant officers. And I think H3Africa really is the example of going far by going together. Thank you.
Thank you, Nikki. Thank you for it. Can I? Okay. Thanks so much. We will take questions. Phenomenal resource there in H3Africa. Any questions for Nikki?
Thanks, Nikki, for the great presentation. And I just want to add that, yes, AGMT, everybody asks about it when it's running again. Also the AGMP. Till yesterday, when we were in the meeting, everybody from all over Africa was asking, can we join? And I was just want the opportunity to please extend the deadline for candidate joining. And at the end of this, I would like myself to thank you very much. Thank you, the whole H3 BioNet Consortium and H3Africa Consortium for letting me this chance to be part of this great scientific and fine scientific community. Thank you.
Thanks.
Thanks, Nikki. And I think that it's such a powerful resource already, but I just want to make the point to all our H3Africa PIs that the more data we put in, the more powerful it gets. For discovery, for use by everybody, including all of you. So just a reminder that if you're not all the way there yet, keep working with Nikki to make sure that you get there. But also, I think thinking about all of this and what you've done with what we heard from CAFGEN, I'm just struck by, so 10, 15 years ago, we were doing these GWAS for HIV in European populations with many, many more people and not getting the signals you're seeing. And so the idea that there's so much more to be found here because of the diversity, because of the structure of the genomes. So much to come in the future. So I don't have a question. That's just my comment.
Yeah, I think what it shows and what Moko has shown is that the African genomes are not just relevant for Africa. I think they can actually improve our understanding of diseases globally because you can get to that fine mapping. Yeah.
Yeah, absolutely. And the resource, and Nikki, you talked a lot about Africa as a continent versus Africa as a country and changing that mindset.
Yes.
I think H3 BioNet and H3 Africa as a whole does that very well. Another question?
Yeah, I'm Dominic from Ghana. That's a very wonderful work. I'm really, really excited because it brings together all what we've been waiting for of different population across the continent, different data sets, and how we can kind of analyze it together and see the bigger picture. My question is, I know you are part of the HCA advisory committee. So I wanted to know how
this fits in because we are done with the Human Genome Project, and now we are moving into the Human Cell Atlas project. So how does it fit in? And then secondly, what are the ethics around access to some of these data sets? Thank you.
So the HCA, I think, is the next step. So one of the future things is really to go to the other omics now and to look at, I think they've also just shown the difference in transcriptome, in the population-specific differences in transcriptome. So I think better participation of African samples in HCA, again, I think is going to increase the knowledge. There's a huge gap in terms of our actual knowledge about the protein expression, gene expression in that is population specific and that is relevant to diseases. So I think that's where Africa can contribute significantly to the HCA. And I think it's really important to take that next step to the next omics. Genomics gives you what exists, but actually everything else can be impacted by environment, by many other factors. So I think that's a very important next step. In terms of the ethics, so that's why we have, so all the public data is completely openly available. The aggregate data, so we have agreements with the individual PIs who've submitted their data that have checked that it's all within the consent of the original participants, that it's okay to share this data. And anything that is unclear will then go to one of the ethics boards just to clarify. But we would never show individual level data in that. And even when deciding what population clusters to use, for example, we're looking at what can't be too fine grained that might cause, if possible, for stigma or any sort of discrimination. And so we're quite careful in terms of how we display the data and who gets access to what kind of level in terms of individual level data. And even any individual level data would go through an access committee.
Okay. In your program, it says our next speaker is Ambrose Wancombe, but Ambrose's talk will be tomorrow and it will be virtual. So we're going to move to the next speaker, who's Mayowa Olabi, and who is a professor of neurology and the Dean, Faculty of Clinical Sciences, University of Ibadan, Nigeria. And his talk is titled Translating Novel Insights into Interventions Against Stroke and Cardiovascular Diseases in Africa.
Thank you very much for this great opportunity. Africa is like the global capital, not only of stroke, but of cardiovascular diseases. Africa, as far back as 2014, had the highest incidence of stroke globally, and every minute 6 Africans develop a stroke. Also has the highest prevalence of about 14.6 per 1,000, and this actually also has some environmental undertone. That's in the Niger Delta region of Nigeria, where there is air pollution, gas flaring. And then highest case fatality, 84% in 3 years, and the burden is increasing. Actually, I am the lead co-chair of the World Stroke Organization Lancet Commission on Stroke, and we did a 30-year projection of stroke burden across the globe up till 2050, and the burden is actually still increasing in terms of numbers. So we proposed the stroke quadrangle to tackle this. Epidemiological surveillance, prevention, acute care, rehabilitation. But the key driver of all this is actually research, which gives us information about the determinants, and that can then be used for prevention, for acute care, and for rehabilitation. So Siren came on board, funded by H3Africa, extremely grateful for this, to unravel the genomic and environmental determinants of stroke in Africa, using the tools of Phenomics, Genomics, and bioinformatics. And this is actually the core, these are the core members of the Siren team. This was in Washington, where we had a presentation at the NIH campus. And we had to develop, because stroke is a very, very complex trait and the culmination of a lot of pathways, including intermediate phenotypes. So a very rigorous algorithm for accurate phenotyping, which is very important before any discovery or association, meaningful association can be made. And another thing we needed to do was to ensure that the phenotyping was done in a manner that is reliable and valid across sites. So we needed to develop a software which actually got a patent, actually the first of its kind anywhere in the world, for phenotyping both hemorrhagic as well as ischemic stroke in a reliable way. Another huge problem we needed to solve is that of biobanking in a very, very challenging environment where it's actually unusual to have power. And so we had to rely on alternative sources. And we had to invest heavily in our biorepository facilities, which actually tripled the cost of storage of samples. We have more than 160,000 samples in our biorepository. And we also had to do a lot of community engagement, ethical, legal, social implication, and so on. And you can see we had quite a number of publications on how our samples are collected, how we interact with the communities. And then using the information that we have so far, we were able to generate, we already mentioned the patent, 2 Bruce Schoenberg awards for neuroepidemiology by the American Academy of Neurology, 4 Paul Dudley White awards by the International Stroke Conference, about 100 publications, more than 40 presentations. And then this landmark publication, which unraveled the dominant modifiable risk factors for stroke in West Africa, hypertension being the lead, and dyslipidemia, which was previously thought not to be common, being actually the second most common risk factor for stroke, mostly undiagnosed, undetected. Then the others that we knew before, diabetes mellitus, cigarette smoking. And then surprisingly, we found that and we've said this over and over several times, and we are trying to explore this. This was actually pointed out by the reviewer in Lancet, that the confidence intervals were non-overlapping. Dose response relationship between daily consumption of green leafy vegetables, protective effect against stroke occurrence. We also looked at risk factors for ischemic stroke. I don't have that here. We also looked at risk factor for hemorrhagic stroke. These also got an editorial, just like our Lancet publication got an editorial, and those are the risk factors. And we looked at so many other risk factors. We zoomed in on alcohol, on many others. I'll just give some examples. This is about secondhand smoking. And then we did not only look at the risk factors for the occurrence of stroke, but we also look at determinants for severity of stroke. And again, pleasantly surprised, we find that consumption of vegetable was protective, meaning low vegetable consumption was actually a very, very strong risk factor for development of severe stroke. Meat consumption was also a risk factor. And then lesion volume in one of the regression models was also a risk factor for severe stroke. And then this is just hot off the press, just published February 16. Again, this is in Lancet. It also attracted an editorial. And this is looking at the risk factors for, or the determinants of stroke fatality in the prospective cohort of stroke patients across the Saharan sites, 16 Saharan sites. Again, the determinants, the lead ones, low consumption of green leafy vegetable, blood pressure, systolic blood pressure, and of course, lesion volume, and then aspiration pneumonia, and raising intracranial pressure. Those were the things that determines whether a patient is going to die or not. So, and this is something that we can tackle or target to reduce the fatality. We already mentioned that the highest fatality from stroke is actually in Africa, 84% in 3 years. And we found some of the factors that can be tackled to reduce that. Well, the NCD cascade or the stroke cascade is very complex. And many of the factors we've been talking about are actually intermediate risk factors like hypertension, dyslipidemia, and all that. There are other drivers like non-modifiable risk factors, age, sex, genes, socio-cultural factors, behavioral factors, which are very important. And the great thing about tackling the intermediate risk factors is that we will not only be controlling stroke, but also preventing other cardiovascular diseases. Social determinants are extremely important. As you probably would have seen, dietary factors, stress, which are heavily influenced by social and political factors and economic factors, are really very strong and potent factors. Now, talking about genetics, which is really spinning a new angle into the mix in terms of the drivers of the unique burden of stroke on the African continent. It's been said that a lot has been said about this. So inclusion of African ancestry samples actually gives a lot of opportunity for discovery, even with relatively smaller sample sizes. It's been said that adding just 1,000 African ancestry individuals could even give greater discovery than 10,000 European cohorts. So, well, we did some candidate gene analysis, and we were the first to find the association between apolipoprotein L1 and lacuna stroke, IL-6 and stroke. And then in the first ever GWAS of stroke on the continent of Africa, with the samples that we have already genotyped, 1,683 ischemic, 1,738 stroke-free controls. We used TopMed imputation panel, performed best. We also used Sanga. We also used H3Africa Biobanking, but TopMed performed best. We were able to find 2 SNPs consistently. I mean, we repeated the analysis almost over more than a year, several times, at genome-wide significance level with a lot of functional implications. And we found 30 additional at suggestive significance of 10 raised to the power -6. There is no other indigenous African data for replication, which could have been the ideal replication cohort. These were the models we used, the logistic regression models, and 4 of the models actually produced 2 consistent SNPs at genome-wide significance level. We had to do some meta-analysis comparing with COMPASS, comparing with Megastroke, and you see the scatter plot showing that about 90% of our variants that we reported were also found showing the same direction of effect in COMPASS. Even though COMPASS is an admixed population of African Americans, and you can see its comparison with Megastroke, as well as the relationship between Siren and Megastroke. So there was no ideal replication cohort. Well, the functional implications of our findings, the 2 major SNPs that we found are protective, microRNA regulatory, and then there was significant interaction when we did very, very rigorous in silico functional analysis, transcriptomics, and all of that. Significant interaction with multiple loci, with expression in brain, vascular, cardiac, and neuronal tissue. And this means that we need to go further and do some multi-omics. We also participated in the largest study so far of stroke genetics, the Geiger stroke, and 89 SNPs. We will see that not all of them really, in fact, only a few of them had cross-ancestry representation, meaning that unique data and unique analysis is required for unraveling the unique determinants of stroke on the African continent. We participated in the polygenic risk score, and the polygenic risk score developed using the European data sets did not perform very well in Africa. And this is similar to what has been reported in other NCDs on the continent. Intracerebral hemorrhage, we haven't done that yet. We haven't even genotyped our samples, but it's a unique opportunity because hemorrhagic stroke is 4 times more common in Africans than elsewhere, accounts for 50% of stroke deaths. And there's early age of onset. And so genetics really have a lot of role to play And this is something we intend to explore. Now, in trying to understand stroke, we know that's not possible without understanding cardiovascular diseases. And in 2019, we will see that cardiovascular diseases has overtaken infectious conditions, which Africa is usually known for, as the leading cause of death on the continent, and that includes stroke. So during the course of the H3Africa project, which is still ongoing, we set up the SHARE Consortium, over 55,000 subjects, to explore hypertension, DM, and all that. I pioneered that along with several other PIs who are in this audience. We developed the database. We add this publication on the data resource profile, and then we describe the regional patterns of hypertension and obesity across the continent, as well as we found that obesity doubles the risk of hypertension on the continent, which is very important. So if that's tackled, also that helps to tackle hypertension, that helps to prevent the stroke. And then using that data set, we explored the relationship of vegetable to its association with hypertension. And we found that indigenous Africans who consumed at least 12 servings of vegetables per week were significantly less likely to be found hypertensive, particularly among males and young adults. This is published in the European Journal of Preventive Cardiology. What are the emerging solutions? Well, we tried alongside, we had a very, very strong community engagement, 27 FGDs, 168 subjects across different sites, community advisory boards, 8 of them, more than 6,000 subjects were surveyed in terms of community engagement and their perspectives about stroke. We did cardiovascular screening for more than 15,000 subjects, and we also tried to explore people's willingness to undergo genetic testing for stroke. And we found that those who had a family history were more likely to be interested in that. And then this is just recently published in January. We did focus group discussions, qualitative study, to find out what people think about releasing genetic results about stroke, genetic testing for stroke, and what they feel. Well, they feel that it should be released to them through professionals, relatives, or religious leaders. And the key facilitating factors for wanting disclosure is those who are educated, have an enabling environment, and disclosure inhibitors are inadequate information, fear of marital breakup, family displacement, fear of stigmatization, and so on and so forth. So our communities are already yearning for point-of-care device or tests to foretell the quantitative risk of stroke, combining genomic and environmental risk factors. We were pleasantly surprised that in the communities where we went to, they were asking us to give them the quantitative on the spot, their quantitative risk of developing stroke in the future. And we told them it's still in the works. So, and we've been working with policymakers. We developed this stroke prevention manual and bill, working with legislators. And we try to convert what we found so far into impact. So we had a cartoon based on the dominant risk factors for stroke on the continent of Africa to be used as a digital tool for controlling stroke. And in addition to that, we also developed the first ever Afrocentric stroke risk prediction model, a riskometer. We developed that actually into an app. Combining these two tools, which came out from the findings of the determinants of stroke, we actually already did a pilot trial, which is very, very positive, that show that there is a significant reduction in the overall risk of stroke for those who are exposed, the intervention group who are exposed to a combination of the individualized stroke risk score, as well as the stroke educational video. This is under review. And then we try to see how we can change the landscape for stroke on the continent. And this is in Nature Reviews Neurology. We looked at the profile, progress, prospects, and priorities for stroke in Africa. And we established we established the African Stroke Organization. And we've had two very, very well-attended, world-class African Stroke Organization conference, and we're going to have a third one this year, and it's going to continue. We also got support of NIH actually for those conferences. Then on the global front, we lead the WHO Lancet Commission on Stroke. And this is remarkable. We've been working on this and we had a lot of publications on this already. But this is remarkable because our report is finally ready. It should be ready latest by April. We're working with the editor to finalize it. And it's going to be launched in a grand event as part of the bicentennial 200-year anniversary of the Lancet series. And then this is going to be publicized, disseminated across the globe through the ecosystem for implementation for reducing the burden of stroke, through prevention, through acute care, through improvement of surveillance, and through rehabilitation. And then we have this African rigorous innovative stroke epidemiological surveillance study, the ARISIS. We set up the stroke information surveillance system. We already have recruited over 100,000 urban and rural dwellers, and we're going to follow them for as long as possible to determine the incidence of stroke, but also we'll spread this to other vascular risk factors and other NCDs. Our university has a long-term relationship with the rural sites that spans over 5 decades. And Professor Charles Rotimi has worked in those sites, the Barakwa sites as well, for diabetes and for other things. So this is for primary. This is one of the publications of, in Lancet from the Lancet Commission, Primary Stroke Prevention Worldwide Translating Evidence into Action. And we're using this implementation science model to ensure that we are actually able to tame the burden of stroke, not only in Africa, but also globally. And this is the ecosystem, multi-level ecosystem that is very engaging, that is going to help achieve that goal. And we are also leading the call for action against hypertension in Africa through the Achieve ecosystem. I don't have sufficient time to talk about this. We have a publication in hypertension about this already. And then the neurology revolution, which we are championing, not only in Africa, but also globally through the Brain Health Initiative. We've looked at barriers for implementation of the WHO IGAP, and this is also published in Nature Reviews Neurology. So optimizing brain health, we're leading the WHO IGAP, that's Intersectoral Global Action for Domestication Plan for 47 countries in Africa. We are preparing, the report is almost ready. This is about brain health, and our work has not been unnoticed, and this was a pleasant surprise that competing with all researchers, stroke researchers across the globe in 2021, we got this global award for outstanding contributions to clinical stroke research. And this was at the Washington meeting. And what are the things we need to do differently to move forward in terms of the next chapter for African genomics? We need to improve our capacity for phenotyping, genotyping, sequencing on the continent, biorepository to be expanded, collect more data, bioinformatics, precision medicine, transomics, and of course, big data, both Afrocentric and transcentric transomics. The ICH, that's intracerebral hemorrhage, we mentioned that, drug target discovery, combined polygenic and clinical riskometers. We already have the clinical riskometer, but combining that with polygenic to really be very powerful. And then the African biobanks and longitudinal epidemiological ecosystem, which we are exploring through a quadruple helix approach involving industry, academia, policymakers, and of course the community, to completely change the landscape of understanding health, understanding disease, and improving the health, wellness, and well-being of Africans. We have a large team, and we're extremely grateful to all our funders, particularly NIH. And we have the Talents Program also, which is a mentorship program also funded by the NIH D43. And we are taking the second round of mentees this year. I gave this presentation on behalf of myself and one of my mentees who is not physically around, but I have two of my other mentees who are giving independent presentations during the course of this program. Thank you so very much.
Thank you, Mayowa. Fabulous work. We have time for one question. There's somebody over here with a question. Where is our mic? All right. Great work combining your basic science discovery work with studies on the ethical and social implications and legal implications. I love that paper on the legal implications of stroke. Great integrated work.
Thank you very much for a wonderful presentation. I have a question about the prediction for stroke or for other components of the metabolic disease. The app that you mentioned, did I get it right that this app will predict on base of the inputs how susceptible the person is for stroke or other metabolic diseases, and whether these inputs were a population dependence, for example? So does it work for different population from Africa? And for the part of obesity, does this app include also some of the metabolic indicators, because to differentiate between healthy, metabolically healthy obese versus metabolically unhealthy for for the prevalence of inflammatory factors and these stuff? Thank you very much.
Thank you very much. That's a very loaded question with multiple prongs. Okay, first of all, how did we develop the app? We used the data that we used for the Lancet publication. That was what we used to develop the app. We weighted the different risk factors for stroke. So it's a yes, no. I actually have the app on my phone, but because we're using it for clinical trials, so it's not public yet. That's why, although to avoid contamination of the control group. But I actually have it on my phone, and you only need to click yes, no. When in 1 or 2 minutes, it tells you your overall risk of stroke in percentages. Your likelihood of developing stroke, maybe it's 50%, 70%, whatever it is. Okay, so that's for stroke. But interestingly, using this same model, we also have developed something which is under review in Hypertension Journal. Actually, we got a favorable review. We're trying to finalize and send back to them, responding to the comments in Hypertension Journal. We also developed an Afrocentric model for predicting the occurrence of hypertension, which if finally that's published, we can also use that for an app. So, but that has not been done yet. Okay. Now for metabolic syndrome, we also have publications on metabolic syndrome and stroke, which of course I don't have the opportunity to present because of the short time. So for the app, you just need to input yes, no for each of the, and then it just tells you, it just pops up, tells you your risk of stroke. And this is something you can then work on, to see whether you can bring it down. If it's 70%, you can work with your clinician to see if you can bring it down to 50%, to 30%, and things like that. And it could be very motivating because we tell you all the implications. And why this is important, because we also found it has a collateral benefit in terms of reducing controlling hypertension. The intervention group had 11 millimeters of mercury reduction, in their blood pressure over a 2-month period compared to the control group, which had only about 4 point something. So it's a very powerful tool, which can be used not only to prevent stroke, but also to control all the cardiovascular risk factors which are related. Thank you. Okay, now other populations, yes. What we plan to do is that, we developed this from what we gathered from Nigeria, from Ghana. We plan to test that model using the share data set across other 12 other African countries, using the data set, part of which we've been using for our publications, including the one on relationship between vegetables and stroke. So we're going to test that and see what's the predictive value in other African populations. Thank you very much.
Thank you so much. Thanks so much, Mayowa. So our next pair of speakers will be speaking on H3Africa Consortium Genome Analysis, Bridging the Genomics Gap. And we're going to have Neil Hanchard and Ananya Chowdhury who are going to speak. Dr. Hanchard is a clinician investigator at the National Human Genome Research Institute of the National Institutes of Health in Bethesda, Maryland. And Dr. Ananya Chowdhury is a senior scientist at the Sidney Brenner Institute of Molecular Bioscience, University of Witwatersrand, Johannesburg. Neil and Ananya.
So thank you very much to the organizers for allowing us to, again, talk a little bit about the work we've been doing in the genome analysis group, and then trying to walk you through the sort of where we've been, where we are, and where we hope to go with time. And so the genome analysis group had the sort of primary aims of sort of facilitating cross-consortium analyses among the many projects that were occurring in H3Africa. And then utilizing that data to try and maximize it, to ensure that it is being sort of used to its full utility within the consortium. And then using that then to sort of extend the H3Africa brand through collaborations, through grants, and then ultimately through training of other individuals who would continue to populate it. And so this was initially run by Debo Adiemo and Zane Lombard, who were the first two chairs. And then over the last 2 or 3 years, myself and Ananya, who you hear from soon, have been the chairs. And so the real genesis of one of the major projects was that looking across the many projects that are occurring in H3Africa, and trying to facilitate a number of diseases that were being evaluated, everything from sort of non-infectious diseases to infectious diseases and everything in between, and thinking about the sort of number of countries and the number of ethno-linguistic groups that were part of this, it was felt that there needed to be an effort to try and have a cross-consortium analysis that would facilitate this. And at the time when H3Africa started, this was kind of the landscape of genome sequencing across the continent. And it's sort of centered upon Nigeria, where the Yoruba and the Igbo people had, for many unforeseen reasons, become sort of proxies for Africa and West Africa, somewhat unfortunately. And so knowing, of course, that the African genomes are the sort of deepest tree genomes, and there's incredible diversity among populations that have been living on this continent for thousands of years. And also recognizing that it was sort of well known at that time that the amount of genetic variation in a genome is more in African populations than not. But that these very populations are not sort of isolates. There's well known that there's sort of migration, introgression, and admixture that occurs across them. There was seen as a need to sort of expand the landscape in order to facilitate the subsequent studies. And so the PIs of H3Africa were asked to contribute appropriately consented samples for whole genome sequencing. And that, after a long time, kind of looked more like this. So the red dots represent those genomes, populations that were sequenced at a high coverage, and the green a sort of a lower coverage through the trypanogen group. But ultimately, we ended up with a group of 426 individuals, now spanning 13 countries and 50 different ethno-linguistic groups. So we're sort of filling in the gaps somewhat. One of the major efforts you've heard about previously, which is to create a genotyping chip that could be used across the consortium. And Nikki's talked about the sort of genesis of that. Suffice it to say, it did include many hours of people sitting together in a room to do these analyses. But since that time, it's been used by more than 90,000 individuals. This is all courtesy of Illumina and Javier David. And about 2/3 of those are on the continent, but another 1/3 are around the world. So it's being used quite widely. And the other part of what the genome analysis group had sort of leveraged with the chip development was the placement of an iScan, so a genotyping machine in South Africa at the initial launch of this. But then subsequently, so if we fast forward to 2022, you can see that now the number of machines across the continent has massively increased, and that's sort of able to facilitate additional studies. So we wanted to also analyze this data in a broader sense and sort of understand more about the populations. And so Nikki Mulder locked us in a room for a couple of days, but she was kind enough to join us and bring the key, which is also important. And through some of the subsequent analyses, we're able to sort of demonstrate really the sort of rich tapestry and complex tapestry of genetic variation across the continent. And you can see that in the admixture plot on the bottom, where again, each line is a different individual and each color represents their sort of parental ancestry. And as you move across the continent, going from west to central to east to southern Africa, you can see that there's this sort of tapestry of inter-admixture between the various groups. Along with these analyses, we're also able to demonstrate 3 million new variants that had not been previously described or not present in the existing databases of the time. And you can see that those variants had a great deal of variation between each population that we looked at, such that, for instance, if you look at Botswana, where on average we found about 12,000 new variants, the Botswana population has a strong admixture with the Khoi and San hunter-gatherer groups. That's the pink bar that's there. And in fact, we're able to show that if you plot the number of novel variants as a function of the amount of Khoi and San hunter-gatherer ancestry, you see this almost perfect correlation. And this really demonstrated to us that as you start to go into these populations that are not well represented in the existing databases, that you start to see that's where you get all the maximal amount of sort of novel variation and new information. And in fact, when we looked, if we started from Botswana and then asked with each additional population, how much new information do we gain? We really didn't reach a plateau. So this is just common variants to begin with, but even over those last two populations, you can see we're still seeing 6,000 novel variants. And so it's clear to us that as we go into more of those populations, we're going to see more variation, and that's a major push of what we want to go through forward. So we published this information in Nature and had a cover that accompanied it. This was presented in front of the 8,000 strong at the American Society of Human Genetics meeting in Houston, and then led to a real outpouring of a lot of press. And so we were very busy with interviews for some time after that. And it just sort of really showed that this was data that people were really interested in, people really wanted to see more and more of. And so it was now, that was then the sort of genesis for the next phase, is to taking some of the very goodwill that had been developed through that and doing additional analyses. So we developed teams to continue doing new work, some on the existing data and then some on new data. So this is courtesy of Ezekiel Adiebi in Nigeria, where he's working with Melissa Gimrick at the UCSD and showing that if we take the same data and start to look at repeat expansions, they're able to show sort of, they were first able to develop a new method called Ensemble TR that was able to integrate these triplet repeats. They found dozens of novel, again, population specific expansions. You can see the H3Africa populations again with having most of these. And in fact showed that there are some of these variants that are expanded in African populations, but not in European populations. And they actually developed a phased haplotype panel that can be used to impute these triplet repeats into the existing SNP genotyping panels. And so that data is going to be on bioRxiv very soon, Ezekiel tells me. In addition, we started to look at structural variation. Another team led through Scott Hazelhurst and Zene Lombard, showing that there are thousands of structural variants across these African genomes, on average, something about like 5,000 structural variants per person. And many of these impact coding genes. So these are going to be things that are going to be clinically relevant for the groups on the continent. We've also started to look at the HLA region. So given the sort of core central importance of immunity to the continent, but the complexity of the most complex region of the genome, and having then to integrate multiple different pathways of software to look at this. But this is work that's been started by the HLA group, and even just trialing the pathway, the software on existing 1,000 genomes data, you can still see again that even just in the class 1 region that we're seeing lots of variants that are unique to the African population. And so kind of going forward, this is kind of the efforts that we want to build for in terms of cross-consortium analyses. So we're very interested in sort of building out the transcriptome landscape across the continent. So for instance, there is whole blood RNA-seq data that's been generated through the Tripanogen and the CAFGen groups. And alongside these, there are genotypes that accompany them. But even if you look in the public databases, we know that there is RNA-seq data from whole blood that's been already generated. And our hope is that we can get to a point where we're able to not only expand that transcriptome landscape, but integrate it with genomes so that we can understand the sort of genome transcriptome regulation. And this is kind of an ongoing work and plans and hopes and aspirations for where we're going to go in the future. So all of this, again, a big part of this was to sort of leverage collaboration to be part of this. I've mentioned a couple of those. You'll hear in a second from Bamidele Tayo about the Cardinal project, which is, again, another effort to try and do cross-consortium analyses, and the Genome Analysis Group participated in the funded grant application that came from that. We're also helped to contribute to the DSI Africa, some DSI Africa applications, including the coordinating center. And then Shegun Fatmo had a application to look at blood traits across the populations in Africa. And then additional studies to try and say, look at cancer across the same groups and populations. And then this is all underpinned with efforts, again, to work with the reference genome and Nikki's group in the BioNet to sort of, again, fill out the missing gaps in the genome. But even outside of H3Africa, you'll hear some more about Genome Tunisia, which is another project that we have been trying to support as well. We've had interactions with the ClinGen group as part of their general interactions with the consortium. We had some early interactions with the 54 gene, looking at population genetics and submissions that went to H3Africa as a consequence. Sorry, it went to the American Society of Human Genetics as a consequence. And then more recently, we've had lots of input from individual researchers who want to work with the genome analysis group to understand more about their particular populations and the things that they're up to, including, for instance, the Brazil Genome Project, where they're trying to understand the African ancestry that they have in their populations that resulted from the slave trade. And so if this slide looks busy, that's because we like to think we have been, and that we're hopeful that we can sort of leverage these things as we move forward. And so to sort of give you an idea of what we're going to do and how going forward is going to look, I'm going to ask Ananyo to come up and talk about the next phase of H3Africa.
So this is, sorry, not for me, yeah. So this is the representation of African genomic data sets or whole genomes that was kind of compiled in 2021. And you can see there is still major gaps. And so this is a map of Africa where we have kind of shown its real size, and you can clearly see that you can fit in almost most of the continents inside. And still the representation of African diversity is, I mean, there are so many huge gaps there. And I think Zene and Ida wrote this wonderful article. So, and I mean, unlike other continents, geography is only one of the dimensions of African diversity. We know there are at least 5 major ethno-linguistic groups, but then there are uncategorized populations, diaspora populations, and unique populations living in African islands. So 90% of the current public genomes only represent one of these groups. So there is a huge gap. So this is the representation of different ethno-linguistic groups or divisions at the continent level, and this zoomed in version shows the same for just one country. So you can see the importance of nuanced sampling strategy to really capture the full spectrum of African genetic diversity. So when we were wondering about the next phase of the whole genome sequencing study, we not only wanted to cover the underrepresented geographic region, but also underrepresented ethno-linguistic divisions. And we are thankful to NIH and Illumina, NIH for funding us and Illumina for being our research partners. So we are planning to go over 1,000 genomes in the next phase of sequencing. So one of the key questions was that which groups do we sequence and where do we sample them from? And we know that we had this network of H3A projects across the continent, but then as you might remember from Zene's map, that H3A still has a lot of gaps in the African continent. So therefore we thought we should go with like a hybrid approach. So we formed a new set of partnerships, and this map shows you the countries and the country level PIs of this new partnership, which we are calling Agenda or Assessing Genomic Diversity in Africa. So you will see a lot of familiar H3Africa faces, BioNet faces, but also some people who are not a part of the consortium. And to kind of form the basis of this partnership, we had to really kind of dig down into the exact steps and the logistics of how we will do things. I'm not going into details, but this meant dealing with ethical and regulatory framework of 10 African countries, and which were as diverse as African genomes. So we had a very hard time sorting this out, but we are almost there with this paperwork. And as you can see, there was a lot of paperwork. We would especially like to thank Harriet and Furahini, who really helped us navigate this difficult space. So, I mean, our timeline, we are very close to where we are really going for collection and shipping up the samples. And by the end of the year, we want to have the sequencing completed and then move to the next phase of the study. So watch the space. So coming back to H3Africa, I think most of us, as this project was kind of reaching its conclusion, were making plans or reinventing ourselves for this sunset. I mean, how would you survive this, right? So I think the genome analysis working group was also thinking about it, and we thought we would try to have a new name, a new identity, but we would still carry the same legacy and the core vision of empowering African genetic diversity. So, So that's what, so let me introduce to our new name or new avatar, which we are calling Network for Genome Analysis in Africa, or Ngena, which in Zulu means enter. So this is our attempt to enter the next phase of genome analysis in Africa. So we envision ourselves that we would like to be a platform for facilitating or connecting African researchers, but also be an interface between African genomics research and global genomics research. We would love to scaffold generation of new genomics and omics data and kind of take up a few flagship projects to do this, and also kind of contribute to generation of resources for human genetics or human genomics based on these data sets. So, why am I not able to? Oh, sorry. So, Nikki, you stole our thunder, but yeah, these are the partners who have been along for the genome analysis working group, and we thank each one of you. And we really want to have all of you as well as new partners when we go into the next phase. So we really want to go far and go together. Thank you.
Thank you, Ananya and Neil. Fabulous work. I'm so happy to see the collaboration with Genome Tunisia. We start moving further north. All right, questions? Philip, where's our mic? They're coming. They're coming. We want to make sure we all hear you, Philip.
Great presentations, Ananya and Neil. Future directions, will that potentially include long-read sequencing technologies, involvement in, say, other activities like telomere consortium, try to fill in some of those missing gaps, potentially with regard to the genome size, structural variants, et cetera?
Yeah, so I think that we didn't emphasize this, but it is a part of the portfolio in that we have had sort of interactions with the Pangenome group. I think that Nikki's group in Bionet has kind of been the leaders in terms of pushing that side of things forward. But we've continued to have interactions to try and sort of leverage that and be a part of that, because we see that as being an important part of the next step as well. So I think there's sort of a lot more cross-pollination than perhaps was represented, but it's definitely on our radar.
The mic is not on.
Oh, keep talking.
Yeah, again, I just want to thank both of you for excellent work and creating opportunity for things to continue. I think that's really wonderful. I think one of the primary goals of the analysis working group should be not only to find new populations, but to actually work very hard to integrate all whole genome sequences that have been done on African populations. Because we keep saying new variants, understudies, but nobody really know what has been done comprehensively across Africa. H3Africa has done some 400 or something, or maybe 500. My lab, for example, we've done and we continue to sequence. Sarah Tishkov is part of TopMed. They've done over 8,000 individuals in TopMed. Why can't the genome analysis working group pull together all of the available human genome sequences? And in that process, we'll also find out who doesn't want to cooperate but have sequenced African populations. And then we should put them on notice. So I think there really needs to be a need while we are pursuing new variants to see what we already have done and to make that data available in a way that people can download it and use for the work that they want to do. So I would seriously encourage the genome analysis working group to work on that. I wanted to ask a question about the new initiative. What is the role of Illumina in this process? And what kind of understanding has been reached with them in terms of access to this data? And how is this data going to be put in the public domain? Again, my interest here is, yes, we need to keep emphasizing the deficit in terms of African genomics, but it should be our primary responsibility to make sure whatever African data is available, that it is public and that we are all using it to work together to enhance work across Africa and the globe.
Great questions. Thank you so much, Dr. Otimi. So I think for the first question, that's certainly part of our plan. So what we try to represent here are our attempts to get new sequences and to be able to do that. But we intend to integrate that with the existing sequences that we had the first time around. You saw the map with all the places that have been sequenced, to integrate as much as we can with researchers who are willing to share with us, but to also integrate, say, the Egypt genome sequencing project, and there's the Qatar genome sequencing project, just to have, they've approached us about some of the interactions that occur across there. So we do have this grand plan idea that we'll be able to integrate across all of the available things, as long as people are willing and available to share that kind of data with us, which is an as yet unknown entity. As far as the way that we are envisioning the future, I think that we sort of think of no, I've forgotten the question. Illumina. Yeah. Illumina. Yes. So that's a long, I don't know if we have enough time to discuss all of the things that went into this. So I think the primary effort here was that we wanted to ensure that as these genomes become available, they will be made publicly available. And I will say that bar in and by itself is one of the reasons why the map isn't as full as some may like. There are many countries that were approached and there were dots that were on the map that then had to be removed as people were not willing to participate if the data was going to be publicly available. I think that for the existing partners that were there, part of the reason why it's taken us a year to get the ethics and all the paperwork in place was because we have an industry partner, and there was there's a lot of backing and forth and a lot of paperwork, both with the NIH and with Illumina and with all the partners, to ensure that this was going to be an equitable kind of event journey. And so as it stands, the data will be public available. That's the primary bar that we had available to everybody. It will go through the H3Africa DBAC, so that there will be the data access committee will have to sort of vet all the ways that they do now as the data comes through EGA, and the data will be put into EGA. And Illumina's role will be producing the sequencing, and then they're going to use that to sort of help their own sort of in-house kinds of things. But they won't have access to the data before anybody else, and they won't publicly make the data available. And I think that the provisos of that have gone through a lot of iteration to get us where we are. I don't know if Michelle Ramsey has anything that she really wanted to add to that, but it's suffice to say it's been a long process to get this far.
Okay, just one very quick question. Thank you. Very great work.
Quick
Trying to
question,
fill the
Daniel.
gaps. Very quick. Yeah. So with the genome, you were able to find over 3 million new variants. I'm sure similar things probably, similar gaps probably exist for transcriptome, for proteome, for metabolome. And these are very, very important, extremely important for post-GOAs in silico analysis. Are we looking at these gaps and how we can try to enrich the databases with African data?
We are. I think that's been that's one of the things that I was trying to sort of get across there, is that we recognize that to be able to facilitate all of these genome analyses that are being done by individual groups, that having some kind of context for them is really important. And that's why we've been trying to push forward the idea of trying to understand the transcriptome landscape as a starting point. It's kind of the next step that's there, particularly as it relates to the genome. Now, we need money to be able to do that. And so, a big push is to try and understand where we can find some additional funding to be able to support that. But we have certainly a number of willing partners. We have a whole team who has developed already developed a harmonization method for dealing with all of the RNA-seq data that they're getting from different places. So we're ready to go. It's largely a matter of the data generation and the funding to be able to support it.
Thank you very much. All right. Okay, so we're gonna move along. We have a few speakers left and a little time. So our next speaker is Dr. Bamidele Tayo, and who is a genetic epidemiologist and statistical geneticist at the Loyola University Chicago Stritch School of Medicine in Chicago, Illinois. And his talk is PRS for cardiometabolic disorders in African ancestry populations, a Cardinal project, which Neil mentioned earlier, an H3Africa CVD working group, cardiovascular disease working group collaboration. Bamidele. Do you have it's over here?
Thank you very much. I appreciate the opportunity to come around and showcase the collaborative efforts that we name Cardinal, which, as the title indicates, is an example of the type of collaboration that we can get within H3Africa and also partnering with other research groups within Africa. So, as a way of background, what are polygenic risk scores that we develop this project around? These are genetic estimates of risk of diseases or traits. And they basically are built as sum of weighted estimates of genetic variants that are generated from such activities as GWAS or sequencing activities. And the variants could either be risk factors or those that are protective. One assumption or the main assumption for polygenic risk score is that the conditions under study or the traits are influenced by multiple genes. We're not referring to monogenic conditions here. And so the polygenic risk scores are then used to categorize the risk of individuals or individuals to different categories of risk. And the category could be binary and it could be, ordered in such a way that is increasing or decreasing. However, the available PRS that have been developed so far have some limitations or challenges or opportunities that include the fact that the development and the performance of the PRS are actually affected by such basic epidemiological things that, ordinarily should be taken into consideration in studies, such as study design, the sample size that I use to generate this PRS, the population characteristics of the sample, the variant association, the degree of association. Also, even though the list is not exhaustive, something as simple as the genetic model, that is the inheritance model on which the PRS are built, also affects the performance of this PRS, whether they capture such thing as gene by gene interaction. Are you talking of additive, model, dominance, or recessive? All these play a role there. And the fact that current PRS have limitations in their portability to other populations, such as African ancestry populations, and the reason being that the available estimates from GWAS are mostly from European ancestry populations, which we all know are part of the reason that H3Africa is also trying to address that there could be enough representation of African data in genomic environments. And the PRS that we currently have also do not adequately address ancestry diversities. And like I mentioned, not only are they poor in terms of transferability to African ancestry populations, also other populations outside of European ancestry. There's also the depth of methods to develop PRS that actually can perform better in multi-ethnic data or population. So this is where Cardinal actually comes in, to leverage the data that have been generated or have been generated by H3Africa and also other study projects in the continent of Africa. So the cardiovascular working group of H3Africa decided to leverage the H3Africa data and came up with this idea of Cardinal, which was in response to the RFA from the NHI, from NIH. And that RFA was actually developed to improve the applicability of PRS in diverse populations, and also to optimize the integration of large-scale harmonization of genomic and phenotype data. So under the leadership of Drs. Michelle Ramsell and Sally Adebamawo, the cardiovascular disease working group of H3Africa, came together, to come up with the idea, and this involved a number of research groups that are already involved in the working group. And the idea of this collaboration actually was patterned after the shared collaboration which Mayowa, Dr. Owolabi mentioned earlier, that involved everybody contributing, both scientific ideas and data. So overall, there are 5 core H3Africa projects involved in this collaboration, and 3 other non-H3Africa projects that are prosy because of the investigators in H3Africa and their relationship. They are also participants in this Cardinal. So the aims of Cardinal include to integrate phenotype and genomic data from over 50,000 African ancestry individuals that we were able to put together by the coming together of the projects that I mentioned from within H3Africa and the other non-H3Africa projects that came in. The second project aim is to evaluate existing PRS model, develop a novel PRS method that can actually address part of the limitations that existing PRS have. The third aim is to develop an interactive dashboard for dissemination of PRS-related data from diverse populations. So in addition to the 50,000 data set entries that we were able to put together, we also have a list of 6 other non- and H3Africa projects that will serve as replicating data sets that are listed, as we can see. The phenotypes that are included in all these projects, the data actually, which we kind of look as cardiometabolic, because that's what we are focusing on, include the non-modifiable variables like age, sex, and then the continuous cardiometabolic traits, and the endpoint of, the endpoint phenotypes representing cardiometabolic traits. And also we have the opportunity of looking at some intermediate, phenotypes. Because Cardinal was funded as part of 6 other grants by the NIH, so by default, Cardinal is a member of the primed consortium, that is the polygenic risk score method, a risk method in diverse population, which actually includes 6 other projects or study sites in addition to a coordinating center. So not only is Cardinal a mini consortium, it's also a member of a larger consortium, the primed, and primed as representation from 48 assets, the time that I put this together last week, 48 institutions from across 9 countries. And as you can see, currently in Africa, there are, 3 countries there, Nigeria, Uganda, South Africa. But the good thing is, since we have a lot of data that have been generated by H3Africa, there's the opportunity for this map to actually increase, because other projects or studies can become affiliate members or primed. And then that will provide opportunity for this data to be incorporated into the PRS effort that is ongoing, and better representation for Africa in this effort, just as we try to accomplish as part of H3 Africa. So these are the names of the key personnel. It's not exhaustive, but these are the members of the working group and then the individuals from outside the working group. So this actually demonstrates the fact that within H3 Africa, there are opportunities to have functional collaboration. Like I like to mention that the contact PI for Cardinal Isali Adebamawo, the project itself is a multi-PI plan. Thank you.
Thank you, Bamidele. One question for Bamidele. There's someone here, Jen?
Here it is.
Thank you. And it's very exciting to see all of this moving forward and all of the H3A contribution. Looking at this, these are such big data projects, right? And they will not work if data is not shared. So can you comment on how the data sharing is going?
Okay, so the data, there's commitment to share the data. There are some that cannot be shared at the individual level because of the informed consent that was collected, but the bulk of the data are shareable. So, I mean, we have the H3 Africa data available at EGA, and we are leveraging that. And then, yeah. So, but everyone participating in Cardinal has commitment to reasonable extent to share the data. Those that are not shareable because of the limitation of, how maybe legacy data that were collected years before the sharing policy, those will be analyzed and the summary statistics will be shared. And the product, PRS, and other things that are generated from such data are also going to be shared.
Thank you, thank you so much. All right, our next speaker is Dr. Elizabeth Main. She's a hematopathologist in South Africa, currently at Wits University and the NHLS, which is the largest provider of pathology services. And she's going to talk about the H3 Africa biorepositories in continental, continental pathogen surveillance.
Okay, so I have to give a shout out to my current institution, which is actually UCT. And just also to say that I am actually 3 people speaking this morning, not just one. I'm representing Prof. Jaloba from Uganda and also Prof. Abimaku from Nigeria. And I chose to call this Love in the Time of COVID because for those of you who read South American literature, there is a very famous book called Love in the Time of Cholera. And it just highlights how we should be looking at South-South or the Global South collaboration, especially in the time of a pandemic. And also just to mention that in this book, the scientist gets the girl. So it's also got a very good message for us scientists who are looking for love in the middle of a pandemic. Okay, so generally I'm here to shout at people and tell them to put their material in the biorepository, and it's really nice to come here and not have to shout at people because generally you have been very responsive. So what makes the H3Africa biorepository so valuable is that we've been collecting from populations that are generally very underrepresented in other biorepositories. And we spoke about Biobank England or Biobank UK. Certainly we have the samples that they need. In many cases, we have longitudinal cohorts of samples and from different ethno-linguistic groups, and we have a variety of samples. So although our primary mandate was to collect DNA and store it, we also have other samples, including serum and PBMCs. All right, so again, the biorepositories were uniquely placed at the time of the start of the pandemic because we already had the infrastructure that was going to be necessary for the public health response in Africa to COVID. Although SARS-CoV-2 was officially declared as entering the continent in March of 2020, our wastewater surveillance and various papers suggest that it actually entered much earlier, probably at the beginning when it was first described in China. Initially, our cases were primarily from travelers, predominantly high net worth individuals who'd been to Europe for their holidays. And these people were characterized by having very low severity COVID. As it spread, there was a number of questions which repeatedly were asked. And one of those was, why was SARS-CoV-2 not as pathogenic, or certainly not as severe in the African population as it had been in other populations? And here we were specifically looking at African American populations where COVID was described as a very high mortality when compared with other populations in the US. So as an indication, we estimate that 90% of South Africans were infected with COVID, and one of the most pathogenic variants, which was the Beta variant and then the Delta variant. But of those 90% of people, we've had 100,000 case fatalities in South Africa, which sort of translates to 0.25% risk of death. So the question is, why? Why were these people not dying and having severe disease? And we expected there to be much more severity, considering that we had underlying conditions like HIV infection, respiratory infections like TB, and we have a high prevalence, as my colleagues have very eloquently said, of other non-communicable diseases that have translated to increased severity in other populations. The other question, the final question, was where all of the resources were being plowed into countries that were high income. What was Africa's contribution to diagnostic and surveillance efforts more generally? Okay, so thank you to Lashley for this. So there was a SWOT analysis which was done, and I put this sort of here in the middle of the presentation because it really speaks to what all three biorepositories are thinking about. So as I said, the fact was that we had this fantastic infrastructure with cross-trained staff. We had a lot of genetic infrastructure which was linked to the facilities. This created huge opportunities for us to scale up and for us to help provide a public health response in our respective countries. Unfortunately, obviously, as with all of the other biorepositories and the rest of Africa, we had very significant lockdowns that were associated with limited access to the biorepositories. People did get sick. And there was significant competition on the world stage for resources like diagnostic tests, as well as for general reagents, and getting them into the countries was also quite challenging. Okay, so one of the key features that all three of us became involved in very early on was the diagnostic scale up, and it made us very nervous because the FDA, shortly after some of these tests became available in Africa, came out with a sort of worldwide international recall. And so we took our diagnostic validations very seriously. And the three problems we had was, do we have true positives? Do we have true negatives? And what would those look like? And how do we ensure ongoing quality assurance? All right, so in South Africa, the CLS biorepository was subcontracted by our own internal FDA, for want of a better word, SAHPRA, who were responsible for overseeing the diagnostic validation. It was new territory for both us and the regulator. We'd never done anything like this before. But what we had was a vast quantity of pre-pandemic samples which were stored, which we could use as a negative cohort. And we had the infrastructure, the facilities, and the ethical and legal framework for prospective collection as well. Because of that, we were able to share reference material across the continent, actually not just in South Africa. And so because of that, we were able to participate in Africa-wide initiatives in diagnostics. So this is just a summary of some of the patients that we collected very early on. We had that very valuable cohort of people that we collected prior to the 2019 pandemic. This was also the experience of my colleagues in Uganda. So they also established a COVID-19 prospective sample collection, which again was longitudinal. And as you can see, they had over 1,000 samples. And because of that, they were able to conduct significant validations for rapid diagnostic tests. They were also very involved with the public health response more generally. They partnered with industry, and they were also able to conduct genomic surveillance and sequencing of the virus. And this formed a very useful resource in terms of training for PhD and MSCs in both genetics and bioinformatics. And again, this biobank has formed a nexus of their training facilities in genomic surveillance more widely. Similarly, in Nigeria, the Caravan project was responsible for rapid evaluation of RDTs for COVID, and they validated 16 COVID-19 rapid diagnostic tests. All right, what about research and going into the future? What would we be looking at in terms of the biorepositories? As I mentioned, the CLS biorepository collected more than 200,000 individual samples from well-characterized individuals across South Africa. We were also part of a major South African Medical Research Council point of care study, which was presented at CROI in 2021. And we collected a longitudinal cohort of rural and urban patients, both cases and uninfected controls. So what we in the HIV business like to call discordant couples, except they were COVID discordant. I'd just like to point out some of the publications. So to date, this has resulted in 12 publications, but two of the ones that I'd like to point out was a partnership that we had with researchers at UCT, characterizing humoral responses and looking at them over time, but also a partnership with a researcher who utilized our samples to clone COVID proteins in tobacco plants for diagnostic tests. And again, we are working with industry to develop our own in-house diagnostic tests using some of these samples. Similarly, there have been clinical trials in Nigeria where they've been using immunoglobulin to treat patients who had severe disease, as well as interventional studies looking at modifying immune responses to COVID, as well as serosurveillance studies for future research programs and mentorship. And obviously, again, and this is our 15 minutes of fame, we all found our way onto the TV, and there's a Lashley on the TV, so looking very great. So I think the biorepositories have really come into their own. I think into the future, we are looking at the biorepositories as a nexus of the public health response. And we are hoping that these very valuable samples are going to show their worth, because, and I'm going to talk to one of my NIH program managers. She says she doesn't want frozen lollipops. And neither do we. We want lollipops that are going to make everybody's life a little bit sweeter. So that's just acknowledgement to us and our funders, and thank you.
Thank you, Elizabeth. Any questions for Professor May? No, doesn't look like we have any questions here. All right. Well, thank you very much. So for our next talk, it was supposed to be done by Dr. Paulina Tindana from Ghana, and she's not able to be here. And so we're going to have two presenters, Dr. Yantina DeVries and Dr. Mary Kasuli. Yantina from UCT, Mary from Uganda. Okay, I don't know.
A true sign of conviviality, which I think hopefully characterizes the ethics work.
All right.
It's not just the two of us, it's Erisa as well.
Okay.
Who's somewhere here, there he is.
Okay, third person. And the title of the talk is An Overview of Ethics and Community Engagement, Implementation of H3Africa Resources. So they're going to talk about the ethics work of H3Africa.
Cool, and hopefully won't take too long. So what we're going to do today is present, I think, in kind of reverse chronological order and batting things around a bit. So Paulina prepared this presentation because she said yesterday on a call that what she feels is really important is that the people telling the story about the ethics work that we've done in the context of H3Africa ought to be the people that have done the work. And so Paulina put this presentation together, then unfortunately wasn't able to travel, which she only realized on Friday. And so asked the three of us to present this presentation together. So Risa, I think you're kicking off.
Okay, thanks very much. Good morning, ladies and gentlemen. It's been a long journey. Of course, we all know that we all need ethical conduct of research. And basically, that's the outline of our presentation and how it all started. It all started 10 years ago. That's in Accra, Ghana, where Africa are rising with the growth of genomics in Africa. But then we realized that most African countries did not have ethical legal frameworks to govern genomic research. And basically, there was also limited ethics capacity, the regulatory capacities in most of the countries to really govern genomic research and biobanking. And of course, this was informed by a paper that was published by Yantina. Was it in 2017? When you reviewed the regulatory framework from the different countries. So that's when we really felt that we had to come up with guidance documents. And of course, we also were appreciative of the several ethical issues that arise. And of course, consent, very cardinal, community engagement, data sharing and sample sharing. Of course, we've been talking about sharing of genomic data sharing of metadata. So we really need to have ethical sharing of data. Then benefit sharing. This was actually a topic that was discussed in the ethics and community engagement working group for almost 2 years, especially when it comes to sharing our resources with commercial entities. Then ownership, and lately there is feedback of results and findings. This is becoming a hot topic because research participants are demanding for their results. And of course, we need to address consent issues. Which consent models are most appropriate for our, for genomic research and biobanking? Of course, tiered consent, broad consent. There is a specific consent which is so restrictive and kind of curtails progress of research. We've developed quite a number of guidelines, and we'll talk about them. Then, of course, the levels of engagement we have at the community level, we have institutional level, then even at a national level. Because the thing is, communities have their own expectations and preferences, and these have to be taken into consideration. So the informed consent guidelines were developed in the H3Africa guidelines for informed consent were developed in 2014, and were revised to 2017. And of course, the recommended consent models were broad consent and tiered consent. And then one of the biggest problems we realized that for participants to understand consent, they need to understand the concepts and whatever information being disclosed to them. But then there was a problem with translations. You find that if there is a statement in English, in some of our local languages, there are no direct translations, and you have to tell a story for somebody to understand. And the problem is, the more information you tell a patient, the more confused they get. So a participant, the more confused they get. So what is the most appropriate way of translating? I remember, I think there's a Professor Debamo who talked about that in Nigeria, they had come up with, a local library. I mean, like dictionary for local terms. So this is something that hasn't been replicated in other countries. Then, of course, the need for community engagement. Community engagement is very important, especially when it comes to informed consent, genetic literacy, talking to people. How best should the consent be obtained? How best should the communities be involved? Then the community engagement guidelines, these ones were also developed in 2014, and they are revised in 2017. And basically, these ones defined what community engagement is and the key features of genomic research, and why we need to engage communities and how to engage them. Of course, if you have a community engagement activity, then it has to be, the goals have to be clear. Then the community has to be defined. As we all know, there is no easy definition of a community. It can have geographical boundaries. It can be a community of scientists. It can be a community, of let me say WhatsApp, social media, whatever it is. Then, of course, what are the discussion of the roles and responsibilities of the different actors, and what are their expectations of the community, and of course the anticipated challenges. Then one thing we really as ethicists we really push for is when we get information from people, get their samples, we need to give them feedback. The problem is, we go in, get the data, get out. We never give them feedback. So communities have to be given feedback. Then for the next set of slides, I think Yantin about the scholarship and capacity building. But of course, there are some other guidelines we came up with. You have the ethics and governance framework for best practice of genomics in research. We have the DBAC guidelines for for accessing samples and data. Then we had consultants who helped us compile a report on sharing of commercialization, that sharing of our resources as H3Africa with commercial entities and several others. So there's been a lot of scholarship. We've had quite a number of capacity building initiatives and scholarly work. So, yeah, Tina.
Okay. So I think what the sense that Paulina wanted to share with you, thank you, is that an enormous amount of work has happened in the context of H3Africa and in the context of the working groups, but also the LC grants that were funded and that many of you have been involved with. And beyond kind of offering guidance as to, what should the practices and procedures be, for instance, in how we think about consent and engagement and feedback of findings, H3Africa also allowed us to come together as a community to start doing systematic empirical work as well as normative work around the ethical questions that genomics research on the continent raises. And on this slide are just some examples that she put together of really exciting work that has happened in thinking about community engagement, critically, empirically, and conceptually. The guidelines that Erisa spoke about, as well as the process of articulating the data sharing policy at the time, the data biospecimens sharing policy, led to quite a lot of confusion, I think, and quite a lot of concern by ethics committees as to whether and what H3Africa was trying to do, but also whether and to what extent research of this type was appropriate for the contexts within which H3Africa research took place. And so in 2014, which is around the time that these guidelines were developed, that led us to propose to the consortium that really what ought to happen is continental consultations with people involved in research policy, as well as ethics and implementation, to critically think about these things that we were proposing to do. So not just the science and the consent, but also, for instance, practical questions such as the very idea of a data policy, right? A data sharing policy. And these consultation meetings, we held several and reached quite a large number of people, some of whom came back to all three meetings. And one of the people at those meetings was Mary. This was way before Mary was involved in H3Africa herself, involved as an ethics committee member, I think, at the time at the University of Botswana, and giving Kafjan quite a lot of headaches around, Simo goes laughing, around approvals, right? Approving a data policy simply because she wasn't aware or the committee wasn't understanding what it was we were proposing to do. And I distinctly remember Mary in the meeting in Zambia, when we've had now 2 opportunities to talk about the vision of H3Africa, standing up and saying, what we really need is a framework and let's work together. So Mary, let's go to you and talk a bit about the framework that we went on to develop.
Good morning, everyone. We have talked a bit about this framework. When the H3Africa genomic research started, there was a lot of confusion among the ethics committees because I'll admit we didn't know what was happening. And most of us, or a lot of us, were not trained in genomics. So we are relying on the researchers, which was a conflict of interest, to tell us, this is what you want, you must approve this study. But we don't see the data sharing agreement. We don't see ABC. And we were able to work together, which has been a great success. We worked with the researchers to tell us what they are doing, the ethics committees, what we are doing. And that's how we came up with these frameworks, which were guidelines as to how to review this research, new research, which had come in place, approve without really taking too much time. But when I look at the framework that we developed 10 years ago, right now I'm realizing one thing which is missing that is still causing delays in ethics committees approving. And if the researchers, the future is collaboration, it might not happen if you don't train the research ethics administrator. You know, that person who manages the ethics committee, who operates the day-to-day work, most of them are not trained in certain ways, leave alone the ethics, leave alone the soft skills. So really, I think this is a take-home message. We should think about, you know, training this person to be able to function at the same level as the researchers. Then you will be able to go through the 10 ethics committees without causing you so much delay. The only thing that causes the delay in the start of the project is the local ethics committees, which would not have approved. And partly this is because the person who manages the committee is not trained in many ways. I don't know, we've done a study mapping all the ethics committees in Africa, and we really found that there is this person who is silent, but does most of the work, although they have no position, they don't know their roles, they don't know, you know, the activities that should go on to speed up the process. If you're going to write me a letter, I should have the soft skill of communicating between you and me, the researcher. If you are going to, if we are saying no, it's this, is that, I must know how to deal with conflict resolution. I think I can go on and on about this person who needs to be trained, and now, before we start the collaborative studies. Thank you.
That was just Mary's. So I'll skip through a lot of the rest because I think Paulina was exceedingly optimistic about what we'd be able to cover. All of these resources are available online, and many of you hopefully have known them, have used them, and are proposing to use them and to improve them. But what I do want to kind of end off this very short reflection with is with reflection on what I think has been one of the most wonderful success stories, at least in relation to ethics. And that is the enormous amount of capacity building that has gone on, that has happened in the context of H3Africa, and capacity building in a way that in itself has been very convivial. I remember a particular course in Uganda where Erisa, Paulina, and myself got together and taught ethics of genomics for a whole week, and that was a wonderful experience. But also, we've trained several PhD students, many of whom have now gone on to become leaders in their own right. Changwi Manung was sitting here, she's left the room now, but she was one of my PhD students who's now becoming a really strong voice in ethics of genomics on the African continent. Olivia Machabane is with the NIH doing postdoctoral work around the ethics of genomics. Dimpo Ralefala is leading the Research Ethics Office at the University of Botswana, and so on and so forth. The H3Africa work has given rise to an enormous number of trainees who now in themselves are starting to take on leading roles in the face or in the ethics space, in African genomics, biobanking, and other technologies. For instance, Olivia is particularly interested in neuroscience. So I think that to me has also been a real kind of success story of the H3Africa work. There's more that Paulina wanted to say, but I think we're going to leave it at that because we're already half an hour late. But just to say that the experience, I think, for all of us, Erisa as the current chair, Mary as the past chair, Paulina as the past chair, and myself, Cornelius, and Amina Yakubu, has been very exciting and a wonderful nurturing kind of ground for our own scholarship that complements the genomic and scientific work.
Thank you. Thank you, Antina, Mary, and Lisa. All right, we're well over time, but if there's one burning question on ethics, Charles. One burning question, we'll take it.
Yeah, this is really burning. But first of all, I want to thank all of you and the work you've done. I actually believe that one of the true success story of H3Africa was coming up with this framework and doing all of the work for us to have a little more easy time when we are going through the ethics review process. But as you must have heard this morning from Neil and others, that there's still a very big struggle going on. And I also want to compliment you for the fact that you are still looking at the framework and see what is missing and how do we begin to even improve on that. So my question to you is, are you planning a formal process to revisit that framework and to begin to incorporate some of the new findings to even make us better? Your suggestion about training people so they can be familiar, I think is excellent. How do we do that in an organized way so we can make this process a little more friendly? It is absolutely costing us a lot of time and a lot of work to do. And also for our ethics review and the professionals to find ways to make sure that there's true dialogue between scientists and ethicists so that the answer is not a knee-jerk no, when you really don't understand what is going on, what is the concept and things like that. And also for the scientists to be respectful of the ethical processes. So those are just some of the things. This is why I said my comment and question is really burning.
Oh, go ahead.
Okay, thanks very much. Of course, now these, the framework is not cast in stone. It's evolving. Of course, as we get more empirical information, it will, we'll try to improve it. But of course, one of the major problems is we come from different countries with different regulations and different, ethical legal frameworks. So you find that some of the researchers are given harder time in certain countries. I think there is a need for harmonization. My question would be, how do we harmonize so that at least most, I won't say all, so that most countries in Africa, are at the same level, so that we speak the same language. The problem is we are members of the same family, but we are speaking different languages. We need to learn how to speak one language so that at least there is fairness in all countries. Thanks very much.
But I think we're also late. I would just say two things. One is that I think it's really important to always bear in mind that the ethics that an ethics committee does is not necessarily the same as thinking about ethics critically. So academic scholarship around ethics that helps us understand the nature and scope of the ethics of the science that is happening in a space such as H3Africa is the kind of scholarship that I think Erisa is trying to do in his group, that I'm trying to do in my group. It's very different from the much narrower bureaucratic ethics that an ethics committee does. And that is institutional. It's often about risk management. So I think it's important to bear that in mind because those two things are not the same. And the people on an ethics committee are not necessarily the ones that have been empowered or equipped to think critically about ethics. So recognizing that, and then doing the second thing, which is, I think, what we did very well in H3Africa, which is recognizing that in order to take people along on the scientific journey, you have to build community. Because if that relationship between the science and the ethics becomes locked into opposition, then you're getting nowhere, right? Then you'll just keep on getting no's for answers. Whereas what we did in those framework, in the meetings that we had, was try to create community between researchers and ethics administrators, right? Ethics committee members, so that we can have constructive conversations around the policies, to the extent that even one of the critical people became the chair of the ethics working group, right? So building community around ethics in the context of a scientific project such as this one, I think, is hugely important to anticipate those more acrimonious relations and disarm them.
All right, great. Thank you so much. We've got to move on. All right, thank you, Mary, Elisa, and Yantina. All right. I know we've passed our break and we still have one talk to go. We're going to hear from Professor Lee Matisa, who's going to talk about transgenerational epigenomics of trauma and PTSD in Rwanda. And after that, we'll take our break. We'll do a 10-minute break. We'll come back. We'll make it up later in lunch. All right? Thanks.
Thank you very much. I will try to be brief. I just want to share with you the recent updates from our project focusing on intergenerational epigenomics of trauma in Rwanda. And as you know, there are many examples of events, tragic events like what's happening in Ukraine, for example. And of course, Rwandan genocide, other genocide like Armenian genocide and the Holocaust. It's also generating long-term PTSD, which is being expressed by different symptoms like fear, flashback, depression, anxiety, and so on. So when we zoom in, what's happened in Rwanda, that's when even the idea came in to work on this after 1994 genocide, which started actually back 1959, where 1 million of people have been killed just in 100 days in 1994. This is just to represent you what's happened, but what's happening now, it's really impressive. So how do you, this is kind of a contradiction. How do you reconcile this population where you have a rapid social and economic transformation, where you see the stability, the infrastructure developments in all corners, the stability in terms of leadership and the security, and then people who experience this genocide, the survivors, they still live in this society. So that's how you understand that the PTSD problem became an issue with this contradiction. So when you look at on global data, you see, for example, the PTSD represents around 4%. When we look at in this population, the survivors, it's very high, 28%. And depression also very high in the general population, because these are population living in this in the country which is destroyed with this tragic story. So, and if nothing is done, of course, this will end up with the developing some chronic mental health disease, and especially this can lead to the suicide. Of course, when we started this program, there were some evidences of transmissions by the mothers to the children. And then our initial question was that how do we really cope with this? Because as you know, in this country, every year we have 100 days when we have the commemoration events starting from April to July. And what was really our initial focus was, we were seeing children born from 1994 genocide, women who were pregnant, experiencing this problem when we know they were not directly exposed to the genocide. So that's when the idea came in to work on that. So we started with this pilot program where we worked on exposed women and the children who were present in 1994 in Rwanda. And another group of control, the same ethnic group of women who were outside Rwanda because they have been refugees since 1959. They went outside Rwanda. So then we worked on these two groups, and we founded our first evidences when you compare the PTSD and the level of depression, which was very high in exposed both mothers and the children when you compare to the non-exposed groups. So this prompted us just to also explore other mechanism which could explain this, like the level of cortisol. You can see the level of cortisol is really low in this exposed both mothers and the children when you compare to the control group. The same for other biomarkers like mineralocorticoid and mineralocorticoid. So you can see the variations. But most interestingly, the level of methylation. We found high methylation in one of the two genes that we were really focusing on, NR3C1 in this exposed group. So you can see the level of methylation very high in this all CPGs for these two genes. Although we know that Africa represents a great level of genetic diversity, but epigenetic landscape of African population remains unclear. And we also emphasize this in our recent paper that we published with Michelle and my other colleagues, where we show that there's still that need to explore more on the epigenetic level, especially when you want to study things like a trauma. There are some concepts that we know, of course, like the normal developed epigenetic mechanism, which can lead to the normal process, but also some health conditions like cancer. But what is not known is the impact of the trauma. The war or other type of trauma or violence, like intimate violence, to generate this effect of epigenetics. And what we know also is that there's a negative feedback when you look at on the HPA in the case we have trauma or stress. And there are also animal models which have this strong evidence showing that they can just have the transmission being inherited from the traumatized animal models. So that's really all of those evidences were behind our ideas, just to start to enlarge our research to understand what is going to happen with the different concepts like intergenerational and transgenerational. So actually we are now looking for intergeneration, where we see the first generation of the mothers and the children and who were born in 1994 and the young siblings. So those are really our most focus, but we are starting now to see other children also are born from the children who were born in 1994, which is going to represent our next generation for the focus. With that, we started again, we continued our program, and they started a second phase where in this case, we were focusing on the burden of PTSD and cognitive outcomes, of course, looking into the intergenerational effects and the biological effects, especially by doing the DNA methylation, continuing the analysis of the biological markers that I mentioned here, and the whole of in-community engagement, but also by the same time building the capacity for biobanking. So results here, this is the first paper that came in the second phase from my PhD student, where he shows that with this meta-analytic study, there was still very high PTSD in this survivor group of women and the children born during and after 1994. And then we increased a little bit our sample size, used Illumina chip to study the methylation levels. And for instance of time, I'll just skip this and go straight forward to the EWAS data that we generated. And for this, we worked on different samples from the pilot and also the samples we collected, but you can see here the impressive data, I mean, the results that we collected here. So the DNA methylation is still very high in these both children and the mothers. The level is just measured between 0 and 1. When you go across to 1, that's you have a high level of the methylation. So you can see here the level of methylation for this group, I mean, PTSD in this group. Even very impressive, the discovery of the DNA, I mean, the differentially methylated regions, which are very high in mothers who were exposed versus those who were not exposed. Even this also was discovered in the children. Those who were exposed, 16 DMRs versus those who were not exposed. And when you zoom, I mean, when you combine, we also find the DMR level, the differential methylated level, the regions in these groups, and then you see some of these genes, when you zoom in, have been already characterized previously to be associated with the trauma, especially for the pregnant women. And these trauma are also associated with some mental disorders. And again, very impressive that to see that the intello-genocyte exposure was just having some genes with high methylation. You see here in the pink, these are the genes, the 3 genes here that I mentioned among the 6 ones with high level of methylations in our group. And again, when you look at the comparison between these 3 genes and in both group control and the exposed group, you see they are still highly methylated. Which shows the evidence for this traumatized group. And when we talk one by one, again, you will find the same results. So you see in blue, these are the control, but here they are highly methylated gene. And again, we could not just do this without the involvement of the community. You know, we are now dealing with a very sensitive group, or a group experiencing again the PTSD, living in that society which is now transforming with a good transformation of economic and all corners. But of course, we need just to involve them to get their ownership. So we even did the very great job. We collected more than what was expected to be collected, and also could, with this regard, contribute to H3 Bio repository for the samples that we collected here. Now, the implication of the HPA axis mechanism also revealed recent data for this, which shows the evidence. And here, I want just to show you the level of the PTSD symptoms in the control versus case group. So you see the level of the PTSD symptoms very high in the control group, I mean in the case group versus control group. Even when you compare the symptoms among household members, you still have significant symptoms, a level of symptoms in the case groups, those who are exposed versus non-exposed group. The same for the biological markers, good cortisol, GR, and MR also showed differences in this, also having the strong evidence, proof of concept for biological intergenerational transmission effect. And when you finally look at on the severity of the symptoms, again, you look at for the child one, the children who were born in 1994 and the siblings who were born after 1994, you still see there is a high severity of symptoms in these children when we compare to non-exposed children, which also brings an evidence of PTSD intergenerational transmission effects. To just conclude, I also want to just show that this program raised a lot of interest for networking and the collaborations. This is just to mention some group that we are working on with my colleague Gemini from VUB, who are experienced in the biological markers analysis, and also the group from Netherlands who are experienced in the cognitive and the memory task. But also we had just chance to train our researchers here, our PhD student who's graduating in April this year, and the two senior scientists, especially Segun, who also completed his postdoc and now got promoted to associate professor from the London School of Hygiene and Tropical Medicine. So, no, what's next now? So we, as I mentioned, we collected a number of samples from these both non-exposed and exposed groups, which contributed to the H3 Epigene. But we are looking for developing other studies in memory task with collaboration with the group that I mentioned. I will skip this. These are studies that are just happening. And then finally, what we want is just to do other studies, especially for BDNF, to look at on this methylation on the pre and the post-genocide exposure. Thank you very much for your attention.
Great. Thank you, Leon. All right. Unfortunately, we don't have any time for questions. We're way behind. But you could always connect with Leon and any of the other speakers if you have questions that you were not able to ask. Thank you to all of the speakers. And for the pioneering work, this is just a snapshot of the pioneering work happening in H3 Africa. And we're going to hear more about the work throughout the next day and a half. So we're going to take a 5-minute break. It just got decreased by 5 minutes. And so we're going to come back and then we'll try to make it up later. All right. Thanks.
Yeah. Yeah, so it's fine. She would not use the headset microphone. Yeah.
All right. Welcome back, everybody, after your brief break. I hope you had some sustenance to get us through the next an hour and a half. It is my absolute pleasure to chair this next session, which is on emerging leaders and early career scientists, scientific advancements and vision for genomics. I'm Emer Kenny. I'm a professor of medicine and genetics at the Icahn School of Medicine in New York City and one of the members of the International Expert Committee. And just a few notes before we start. A general announcement. If you are heading to the airport either Tuesday night or Wednesday morning, there is a sign-up sheet out at the registration desk that you can sign up for transfer to the airport from your hotel. So you can just go out at the next break or at lunch and sign up, and arrangements will be made for you. And then the second announcement is that as we are running a little behind on time, I'm going to be a little strict, nice but strict on time in this session, and would ask anyone who wants to ask a question, just try and keep it as succinct as possible so we get as much conversation as possible into, unfortunately, a small amount of time. Okay, so with that, it's my great pleasure to welcome our first speaker, Dr. Simone Richardson, who is a senior scientist at the National Institute for Communicable Disease. Welcome.
Hey, thank you very much for having me. It's gonna go. Yeah, it's not advancing. Okay, I will be speaking about finding function in immunoglobulin genetic diversity. So really going beyond just finding genetic diversity, but I will discuss that. Okay. All right, so antibodies obviously protect us against many infections. They're unique and they are antigen specific. And so we as a group are extremely interested in understanding how they work, where they come from, and how they evolve. So this is the structure of an antibody. An antibody can be separated into two distinct regions, the first of which is the Fab portion. That Fab portion binds to the antigen and is responsible for blocking of infection or neutralization. But the other half of the antibody is equally exciting. It's called the Fc portion. It binds to the Fc receptors on healthy cells and essentially recruits a slew of cells to a site of infection in order to cause cytotoxic function. So while the Fab portion potentially blocks infection, the Fc portion clears infection. And there's a huge range of different functions that it does. So engulfment of particles, phagocytosis or cellular cytotoxicity, or nibbling of cellular membranes. So there's all these different functions that the antibody can do, and essentially serves as a link between the innate and adaptive immune systems. I think it's dying. There we go. We as a whole, aim to understand antibody genetic diversity in a South African population with a high prevalence of HIV infection and the impact that that variation has on antibody function against HIV. So just to take you through the first really exciting thing that we found. So this is really just taking you through a tour of 5 years of work. But this work has been done by a master's turned PhD student, Elaine, who essentially looked at novel allelic variation in this top bit of the antibody, which is the variable region, and found that there's about 14% of novel alleles. She also compared the repertoire, so those antibodies that are actually expressed in an individual, and compared them to just the ones that we were able to sequence in the genome. And so it's a very valuable data set with a huge amount of diversity in the South African population. And so she really is the first to describe a lot of this variation. The other part of the variation that, of course, we're equally excited about is the variation in the Fc region. So antibodies come, of course, in many different flavors and many different isotypes, all of which perform slightly different functions. And what we were able to do was actually sequence through a mixture of both Sanger sequencing and PacBio sequencing the constant region of these antibodies. And what we found is also a huge amount of novel variation. So in IgG1 and IgG3, you can see the little yellow, the little colored part of the pie there is slightly smaller, but it's usually because these are very widely described. But in a South African population, we are seeing a huge amount of variation in IgAs. IgAs are typically mucosal antibodies, which of course are important for HIV, but not just HIV, also respiratory disease like COVID. And of course, this became an interest of ours. What we were able to then do was actually show that some of these novel alleles that we were finding in, for example, here, IgG3, actually had functional implications. So what I'm showing here is just the bottom bit of the antibody binding to an Fc receptor. And you can see there in red, the little red dot was a novel mutation that we found in IgG3. And what happened was when we made this antibody shown in yellow on this bar chart here, we could actually improve function. So this is improving the ability of antibodies to engulf particles. And so really showing for one of the first times that these mutations, these allelic variations that we're finding in South African individuals are actually contributing to a change in function. We then took the one step further in a huge amount of work. So what I'm showing here on the, would be the left-hand side view, is the trimer of HIV, which is the envelope protein. And there's a bunch of different sites that are targeted by different antibodies. And in the middle there, we have a bunch of broadly neutralizing antibodies. Those are the antibodies that are able to target or block a lot of different clades of infection in HIV. And we made IgG1 versions and IgG3 versions of these. And we're actually able to find that there was increased ability to engulf particles, increased ability to pop cells, and then increased ability to actually block infection. And so that's what it's showing here in the red. Is the IgG3, and it's actually more potent in combinations than IgG1 at blocking infection. And so really this work has implications for harnessing allelic variation for benefits in therapeutics. One of the kind of really big milestones that we have in our 5 years that we've been with H3Africa has been the establishment of an FC effector function core across a number of diseases. So this is the ability to measure all of those functions that I mentioned before and understand what the genetic impact of some of the changes we were seeing is. We've since gone into several different diseases. So we've done it in influenza, in COVID-19, in HIV, and it's really been a huge success. There's been 2 PhD students, 3 master's students, and then 27 publications since 2018. And so that's really something we're very proud of. And this is all from H3Africa. And then, of course, like many other labs, we also had to repurpose our platforms for SARS-CoV-2. And it's really generated data that was important for policy information in South Africa and for the vaccine rollout, especially the middle one there about Omicron triggering cross-reactive neutralization profiles. And this was all, again, of platforms that were designed for H3Africa. And then, of course, the thing that we're most impressed with always is capacity building. And these are all the students or the degrees that we've generated as we've gone. And what's more impressive, I think, is progression through. So Elaine, who's shown there in the red hair, has done her honors, her master's and her PhD under H3Africa. And I have done both my PhD, my postdoc, and I'm now a co-investigator on this grant. So it's really just showing how this environment has allowed us to grow. So ultimately, and really just in summary, we've begun to understand how to harness genetic variation for real-world applications, clinical interventions, specifically for antibodies. And so that is 5 years summed up in 8 minutes. And just to acknowledge the lab of which I'm part, of course, you can see none of their faces because this was during COVID. But we're very proud to have been part of H3Africa for the 5 years we have. So thank you very much.
Thank you so very much. And we invite, and thank you also for being very timely. We invite anyone to ask questions now, and the microphone will come down to you if you raise your hand. Am I seeing hands? Yes, okay.
Thank you. Just wanted to know if you had different samples from different populations when you looked at the alleles.
Sorry, I can't hear you properly.
When you did the sequencing for the immunoglobulin genes, you use samples from different populations? Or?
So actually, we're primarily interested in South African individuals. So we've only done South African individuals. Oh, can you hear me? Okay. So we've only done South African individuals. This is just looking in that population that is specific for our high HIV burden. It's actually a Caprisa cohorts in Durban. Yeah.
Okay, any other questions from the floor? Okay, well, then we thank our speaker. Thank you very much, Dr. Richardson. And next, we welcome our next speaker, Dr. Gerald Mbuya. Welcome. And Dr. Mbuya's talk will be on building resilient bioinformatics ecosystems in Africa. And Dr. Mbuya is a senior scientist in the African Centers of thank you, Excellence in Bioinformatics and Data Science. Thank you very much.
Good afternoon, everyone. I thank you for the opportunity given to me to present to you our aspect when it comes to building the capacity for bioinformatics on the continent. As already introduced, I'm a CAF Journey Fellow. This journey started in Stellenbosch in 2014 in May, where I think that time it was Professor Nabwami who was heading CAF Gen, and currently we have Moho here. So Moho, what I can say before I start, the tree you planted, it is now, you are seated under the tree. So the implementation science expert for bioinformatics at the Africa CDC based in Ethiopia, Addis Ababa. So away from the science, let's talk politics. Okay, so what has happened in the last 10 years is a journey that has seen acceleration of genomics in Africa, looking at building both pathogen genomics and human genomics. And there are two entities that have played a big role, the H3Africa. For those of you who have been here, you have seen how much has been done on the continent in the last 10 years. And then we have the Institute for Pathogen Genomics, which is based in Africa Union under the Africa CDC, which is the Department of Public Health under Africa Union. And these are some of the achievements for the H3Africa. We have seen many of these in the previous presentations, but of these specific importances, what you see here, I'm part of that group that has that is found in that circle, capacity building. And like I've told you, the journey started many years ago. And you have seen the investment, which is to the tune of about $176 million US. But then look at the other side, which is looking at pathogen genomics, and that is under the Africa CDC. So over the past 2 years, it is not compared to the H3Africa, which is 10 years. The past 2 years, you have seen about 53 member states contributing the SARS-CoV genomes to the global database, that is the GSAID. And we have managed to ship about 33,000 samples across the continent and distributed, as of now, about 49 NGS platforms ranging from the mini-iron MK1C to the NovaSeq, and depending on the capacity of the lab. And then you have also the tune so far has been estimated to be about $100 million US in the last 2 years, contributed majorly by the Gates Foundation and then Illumina and Oxford Nanopore. And as of now, today morning, we have about 150,000 SARS-CoV genomes that have been sequenced from 53 member states. Only 2 countries have not contributed anything. That's Eritrea and the Western Sahara Republic. We have trained over 400 trainees across 53 member states. And then reagents for about 180,000 SARS-CoV genomes have been shipped and distributed to the different countries in Africa. And we have done pathogen genomics mapping capacity in each of the 41 member states out of the 55. So looking at how this capacity from the H3Africa perspective has been, we look at the biorepositories. We have seen the presentation for the biorepository group. We have a biorepository in Nigeria, the Institute for Human Virology. And then we have in Uganda, the Integrated Biorepository for H3Africa, and then also the CLS in South Africa. And that's also the H3 Bionet, from which I greatly benefited. What I can tell you is I'm always grateful to Professor Nikki. I think she's not here, but I attended almost every training that she put across on the continent. And the lessons learned during that, the lessons I used to do when I was offered this opportunity to train at the continent, I borrowed much of that from the trainings that I undertook in H3 Bionet. The bioinformatics training programs that were also launched by and funded by NIH, and one of them was at Makerere University, of which I managed to contribute in my own capacity, though not major. And then the collaborative research centers, we all know about them. You have seen their work that has been presented, from which also CAFGEN is part of the collaborative research project that planted the tree, like I've told you, the story of borrowing the words of Moho. And then the research groups or projects, those were several of them, over 51 research projects distributed across Africa. And then you have seen the presentation from Dr. Elisa Mwaka and also Gentida. So that is what H3Africa has done, and we can't emphasize more, but we're always grateful. Of course, there are gaps that remain looking at this map. You see where the human genomes have not been heavily represented, and Africa still has those very blind spots that you can see. That means H3Africa needs to still push. If you're contemplating pulling out the plug, you need to think twice, because we need all this map to be green, as you can see elsewhere. So how did we do this? Is we leveraged the network of facilities that were already established in the continent. What SARS-CoV showed us was sequencing and data sharing were key components in any health system. So during that time, I think everyone knew the continent was going to probably face the worst crisis in maybe the last 100 years, but this wasn't the case. So what Africa CDC did was to go and talk to the institutes that were already established, including the SGID in Nigeria, headed by Professor Happy, Sambi, and then CRISIP. So these were approached to offer training, but now we realize that this emergency needed the participation of the National Public Health Institutes in Africa. And these were not capacitated with genomics, the platforms and the training. But of course, one benefit of SARS-CoV is now we know anyone, anywhere knows how important sequencing is, but also data sharing. So these are the components that this network managed to ensure. So how it was formed, we had these labs that were equipped with sequencing platforms, and we lined up these countries under each of these labs to ensure that they can ship their samples to those facilities, so that to ensure they can have these genomes sequenced. We all know the two important SARS-CoV variants of concern, that's Omicron and Beta, were initially discovered in Africa before anyone else, and eventually that data was shared. So we looked at coordination, leverage the existing capacity, access to sequencing, but also data sharing, which were key components of responding effectively to the pandemic. And this is how we leverage the network. Yeah, so the emerging opportunities, having learned from what the network could do, like I've already shared with you, we have over 150,000 SARS-CoV genomes in the public database, and these are coming from 53 member states, except 2 countries that have not shared anything. I bought this from some of the talks from Francis Collins from last year around October, where he introduced this. It's an idea that we hope can take over from H3Africa. And like Moho said, CAFGEN will be happy to participate as one of the genomic centers of excellence. So looking at 2019, just before COVID, we tried to do a mapping of the NGS capacities in public health institutions in Africa. And only the countries you see in green had the functional NGS capacity. So if COVID had taken on the continent then, I think we were not in positions to respond. But as we talk of now, this is where we stand. Over 39 national public health institutions are equipped with functional NGS capacities, and they can also do the analysis. That's very important. So the continent, looking at the two big genomic investments we have seen in the pathogen genomics, but also in the human genomics, how do we move on from here? What we propose is kind of ambitious, but we hope it can be taken over. If you are to look back 20 years ago, when we had the HIV epidemic, we all didn't know about the viral load. But as we talk of now, viral load testing is integrated in most of the African countries' healthcare system. So we hope also genomics can become part of the healthcare systems across the continent. That's the only way we can probably sustain this endeavor. So, and what we are proposing is at a country level, we have antimicrobial resistance programs, antimalarial drug resistance program, drug resistant TB, NGS HIV drug resistance testing, and beyond. And then we also need to support the training programs for genomics and bioinformatics in different countries in Africa. And then also we prioritize what we call the high impact genomic use cases. We are demonstrating some of these in Africa using cholera and some immunizable infections like polio and measles. But each country has a priority list that they can try to put forward in their healthcare systems. But beyond COVID, countries like South Africa, they have already started doing environmental surveillance for wastewater, trying to pick up pathogens of public health importance. And then we talk about the collaborative population level human genome sequencing endeavors. I think Professor Neil talked about this. For countries like Turkey, Qatar, UK, they're sequencing batches of 100,000 genomes, well characterized to inform public health care. But I think it is only Nigeria and Africa that was trying to put the first step towards this, but we hope other countries can also move in the same direction. And also looking at promoting personal genetic testing, which is a way of offering direct to consumer genetic testing services on the continent. If you went to the website of 23andMe, I can assure you, even if you wanted a test now, if you're in Africa, they are unable to allow you proceed. Okay, so this is my last slide. Thank you. Yeah. So looking at what we have done in 2020, we managed to sequence 5,000 SARS-CoV genomes. 2021, we went almost 12 times, that is 60,000 SARS-CoV genomes. And then 2022, 81,000 SARS-CoV genomes. But now the problem comes up. We are not ready for this data. The look where we look at over 150,000 SARS-CoV genomes as we talk now. But if you went across these national public health facilities, you can hardly find any storage of the raw sequences. Many of them have uploaded the FASTA files on GSAID. And thank you very much.
Thank you very much. Wonderful talk. So we have time for one question. I'm just scanning the room. Has anybody got their hand up? Oh, I see Nikki over there. Oh, you can't go yet.
Thanks, Yura. So it's actually more of a comment, but maybe you can comment on it too, is that we have H3Africa in the academic space who have built a lot of genomics capacity, and then we have the NHPIs. And I saw at the meeting, the Africa CDC meeting at the end of last year, that there's a disconnect between those, that actually some of the public health institutes should be able to leverage some of the genomics infrastructure and skills that have been developed through the academic sector. So do you think there's a way that we can, I mean, how do you think we can better connect those groups such that you don't get left behind in the public health space with all the advances that are happening in the academic space?
Okay, so thank you very much, Nikki, for that question. So at the moment, having realized that the problem of data infrastructure is a big one, we received funding to set up data platforms or infrastructures in the national public health institutions, but that will take some time. So at the moment, we are reaching out to academic and research institutions to offer these services to those that are willing to do that. But of course, different countries have different requirements to have this data taken from the national public health institutions to academic institutions. But in some cases, it is yes. In some cases, it is no. But still now we are moving into a direction where we can support each of them with a minimum infrastructure for genomic data management. Thank you.
Thank you very much. Let's hear again. Okay, and our next speaker is Dr. Tinash Chikawar. And Dr. Tinash Chikawar is a Wellcome Trust International Training Fellow and a researcher based in the University of Witwatersrand. We met in the elevator on dinner last night, so you're very welcome to the stage. Can you read your title?
All right, sure. No, thank you so much to the organizers for allowing them to put a face to the number of the H3Africa fellows that have benefited a lot from this H3Africa resource. So I'm going to take you through this journey. So I am trained as a nutritionist, and some of you might wonder, who am I even to speak about genetics in this vast audience? But this is just through the benefits of H3Africa. So I come from Zimbabwe, one of the underrepresented countries with regards to genomics, but it's also known about other things. For instance, we have many trillionaires. So you guys have been trillionaires, you should come to us. We have actually been trillionaires, but this also just shows the economic situation in this terrain. But nonetheless, there's also notable scientists that have emanated from this country, Prof. Masimirembwa, Prof. Dandara, Vicky Nembaware, and Alice Matimba that are doing great stuff. But for me, I just started as a nutritionist working in relief agencies. But along the way, I got fascinated about the concept of nutrigenetics. This made me to go to South Africa. And my first brush with people from H3Africa was through the SASHG in 2013. And Professor Michelle Ramsey was one of the organizers of that meeting. I won an oral presentation, first scientific congress that I'd attended, and then began a journey. I was interacting with Professor Zen Lombard. I also met Professor Dandara then, and Ambrose, and many others. And over the journey, Professor Zen Lombard took me in while I was doing my master's. I wasn't even part of H3Africa. I was at the university, but I would come by and I'll get assistance in the analysis of my training. I moved over to even support from Professor Scott and even Sean Aaron. I was able to submit my first job on the cluster while at Sydney Brenner. But over the years, post my PhD, which I then also started with Professor Zanay, and also getting exposure even into grant writings and fellowships. Now, where am I? I'm now even contributing to the training of other fellows through H3 Bionet. Also was part of the first initiative of the African Society of Human Genetics, going to Ireland, actually to train people in Ireland about genomics in Africa, and also partnering with others, the Wellcome Connecting Science, and even going in March to Nigeria, actually to train people about Mendelian randomization. So indeed, I started as a novice, a nutritionist, but boom, I'm now even training people about genetics. With all this, I was fortunate to nest my first ever grant in the CVD working group. And I'm so grateful to the number of PIs that are part of the CVD working group, as we have heard from Dr. Tayo. And this is a fellowship that really has transformed the science and also participating in the Cardinal project. We got the advantage, even though we're junior, even to have sections that we're writing about. And this also emanated in me also being one of the multi PIs in the DSI grant that we have submitted. So indeed, the road has not been always rosy. There've been a number of challenges along the way. But what's so important is the issue of being part of a village. You know, they say it takes a village to raise a child. And indeed, it also takes a village to raise a scientist. You know, one of all the H3Africa meetings that we've attended, the Rwanda just stands brilliantly in my mind as one of the best that I attended. And this is at this moment where I met Shegu for the first time. And later on, this spilled up in one of our greatest collaborative acts, you know, being a nutritionist, now being a senior author, joint senior author in a Nature Medicine paper. Wow, that is all through H3Africa. And even the examples that we're getting, I remember we're sitting and we're reflecting of the duo between Professor Ambrose and Professor Dandara. And we're like, wow, we can even work together and really also get to these heights. And indeed, through God's grace, we also topping on these heights. But much more, aside from the mentors that are directly with you, there's a good culture, maybe that we might not know, but that exists in this community. A number of people are always looking out for even young people like us. We hear someone say, no, I'm doing this, can I recommend you here? And I so remember I was attending interviews last year, and one of the members who's actually in this room had even recommended me to chair a session at the American Society of Human Genetics. And the people who were interviewing me had students that were presenting and I was chairing. And lo and behold, when you go to the main room where the meeting was done, there was actually a poster and there was my name there where I was standing. And you know, I think when these people went, there's like, I think this guy is just doing exceeding things, not knowing that it was just because I'm part of a village, I'm part of great mentors in the society, in H3Africa, who are always recommending these things. And really, indeed, the best is yet to come. So where am I now? So I'm moving in June to Harvard Medical School. I'm going to be starting my research group there in the Bring Up division of genomics and also the Channing division of network medicine. I'm so much fascinated now about the concept of lean obesity that is quite prevalent in East Africa. But if you come in South Africa, a lot of obesity, a lot of diabetes is obesity driven. So in this project, we're seeking to use multi-omic approaches to really understand and tease out mechanistic insights driving this unique phenotype. So indeed, I thank you, everyone. Thank you. Much thanks to H3Africa. Much thanks even to the people that have directly mentored me on the way, and even my collaborators that I continue to do great with. Thank you.
And well done on timing. Thank you. So any questions from folks in the room, please raise your hand. I'm not seeing any. Oh, here we go, Zane. Wonderful.
Thank you, Tinashe, for that overview. And I think it's such a positive message that you've given, but I think I also want to just throw it back to you and say that it is really important to take up opportunities, and that is what helps with success. So that's my comment. My question is, what advice in 1 minute will you give to young scientists in the audience who wants to be successful in genomics in Africa?
Thank you so much. In 1 minute, wow. Yeah, so I think I can only say what works for me. I think we undermine the view even of working together, even as fellows, like collaborating at that level. But I think that is something that has not been explored. Indeed, we have our primary mentors, but we need to explore this option. But above all, I think be approachable, approach many people, be shocked how many people are willing to help you, even people that are way senior than you. Relish advice, visit them at their institutions, tell them your plans. You know, don't just think you surprise people, but as you discuss, people help you refine your ideas. They even give you suggestions. So in a nutshell, make the most of the mentors that are ahead you, and also collaborate even with your fellow peers. Thank you.
Thank you very much. Let's thank our speaker again. Fantastic. So our next speaker is Dr. Yor Hamdi. I welcome her to the stage. Dr. Yor is a postdoc in the Institute of Pasteur of Tunis, and the title of her talk is New National and International Initiatives in Genomic Medicine Toward Data-Driven Healthcare. Welcome.
Okay, so thank you very much. I would like to thank the organizers for the invitation. I'm Yosef Hamdi. I'm from Tunisia, and I'm going to talk about a new initiative that we are launching at the national and international level. So I think that there's no doubt about the great progress that we've made in Africa in the field of human genetics, and this is mainly due to the efforts of the H3Africa Consortium and all those who are working in this great project, but also to other scientists and organizations from outside of the H3A. And however, I also think that we are all aware about the big gap in North Africa in terms of data generation. And that's why today I'm going to really focus on this region and to share with you some initiatives that we are working on in North Africa and mainly in Tunisia. So I will start with Genome Tunisia project, and this project was launched in 2019 and is funded by the Tunisian Ministry of Health and other international partners. Genome Tunisia multidisciplinary project, and the main goal is to really generate the reference sequence of the Tunisian genome by sequencing around 10,000 whole genomes. So as I said, Genome Tunisia is a multi-center, and we are more than 15 centers from Tunisia that are involved in this project, and also it's a multidisciplinary team. And as you can see, we are really a very a multidisciplinary consortium. So we have anthropologists, human geneticists, bioinformaticians, medical geneticists, and this is really important to include clinicians on this kind of consortium, pathologists, and so on. And also it's a multi-phase project. So in its first phase, Genome Tunisia will more focus on building facilities for research and clinical use. And also the main goal is to generate the reference sequence of the Tunisian genome, but also it will be an opportunity to start the transition from research to clinics. In its second phase, So it's in its second phase of Genome Tunisia, we will be working on a program. So it's not a project, it will be more a program that will last for 10 years. And we have around 12 task forces that we will be working on, going from LCs to omics, to the generation of other omics data, to biobanking and digitalization, genomics, entrepreneurship, and so on. So it's really a huge project that we are launching in Tunisia. And we hope that by the end of 2024, we will be able to generate the first sequence of the Tunisian genome. The second initiative that I will talk about is Agenda, and I think that Neil and Ananyu talked about this initiative this morning, and I will go quickly on that. So this initiative, so North Africa is represented, and around 200 genomes will be sequenced from North Africa, from Libya and Tunisia, and now we have all samples that are centralized in Tunisia, and soon they will be sent to South Africa, and from South Africa they will go to UK for sequencing. Now I will move to the disease-based cohorts and projects. So this is really very important to start generating genomes for affected people. And in Tunisia, we decided to work on two main pathologies, breast cancer and lung cancer, because they are the two most prevalent cancers for males and female. And for breast cancer, we decided to work on two main subtypes. So the inflammatory breast cancer and the triple negative breast cancer that are the most aggressive and the most deadly forms. And also because we don't have enough therapeutic options for these cancer types. And also because they are more frequent in African populations compared to other populations. So for inflammatory breast cancer, we will be sequencing around 300 samples, which is a quite large cohort for the IBC, which is a rare cancer. And we will be doing this in collaboration with a new startup in Africa based in Ghana. And this will be a good opportunity for us to start collaborating with other members from Africa. And also, as scientists, our mission is to start encouraging these startups and pushing them, because it's important to get more and more startups and spin-offs in the field of genomics in Africa. The second project in breast cancer will be in triple negative breast cancer. And here we are 4 countries from North and West Africa. And a good amount of data will be generated, around 800 whole genomes for triple negative breast cancer and 200 controls. And again, it's a collaboration with industry, a pharma company. And this shows the importance of collaborating with industrial partners to generate more and more data in Africa. As we are talking about cancer, so the fourth initiative will be on the African Oncogenetics Network that we are putting in place, and it's led by Tim Rebeck from Harvard and Funmi Olopadi from University of Chicago. And so we are trying to work on different aspects related to cancer. So the education, training, research, policy, and implementation. And we are more than 15 countries from Africa that are involved in this network. Finally, it's about personalized medicine, and I'm going to talk about PERMEDINA. And PERMEDINA is a new project that we are launching in North Africa, and it's about personalized medicine in North Africa. It's a 2-year project funded by the European Ministry of Foreign Affairs. It's done within the Institut Pasteur networks, Tunisia, Algeria, Morocco, and Paris. The coordination is in Tunisia. And we are collaborating with different partners from Africa and from Europe. The main goal of PERMEDINA is really to assess the readiness level of precision medicine implementation in North Africa. And we will be doing that by, we go through 4 main work packages. The first is about situation analysis, inventory of existing precision means in the region. And the second one will be a precision oncology pilot phase on lung cancer. And here we'll be also generating lots of data on lung cancer, around 500 whole exomes and 500 whole transcriptome, in order to try to identify new biomarkers that can be associated with lung cancer in North Africa. Also, we will be working on training, dissemination, communication, and the 4th work packages will be dedicated for policymakers. And we are aiming to draft what we call the PM plan, Personalized Medicine Plan for 2030, that will include a roadmap and recommendations for health authorities. And last week, we've been in the EU-Africa PERMED meeting here in Cape Town, and we started discussion collaborations between the 2 initiatives. And so we'll be collaborating along with the IC PERMED, which is the International Consortium of Personalized Medicine. And so a big partnership will take place. So we will apply for the European Partnership for Precision Medicine call that will be funded by the European Commission. So it's hard to talk about all this in 8 minutes. I mean, I will be here for the next 2 years if you have any questions for all these initiatives. But I try to keep these main notes or ideas that I think that are really extremely important to work on. First, multidisciplinarity is important. We need really to include other specialties in our projects and our research projects. It's crucial, mainly the clinicians. It's extremely important. Data generation is a gap in North Africa, and we are aware, and we are committed to generate more and more data at the region. Infrastructure, and here I have to say that for the agenda initiative, we've not been able to sequence any human genome in Africa. So we are, going to sequence in UK, and that means that we are still, we have work to do for the infrastructure in Africa, and I know that in North Africa, 4 Nova-6 have been put in place recently, and hope that it will help to generate more and more data from the region. Functional genomics is also key, and I'm deeply convinced that we really need to work on this field in Africa, because it doesn't make any sense to continue generating data and identifying new variations and millions of variations without really validating the functional effect of these variations. So in Tunisia, we are going to put in place genome editing platform in collaboration with Japanese partners, and hope that, again, it will help us to work more on this field. Healthy and disease cohorts is extremely important. And as I said, we are sequencing healthy and patients because we need to compare to identify variations from affected people. And finally, collaboration with industry, it's extremely important, but also we need to involve civil society in our project because of the importance of socioeconomic partners in the field of genomics. So that's it. I would like to thank all our collaborators, all our funders for their support, and mainly the H3Africa. I'm really very grateful. I'm really very grateful to H3Africa because as a North African scientist, it was the opportunity for me to discover Africa. It was the first time that I traveled to Africa with the H3Africa consortium. So thank you very much. And thank you very much for the networking, for all what we learned from this big family. And I hope that we will continue working and collaborating together. Thank you very much. And here is my favorite quote. Sorry, it's my favorite quote. Where there is a will, there is a way. So let's continue the journey together. Thank you.
Thank you very much. And can you raise your hand if you'd like to ask a question of Dr. Hamdi? Do I see any hands up? Okay. Oh, there, over there to the side.
I'm so sorry for my voice. I'm really not in good shape, but.
Thank you for the presentation. It was wonderful. My question goes to how did you go about convincing the Minister of Health in Tunisia to sequence 10,000 human genomes? Because this is what we want for every country in Africa, but I can assure you it is not as easy as it is, especially getting funding from the government. So how did you go about that?
Yeah, I forgot to say that I've been trained in Canada, and I was I've been 15 years in Canada, and I worked on the Genome Canada project and the Genome Quebec project. And when I've gone to the Tunisian Ministry of Health, I was like, look, I have some evidences that these kind of projects are, I mean, we can do a lot with these projects, and we can really bring a lot of knowledge. And also, one advice that I want to give when you talk with policymakers, talk about economy and about the economic values of this kind of project. So I did a kind of a study on the importance and the economic impact of the genomic study, human genome project. And then they was like, oh my God, it's good, and we will go on. But I also have to say that the COVID crisis helped me a lot to convince the government, because as I said, this project started like 2 or 3 months before the COVID. And when they felt the need of getting the infrastructure of sequencing and to generating genome data, then they were really very convinced, and we started the work together. So I know that it's not easy to bring governments to fund research in Africa, but yeah, let's try. And if Tunisia succeeds, I think that all other African countries will be able to do so.
Okay, thank you very much. Great. Okay, so our last speaker of this session almost needs no introduction. This is Dr. Sejin Fatumo, who is the computational geneticist and associate professor of genetic epidemiology and bioinformatics at the London School of Hygiene and Tropical Medicine, and a Wellcome Trust International Fellow. And the title of his talk is The Emergence of Large-Scale Genomic Research and Genomic Leaders in Africa, the Ripple Effects of H3Africa.
Thank you. I've been asked to talk about my story, and I've not recovered from hearing what Nasha was saying. So during the H3Africa at the beginning, I was a postdoc, and now I'm an associate professor, and that's what I was asked to talk about. So pre-H3Africa, African genomics study was characterized by ethical dumping, helicopter science, and exploitation. And we know that people come, they fly in from high-income country, like in the UK, in the US, so they take sample from Africa and they go away. So that is what is called sample safari. Okay, so there's now a growing number of genomic initiatives in Africa, but African genetic data from H3Africa program is everywhere. If you look at the map, you see there are programs that are coming in Africa. There is a Nigerian on the Cape project, there is Ugandan project, the other projects. But look at that map, look at the red, that is H3Africa, is everywhere, all over Africa. So this slide is showing the parachute science, electrical science versus the African-led science, which is what we do now in H3Africa. So if you look at what is in between them, that's happened last 10 years, the training, a lot of training, education, capacity building, infrastructure building, next generational genomics leadership empowerment, regional collaboration funding. So you see all that are transformed from just the parachute science to African-led science. So African scientists have begun to set the region genomic agenda. We now gather the sample ourselves with strong community engagement. We've heard about it today, with informed consent, with clear understanding of the need to give back to the community. That's what we do now, which is the real science that we should be doing. So even at that, Africa is still underrepresented in genomic study. We know that, but it would have been worse if there was no H3Africa. So we published this paper, which is last year, which shows that even now, Africa, we are still at 1.1% of genomics that have been studied. So H3Africa has taught us how to analyze our data. So this map, what you see here, UK Biobank has been the most characterized genomic data all over the world, 500,000 samples. That includes 30,000 individuals that are not non-European. What you see is that scientists from all over the world, they don't even analyze those 30,000. That's a lot of sample. That's about 1/3 of what H3Africa has, but they don't analyze it. The reason why they don't do it is because they don't know how to do it. But H3Africa has taught us, because anything that is not European population, homogeneous population, is harder to analyze.
population structure and all kind of stuff. But we've been taught how to analyze this data, and we are determined to do it. So H3Africa ripple effect, which is what I've been asked to talk about. There been a lot of, I've built a lot of capacity in the last few years. I'm now leading some 3 major African genomic programs, including Uganda Genome Resource, Malawi Genome Resource, which I'm very excited about. So we are now genotyping about 7,000 samples on H3Africa array here in Cape Town. 7,000 samples being genotyped in Africa is one of the largest so far that is genotyped in Africa. Other samples that have been genotyped either California or Bristol, but we are taking that step to do that in Africa. I'm also co-leading the Nigeria 100,000 genome project. These are very fantastic genomic programs that are ongoing. So the Uganda Genome Program, we published this paper 2019, which is the largest ever genome study in Africa. And since then, we are now leveraging regional and global collaboration to improve power for discovery, leveraging data from African American, UK Biobank, and H3Africa, particularly Augen. So in the paper that we published last year, so we showed that the African-derived PRS enhance polygenic prediction in continental Africa, but this still vary, PRS still vary within Africa. So this paper that we published just last week, on Thursday last week, so here I argue that African genomes holds the key. If you have been to the session that I attended in the conference, you will see me talking about, I think we are doing PRS in the wrong way. We should be doing PRS from Africa and test transferability in other populations. So why should we do it that way? Given that the high genetic diversity is in individual of African ancestry. So that is where PRS should be done that I think will be transferable to global population. So what we are doing now is a project that is called Kidney Gen Africa. So we're now aggregating all the regional data from Western Africa, Eastern Africa, South Africa, to do large-scale study of kidney genetics. So H3Africa has made it easier for me and many emerging African scientists to pursue career in genomics on the continent. If there was no H3Africa, probably maybe some of us will be selling something somewhere, but things have really changed. If you look at this slide, which I like so much, you see me in the middle there, and you see a lot of people that are giving a lot of support. I showed the Kidney Gen program. Everybody's supporting my career, making this to happen, including Dr. Rotimi, Dr. Debo. Many people from the H3Africa, at the beginning of H3Africa was part of the H3 Bionet that are led by Nikki Moda, who is there for all the amazing training they are doing. The last 5 years I've been part of the project that was led by Leon Mutesa, Rwanda, and also other colleague from USF in the US, and many people. So these people made the career to be possible. So currently, this is not the multiple effect. I have a group. I'm now mentoring 3 postdocs, 5 PhD students. I'm the primary supervisor for 4 of them, and also have many MSc and also internship. This is possible because other people are supporting the career. They made career really possible. So here, I'm so excited to say that the ripple effect is that, so currently, 2 of my postdocs are now at the last stage of Welcome Early Career Award. That's a great achievement. I hope they get through. Some of them have won other prestigious fellowship, like the RF fellowship, to other fellowship like the AVH fellowship, postdoc fellowship, 2 years fellowship in Germany. One of the postdocs is going to Germany for 2 years. Two of my students also won 4 years fully funded Welcome doctoral training just last year, starting PhD at the University of Leicester. One of them with Chisholm won 1 year fully funded Commonwealth fellowship. She's currently in the UK. 2 years that fellowship. So many fellowship. You can't be in the group and not win anything, because we train people to also train others and to be able to get a grant that can make them sustainable. So H3Africa empowered many African scientists like me and gave me a voice. For me, it is a very, very strong voice.
In the last 3 years, I've been featured in media, many interviews. As I was sitting down there, I just saw an email from New York Times asking me for an interview. Last 2 years, I had interview from many media, including BBC. So this was possible because H3Africa gave young scientists like me, give us a voice to be able to speak out our science. So also another ripple effect is the impact. Only last year, I had more than 10 invited or keynote talk. In 2022, I had more than 20 papers published, and that include paper in Nature Medicine, 2 papers in Nature Medicine, Biomedicine. I'm a guest editor to many notable journals and advisory board, and also on panel for funding. No, it was not possible for, like when I was just doing my postdoc, I couldn't even imagine that I would be on Welcome Discovery Award as a panel member, but this has been possible because H3Africa gave this to be possible. So H3Africa made peer scientific collaboration between researchers within Africa possible. And Nashi mentioned this, I found it very easy to collaborate with people within Africa. So if you look at this paper, you see name on the line. These are people within Africa at Nashi and Nayo, so it's easier. This paper in the conference that we had, I've been referenced and referenced again. This was led by people of my mid-career in Africa. The many other paper, this one paper also, we just gathered young people within Africa. This paper was published in Biomedicine. You see, it made collaboration easier with peer. This is another paper. You see also again Tinashe's name coming out. You see Kenneth Ekuru from Uganda coming up, people also of mid-career are the same. Another paper here, you see Tinashe coming up here. So all this happened within just 2 and 3 years. Another paper, you see Tinashe coming here as peer. So I'd like to just stop here by saying that partnership, peer collaboration is important. It's good to collaborate with everybody in the US and in the UK, but we must remember that there are a lot of brain within us and we can leverage that. And why I find partnership within the region very important is because I can call Tinashe anytime. I can send a WhatsApp. I can't do that with Michelle Ramsey. I can't do that with Dr. Rotimi. It makes things easier. You can push things much more harder with a peer collaboration instead of trying to push with very senior people. So in summary, H3Africa program enable large-scale genomic study in Africa population and contain parachute as research. H3Africa has built capacity and infrastructure. We've added and over and again. Most importantly, H3Africa has produced new global leader in genomic study, and I am here very proud to be one of them. Thank you.
Wonderful. Okay, so we have time for 1 or 2 questions. Can I see any hands? I think everybody's just elevated by that optimistic note. So that's a great note to end this part of the session. Oh, sorry. Do I see? Sorry.
Yeah, thank you. That's a very wonderful journey, and I'm very happy you are pushing young people. I also believe in mentorship, and I try to push people in my lab from undergrad, and some of them have gone on to even study their PhDs. So my question is, I think someone asked that earlier in the morning. As a young early career scientist, I'm a postdoc now. I'm trying to find my feet. What major advice can you give me and some of my other colleagues here? And then secondly, I like the journey you've had with the other colleague. Is it possible for mentorship for people like me so that I can tag along? Probably I'll be the 3rd generation.
Okay. So the first one, in my group, every Monday morning, so I do a motivation for about 1 minute. And what I said to them this morning is what I would say. So what I say is that in your career or in life, there will always be obstacles or there will always be opportunity for excuse. So there are 2 ways to overcome that excuse. So for example, I have this table in front of me. It's an obstacle for me to move to the next level. If you are very determined, there are 2 things you could do. The first thing is that you push the table away and you move. Second thing is that you just walk around the table and you move. What many people do is that they stay there and they keep on complaining. So in your own case, you either push the table or you move around the table.
Thank you.
Thank you very much. So now for the second part of this session 2, which is on H3Africa Fellow Speed presentations, I pass over to the chair, Rolanda Julius.
Good day, everyone. I'm Rolanda Julius, and I'll chair the session today for the Fellow Speed presentations. May I ask the following fellows, Alfred Sekagiri, Esther Assen, Fusieni Kane, Nora Nganewe, and Vincent Ntuijok to please come to the stage. Thanks. Right, it's my pleasure to introduce one group of the H3Africa Fellows that will be giving their speed talks today. So I will call on, well, before I give that, they will all speak for 3 minutes each. And at the end of all the presentations, there'll be a combined Q&A session. So not to delay any further, may I please call on Alfred Sekagiri. So Alfred will present to us the role of minority HIV-1 drug-resistant variants on virological failure among HIV-1 patients on DTG-based regimens.
Thank you. Good afternoon, everyone. My name is Alfred, and I'm here to talk about an overview of my project. I'm a H3Africa PhD fellow in bioinformatics with Brecker at Makerere University. So fast forward, HIV drug resistance is an issue, and it hampers the success of antiretroviral therapy. And what we are trying to investigate is what usually happens is that there are these variants in the viral population of HIV patients, which are not very easy to detect using the current technology, which is Sanger sequencing as recommended. What happens is that when these are not detected before initiation or switch of treatment, they kind of dominate the population all the way during treatment, and they sometimes cause treatment failure. We are trying to investigate that in the Ugandan population and to try to see their impact. So with the preliminary data that we have looked at so far, we have identified a bunch of drug resistance mutations. One of the key results that we have is that at a prevalence of at a detection threshold of 1%, we see over 80% prevalence of these mutations in subtype A and B, subtype A and D, which are the predominant HIV subtypes in Uganda. And of note, we see 3 times more risk of treatment failure for patients who harbor these drugs before treatment initiation or switch. So as part of this study, we have formalized our analysis procedure. And so far we have published in Bioinformatics Advances the next flow-based pipeline that is used to analyze this data. And we are trying to create a web server equivalent of that. And with that, I would like to thank H3Africa and my mentors and everybody I'm working with. I'm so grateful to H3Africa. Thank you.
All right, so next we will have Esther Assen. The talk, please. Thanks. So Esther will present to us my inspiration to research on vitamin D genetic polymorphism as a potential biomarker for TB diagnosis.
Thank you so much. So I'm just talking about my background and motivation of getting into genetics. It was during a training on results interpretation, data analysis in TB and malaria, when I visited an immunology lab and genomics. Then I developed passion for immunogenetics. Then I took time to look at the background of TB, and I looked at the sanatoria story. Then I tried to explore the involvement of vitamin D pathway in the disease. So there is need to distinguish between active TB and latent TB. So the latent TB that has always been ignored should be looked at early enough to be detected in order to prevent active TB. And the role of vitamin D genetic polymorphism is important in immune control in order to prevent TB. So my achievement in this journey of studying my PhD, in 2019, I was one of the award winners of H3Africa meeting, and that improved my CV. From that, I obtained 4 different fundings, one from the World Bank and another from NIH to complete my PhD research. Now, it has also helped me to get network in bioinformatics, one from a professor from Rhodes University, another one from an H3Africa fellow. I was able to get in touch with some colleagues from National University of Korea. Then through the nature classes of last year, I was able to meet with nature editors, and that helped me to improve on my writing. I was able to write 3 papers. 2 of them are under preprint, and they have been revised. Another paper is yet to be submitted. Sorry. Anyhow, I wanted to show you that I was able to complete my studies and graduated just 2 weeks ago. I've completed my PhD. And through my PhD journey, I was able to train and supervise 3 undergraduate students to completion, 1 master's student who is about to complete. And I've also been able to begin grant writing. I've written about 2 grants where I've written with some colleagues, and I'm a copier in two running grants. And I'm also trying to write another grant right now. Now, there is one popular question being asked now, what is next? So I'm looking through to see how I can go continue with my genetics work, use through a postdoc, and hopefully I'll continue mentoring other people. Thank you so much.
Thank you so much, Esther. So next I call on Fusieni. The slide, please. Thanks. So Fusieni will present to us the genomic surveillance of SARS-CoV-2 variants by nanopore sequencing technology in Mali.
Good day, everyone. My name is Fusieni Kane. I'm a PhD student in bioinformatics. It is my pleasure to talk about genomic surveillance of SARS-CoV-2 variant by nanopore sequencing Mali. As you know, the SARS-CoV-2 is at the origin for COVID-19 disease. So it is important to know that many countries has found it difficult to implement this genomic surveillance. Hence, this objective in our studies to characterize the temporal spreading and the molecular evolution of SARS-CoV-2 variant in Mali using the nanopore sequencing technology. So we selected the samples with CT value less than 30 and collected in 2021 in Mali. So the next Arctic standard protocol is used for library preparation, and the bioinformatics pipeline you see from bioscaling, demultiplexing to phylogenetics assessment. So we sequencing 150 genome with coverage, mean coverage, over 97%. So for the phylogenetic analysis, we found the Delta variant where the majority according for more than 80% in variant circulating, and 55 and 29% were clustered respectively under 21E delta and 21G delta. So the clade 21E delta was constituted by 74% by lineage B16172, and we see that for this graphic, the distribution of variant according by a number of nuclei. We think that the nucleus in 2021 in Mali is potentially related by a Delta variant. So in conclusion, we can see that the SARS-CoV variant in Mali in 2021 predominantly by a Delta strand, and the implementation of nanopore technology show a cost-effective and rapid approach for the investigation of full structural mutation in SARS-CoV-2 samples. So these studies show characterize the genetic diversity and viral strain and different lineage circulating in Mali, and we think that may be deployed for monitoring and over pandemics outbreak on the country level. So thank you for supporting and thank you of my mentor and a colleague. Thank you.
All right, next I call on Nora. All right, so Nora will present on the recent increase in low complexity polygenomic infections and sialic acid independent invasion pathways in Plasmodium falciparum from Western Gambia.
Thank you. Good afternoon, everyone. I'm presenting part of my PhD, which is centered around the erythrocytic stage of the malaria parasite, and my name is Nganyewo Nora, PAMGEN PhD fellow. The erythrocytic stage of the malaria parasite is a very crucial stage because this is the stage which is associated with morbidity due to malaria and the transmission and multiplication of the parasite in the host. Also, the parasite uses multiple pathways to invade the red blood cells, and the molecular interaction involved in this process is still remain complicated, and the use of these alternative pathways is still heterogeneous, hence the need for continuous evaluation and study of this pathway. So we hypothesize that intensified malaria control interventions and declining prevalence in the Gambia have resulted in a selection of parasites with the most efficient invasion pathways and ligand expression. To test this hypothesis, we used 3 approaches. The first approach was an invasion experiment wherein we treated uninfected red blood cells with different enzymes targeting different pathways. And then we labeled these red blood cells added with infected parasite from different individuals, and we cultured for 48 hours, after which we checked for reinvasion using a flow cytometer. We went further to genotype the infected red blood cells from the people for MSP2 gene, which is a very polymorphic gene in the malaria parasite. And finally, we checked for gene expression for 6 ligands using RNA extracted from the Chison's culture parasite. These experiments were done with samples collected across 3 years, 2015, 2016, and 2021. And we found out that the parasite mostly uses sialic acid independent pathways for invasion, because we realized that the enzymes that were used to treat this pathway were highly inhibited for invasion by the parasite. And secondly, in figure 2, we are showing the genotyping result, wherein we realized that the multiplicity of infection using the MSP2 approach was increasing across the year, while the heterozygosity was decreasing. And finally, in figure 3, where I am showing the expression profile of the 6 genes, we see that the last gene, which is RAH5, involved in sialic acid independent pathway, was highly expressed, and likewise CLAC2, which is a cytokine gene. And finally, we concluded that there is an increase in low complexity polymorphic infections, and this suggests that clinical isolate of P. falciparum in the Gambia are switching invasion mechanism from sialic acid dependent to sialic acid independent. This is because previous studies have shown that the invasion pathway was sialic acid dependent, and we therefore recommend continuous molecular and cellular surveillance of parasite, especially as the Gambia moves towards elimination. I would therefore like to acknowledge PAMGEN, my supervisors, my research institutions, and H3Africa for the training so far, which has enabled me to carry on with my PhD up to this level. And the training has also enabled me to gain additional funding through the UNESCO for Women in Science Fellowship. Thank you.
Thanks so much, Nora. So next I call on the last speech talk for today is Vincent Nduwijak. Right, Vincent will present to us low parasite infection density and high allelic frequencies of MSP2 in Plasmodium falciparum isolates from pregnant women across the different malaria ecozones in Cameroon.
Thanks. Good afternoon, everyone. I'm Vincent, a PhD PAMGEN fellow from the University of Boya. I'll be presenting on the low parasite infection density and high allelic frequency of MSP2 in P. falciparum isolates from pregnant women across the different ecozones in Cameroon. Malaria remains a huge problem in pregnancy, and P. falciparum, it's the most dominant species in sub-Saharan Africa, and the high complex genetic diversity of this parasite might contribute to the failure of malaria control measures. And this may jeopardize the effectiveness of acquired immunity to malaria in the concept of malaria in pregnancy. So knowledge of the genetic structure of this population will give an insight to the pathology of the parasite infection and epidemiology of the disease, with implications in control and prevention. So with this study, we looked at the epidemiology of malaria in Cameroon across 5 different ecozones and the genetic structure. We recruited 3,313 pregnant women who consented for the study with ethical consideration from competent authorities. Blood was collected, used for aridity determination MP, and we also did TBS, which we extracted DNA for molecular analysis and MSV2 was genotyped and analyzed using capillary electrophoresis. So interestingly, we had a very high prevalence of P. falciparum malaria compared to the other species. However, there was a high prevalence, but very low parasite density in the population, with over 80% of the positive cases having a very low parasite density. We equally computed the allele frequency of the study population and compared with the non-pregnant population, where we had no significant difference. We had the multiplicity of infection and heterozygosity, which we equally compare with the non-pregnant population, and there wasn't any difference. But interestingly, with the multiplicity of infection, with areas with a very low complexity, we had very low parasitemia, we had very low complexity. So we therefore concluded there is a low parasite density and high allelic frequency of MSP2 in the study population, with slight variations in multiplicity of infection, though a constant HA value, and there is no significant difference in the parasite structure of the pregnant women and the non-pregnant population. We recommend more markers as well as the neutral markers to be analyzed and compared with these results. I acknowledge my supervisors, my affiliations, PAMGen through H3Africa for the opportunity. Thanks.
Thanks so much, Vincent. So that's the last speaker. Are there any questions? So the floor is open.
Very nice talks, all of you. So rather than asking each of you a question, I'm going to ask one question that anybody who wants to can answer. So I'm actually impressed with the variety of technologies and techniques that you all used, but is there one genomic technology or resource that you wished you had in doing your work that would have made it easier?
Do you want them all to answer, Jen? Jane, can you please repeat the question?
Is there one genomic technology or genomic resource that you didn't have access to that you wish you had that would have made your work easier?
You can press the button there.
Okay. One thing I said for my transcriptomic work was to use single cell sequencing approach. I am in the 4th year now. Thank God we have it in my institution now, but it's almost my finishing time, so maybe I carry that. But I really wanted to learn and to do the single cell approach for my transcript.
Thanks. Barry, you had a question?
Just a comment. I remember the talks at the early meetings of H3Africa from the junior faculty and students, and they were not very professional. I just want to say the talks we've heard today and the presentation of the slides is absolutely top professional, and you're all to be congratulated on putting together a great talk.
Are there more questions? No. So Segun, just wanted to say something.
If you have been a trainee in the last 10 years and you have benefited from H3Africa, can you please be on your feet? Can you just stand up? You've benefited from H3Africa in the last 10 years. Can you please stand? Just all this, please. I would like all the PIs, before we start clapping, we want to say thank you to all the PIs all the senior investigator for amazing work that you've done the last 10 years. You don't know what you have done. And that's why I was showing a ripple effect. Because in Africa, when you train one person, it's like you are training multiple people. So all the senior people, please sit down. We want to stand up and give you a standing ovation. I'll keep on clapping for you just to show our appreciation. Please clap for all the senior people. Young people, stand up, please. Young people, stand up. You are a trainee, please stand up and give a standing ovation and keep on clapping for all the senior people. Keep on clapping, keep on clapping. Keep on clapping. Keep on clapping. Thank you very much. God bless.
All right, just a few announcements. I just want to thank all the speakers, the speed presenters. They were sticking into time, so that was really wonderful. And I just want to add that lunch will now be served, and we ask that you please reconvene at 2:15. Thanks.
Huh? Hi, which guy? Hi, Francis.
Morning. Good afternoon. Oh, excuse me.
Please hold on. We are trying to sort out the audio, then I'll be able to hear you.
All right.
Okay, now I can hear you.
Oh, you sorted it already.
No, so we are running about 15 minutes late. So, yeah, so it means that we will delay you for about 15 minutes, but I'll try as much as possible to get them in the room. And I'm also looking at if it's possible, would you want to go first before Claudina Lutz?
No,
Because
I'm fine. I'm fine with the order. I think the order is probably important for other reasons. So I've got a little flexibility on the far end. So I think we'll be all right if it's only 15 minutes behind.
Yes. Okay. Yeah, so that's fine. I'm on standby and I'll let you.
So it will still be the case that this session will need, it looked like you gave us a little extra time just because
Yes.
It was going to start at 1350 and end at 1500 hours, but there was not that much time for Claudina and Tom and myself. So as long as we are finished by 1500 hours, I'll be fine.
Oh, okay. That's fine. Okay. Nice to have you here. There are kind of multiple email communications trying to get to know whether I'm getting a recording or I'm having you present live.
Yeah, I'm sorry I can't be there in person. COVID kind of spoiled my Cape Town plans.
Oh, sorry about that.
So I'll turn off my video and I'll mute myself and wait for things to kick in.
All right.
All right.
That's fine.
It doesn't go. Okay, so we were held up with
Yep, 1, 2, 2, 2. Yep, 1, 2, 2, 2, 3, 1, 2, 2, 3, 4, 1, 2. Yep, 1, 2. Yep, 1, 2, 2, 1, 2. Yep, 1, 2. Yep, 1, 2. Then 1, 2, 3, 1, 2, 2, 2, 2. So I'm gonna post this. Okay, I just wanna post post. Post.
Yeah, 2. Yeah. 1, 2. Yeah. So I wanted to test that like then. Yeah, 1, 2. Mic check. 1, 2, 3, 4, 5. Yeah, 1, 2. Is it good afternoon? Make quick, I need to present my presentation. Yeah, 1, 2, 3, 4, 5, 6. This is a mic check. Lunchtime now. Yeah. 1, 2, 3, 4, 5, 6. Yeah, 1, 2. Yeah, speaking far. Yeah, 1, 2. Yeah. 1, 2, 3, 4, 5. Mic check. 1, 2. Yeah, it's good. 2, 2, 2. Huh? Yeah, 1, 2. Just a little bit high, but yeah, that's good. Yeah, 1, 2, 3, 4, 5. Yeah. Yeah, 1, 2. Hey. Oh.
Yep, 1, 2, 2, 2, 2, 2. Yeah. 1, 2, 2, 2, 2. Yep, 1, 2. Yep, 1, 2, 2, 2, 2, 2, 2, 2. Yep, 1, 2. 1, 2. Yep, 1, 2, 3. Yep, 1, 2, 2. Yep, 1, 2, 3, 4. Yep, 1, 2, 3, 4, 5. 1, 2, 2, 2, 2. Yep, 1, 2, 3, 4. 1, 2, 2, 2. Yep, 1, 2, 3, 4. 1, 2. Yep, 1, 2, 2, 2, 2, 2, 2. Yep, 1, 2, 3, 1, 2.
Give me a slide. Hi. Do you have Tom's slide? So apparently confidence loaded it in the shared drive. I'm not sure. I'm just trying to track him and confirm. Is it there? Okay. I'm coming in.
Yep. Yep, one, two. Yep, one, two, three.
Good afternoon, everyone. We're going to get started. We have a busy and tight schedule for this afternoon. So again, we will try to keep everyone on time. So welcome to session 3 for H3Africa. I'm Philip Awadala. I'm now part of the IEC. I'm from the University of Toronto, and I'm the National Scientific Director for the Canadian Partnership for Tomorrow's Health. Today's session 3 is titled Vision and Strategy for Genomic Medicine and Health Research in Africa, as well as big picture funding perspectives. Our first speaker, I'm very happy to introduce, is Glaudina Lutz, who's the Director for Health Innovation at the Department of Science and Technology in South Africa. So welcome, Glaudina. Yes, they should be coming up.
Yeah, thank you. Welcome to our guests from the non-South African guests. It's good to see you. The last time I spoke at the H3Africa meeting was in Kigali. So it's quite a time ago. Then we had COVID in between. So I will just take you through what we are doing locally, and then what we are implementing in South Africa is in the collaboration with the South African Medical Research Council. So I know Rizwana Mia is here and she's actually our project coordinator. So in South Africa, we've developed a new system of 10-year innovation plan for South Africa. And for health innovation, we specifically looked at the component of support for research, what we need to put into place, therapeutics, diagnostics, and devices. And then the most important component is actually the utilizing your fourth industrial revolution interaction of things to actually start developing products that will work for Africa, but utilizing those technologies. As you can see at the bottom, it is the combination of nano, bio, cogno, and IT that's combined, and see how we can then actually use it. So the three most important areas that we're focusing on is development of new treatment and prevention technologies, but within that is to see how we can utilize these convergence of technologies. Then digital health, because that's such an important component to have a better understanding of your patient, and for the patient to also have a better understanding. And then lastly is actually precision medicine as such. So in order to do the precision medicine program, we had to really start concentrating on what is needed. So a strategy and framework for precision genomic and precision medicine was actually developed in South Africa. And those are the kind of elements that we looked at: what is needed, what legislation is what is impairing what we are doing, then your sort of what are the breakthroughs that we need to look at, where do we can build capacity. And so we actually then unpack that, all of it. I'm not going to go into detail of that, but the framework is available. And if you need more, you can actually then ask Rizwana as well to share it with you. So what is important, we start looking at the important component is the ethics, to understand what is going on, and the regulatory environment. And also importance, we look specifically not only at your material transfer agreements, but also your data transfer agreements and what is involved with that and what is required from us. We're also working with the African Centers for Disease Control and Prevention, that the data sharing agreement that we will utilize will also be then made available from them. But I must say, we actually copied a lot from H3Africa. I think you've done a wonderful work, and it is now just to make it adapted to the locals in the local areas, then to take it further. The next component specifically is then your infrastructure that's needed. And for that, we need to look what sequences do we need, but not only that, the biobanks that we need, and then take it from there and make sure we can say we are going to do precision medicine, but if we don't have the toys, forget about it. And this is what we need to look then at the whole thing. Skilled people then, in order to assist them then to do what is necessary. And then the three components, genomics and proteomics, data science and health systems, and how they actually then converge to how we utilize in the information that we get from your samples to find better solutions for our people. So the main aspects that's actually is looking at the systemic issues, and this is why we're concentrating on now to put these agreements in place to make sure that the universities actually know what they need to do, then to look at the R&D programs that we need to put in place, and then to utilize that knowledge of the African genome to start looking at how can we make it better treatments available or better diagnostics and utilize it for that. So we're looking at cost-effective diagnostics, and then the changes that's necessary in the health system. So when you start looking at normally precision medicine, the idea is it's not for something for rural areas, but our specific idea what we are following. We want that point of care diagnostics that's based on genomic information. So how can we combine it, but also keep in mind at the same time, your access to the internet to send the data somewhere, but also then to make sure that the medicine that's available is appropriate as such. And then, so that is a stepwise system that we are busy developing. Unfortunately, with the onset of COVID, some of the activities is a little bit on the back burner, but we're still sticking to this plan on where we are going. So just to give you some kind of idea of what is available right now, the infrastructure that has been built, driving genomics on the moment is specifically what we call the distributive platform for omics, or short diplomics, where they actually looked at first at equipment and then to looking at what training facilities need to be made available and what bursaries or training grants needs to be made available in order to build the people. We've also got the platform for metabolomics at Northwest University. We've also got Siri and CRISP, and I think they were quite, through the COVID pandemic, they were quite renowned. They got South Africa banned twice from the rest of the world because we actually got good scientists and we actually spread the information and we were punished for it. So we've showed that we can do it, but now we utilize the knowledge that's there and the sequences that's available, and then to take it further from that. We also have at the CSIR, Council for Scientific Industrial Research, they're also looking at precision medicine, but also from a more industrial component. How do you take it further? What is needed to develop the necessary panels and so on? And also at the same time, look at how to make it then available for your rural areas as such. And at the Medical Research Council, we've got what we call SHIP, the Strategic Health Innovation Partnerships. That is actually where most of our funding is channeled through. And then there's also a Genomic Center that was created. And I think it's something that we can really be proud of. So on the moment, in a lot of the genomic abilities is being centered here in Cape Town and environment. And this is something that we now need to look how to build in the capacity in the rest of the country as such. There's some capacity in KwaZulu-Natal and some capacity in Gauteng, but how do we make sure that this whole network actually works for us? And then lastly, we've actually got an entity called SAPRIN, and I see Kobus is here. They did population studies, but it is a vast network that is actually available that we should and could tap into in order to do more population-based genomic surveys. So that is what's available. So this is the kind of infrastructure that we are putting together. Now, the Technology Innovation Agency, they're in the middle. We use them to fund some of our programs where we actually then take the information from universities then to actually look into creating small companies and what's needed for that. One of our major one of our major programs that we are involved with is H3Africa permit. And in the analysis that we did beginning of last week, one of the strong actions that was set is the actual H3Africa network. And it's now we need to start looking how to integrate that what H3Africa is doing and that what the EU Africa permit group wants to do, because there's not enough space for everybody to have its own little claim, but how we can actually then start integrating the activities and how can the one build onto the other one. And how can the fact that EU Africa is prepared to look at infrastructure, the training and so on, how can we then, as the African component of H3Africa, how can we utilize those going forward? So we did a gaps analysis for that. And I think this is also relevant to H3Africa, is what we found, what do we need to do, what are the things that we need to concentrate. And this is not only for South Africa, this is actually as part of the Southern Africa network of EU Africa permit. And this is the areas that has been identified that we need to start looking at and how we can start sharing the information between countries and taking that forward. I'm not going to read it into detail. It is the slides can be made available to you. So yeah. And then the main thing that you are talking about, that they are talking about, is funding. How do we actually make sure we have the necessary funding going forward? So one of the things that we from South Africa's side has created is actually a funders forum where we brought together the major funders of health research in the world. So you start looking at Wellcome Trust, Bill and Melinda Gates Foundation, NIH, and it just carries on. And we even discovered once what we didn't know, that is funding specifically genomic work. And what we are doing is bringing the funders together and to get them to a point not to duplicate funding, but let's rather look at the systems approach, what is needed where, and then decide who's going to fund that. The whole discussion around genomics with the funders is one of the next areas that we're going to start tackling with them. And then have to really convince them why it is necessary within the African context to also invest in infrastructure and not just clinical trials and getting the data, but actually to build up the capacities as such. Then the areas of interest that was identified by the EU Africa Permit group was specifically your pharmacogenomics is very important, and we need to see how we can look at, but not only use it for your rare diseases, but your infectious diseases. And one of the programs that we're actually funding is to look at on HIV and the impact of your genomic diversity on the outcomes of specific of the medications or treatments that they use for HIV. And that is, for our context, is very important because we need to understand why do we have treatment failures, and need to start having the system in place to go deeper into understanding treatment failures. And this is where your data from your patient record, then combined with genomic information, will be able to make breakthroughs and say, but this is actually how it works. This is a different way that we should be doing. So we also looking at specifically at your system of data banks as well as biobanks that needs to be put into place, and then how to maintain them and manage the access to that and the outcomes of that. So that is all part of what we are busy putting together. Now, approximately a month ago, we signed an agreement between the South African Medical Research Council, the Department of Science and Innovation, and the Innovative Pharmaceutical Association of South Africa. They are a chapter of iPharma. So we do have this agreement. And one of the things is that we can also start looking at how do we utilize the information and the expertise of South Africans then in the bigger research projects, and to make sure that the big pharma projects are actually relevant to what the treatments that should be available within Africa, and then to start talking about the cost and funding around that. So all of that we're busy putting together. And what is important is that, yes, we need your input to say, these and these and these are the pain points that needs to be addressed, and that we can collectively, from the government side, with the academia, start looking at what is the best way forward then. And that's, thank you.
Thank you very much. We're going to have to continue on to our next speaker, who is Thomas Kariuki. And Professor Thomas Kariuki is the Executive Director of the Science for Africa Foundation, which is a not-for-profit Pan-African organization that aims to support, strengthen, and promote science and innovation in Africa. Thank you.
I'm assuming I have some slides to share, but thank you for the opportunity. I guess I'll just focus a little bit on the kind of work that we want to do across the continent. As a Pan-African organization, I may touch all over the place about how we want to move forward. What did I do? Glenn one, yes. Let me start there with a big congratulations to the H3Africa community of researchers. So much has been said over the last few days, not just here in the context of H3Africa, but also in the context of ICG. But anything that one picks there, that speaks to how much this community has actually achieved. So for me, it's just to start with a huge congratulations to all of you for what has been achieved so far. In terms of the Africa's genomic context and the way we see it ourselves is to the fact that the lack of understanding of disease etiology in population that is limited in a strong way for support of health research or medical training. The 3,000 ethno-linguistic committees, communities across the continent is a problem, inequalities, issues, implementation, all of that remains, I think, a context for us to keep in place. And of course, to leverage what has happened in the context of COVID, in the context of the role of current funders, current proposed new funders who may come into that. And I think, she talked about the EU-Africa Permit and the proposed African population cohorts and the longitudinal population studies that needs to come into all of the discussions moving forward, because we are all thinking about what next, what is happening next. And I think these are all the context that is important. I like this from Eric Green reminding all of us that and I think a lot have been said about this lack of diversity. So we will continue talking about this because this is also part of the next phase that we should be discussing, that if we just limiting everything to research with populations of European descent, then that means that we are not talking to the rest of the rest of the world. And therefore, that is why researchers around the world are working to include people from all kind of different ancestral backgrounds. So in terms of our interpretation, there are some missing links in all of this. We are missing out on the potential benefits of whether it is understanding the disease causes or looking at early detection and diagnosis, and how we are looking at the rational drug design and improved clinical care. And that, of course, has been part of all the discussions. So I want to say a little bit more about how we see that future and what our modest contribution to all of that would come into play. This has been nicely outlined in the paper by Zain and Guido, and I think that really is a very compelling reading for somebody who wants to, whether it is we're engaging other people who we want to bring into these discussions, even for us as a Pan-African funder, because that is what we are, we would actually be sending out this kind of information to say, hey, this is the vision that is coming out from the African researchers themselves, looking at how that future would look like. Now, but who are we? What do we do? So we have been known by different names in the past. We have been engaged with H3Africa for the last 7+ years. We were going by the name of a platform called IESA platform. We are now an independent public charity, the Science for Africa Foundation. We are based in Nairobi, in that beautiful green office that you see there. We support scientists. We focus, like all of us, the important things: quality science, data, knowledge, evidence, collaborations, whether they are intra-Africans or global partnerships. So we are looking at strengthening the entire ecosystem, and we are looking at how we engage policymakers throughout. So we are driven by the fact that strengthening African science for a better future is our vision, and we want to go from science to impact. But importantly is the fact that we are very much connected with through our strategy of the role of genetics and omics moving forward, because we want to focus on making investments as they become available into discovery, in transitional aspects, and how we move across the entire value chain of implementation and policy. So yes, health predominantly, but also we must always say we live in very difficult period of times. This is climate change, it is devastating us. And all these things, including food security, are actually interconnected. And so healthy people, yes, that's what we want, but hungry people are not healthy. And so we have to also make sure that this interconnected, our health is linked to our climate and the environment, and our food security is all part of that interconnectedness that we must keep an eye on that. So our core business is we convene, and by convening we bring people, dialogues, African scientists, they sit, they tell us what the priorities are. As a Pan-African organization, we are calling from everybody from every corner of the continent. Our footprint is actually very good. And those experts then come up with what the targets should be. We design programs, and by designing, we mean we deliver programs designed to address critical gaps in the science ecosystem in Africa. This is a good emphasis for me because a lot of discussions have been around who is defining the agenda. And I think I want to make sure that we are making sure that whatever it is that we fund is actually coming from these conversations, and it is not being driven from outside the continent. So funds will be available, but we will deliver impactful science in the continent for Africa's development. And of course, it comes with a big requirement for accountability, which means guided by principles and standards and the best metrics one can put together. So we have some opportunities that I would like to dwell upon because these opportunities may not necessarily be just a transitioning of where we are, but I think it is a nice connection to what could have some very good collaborations emerging out of that. H3 Africa, developing excellence, leadership training, and sciences in Africa, has been one of our most successful flagship program across the continent since 2015, with great support from Wellcome and with the UK government, FCDO. And this is now phase 1 concluded. We are getting into phase 2. Now, phase 2 will continue the same format. So direct funding to the leaders themselves, directly from where we sit, where the money is flowing is from Nairobi to wherever else that this money is going, to any corner of the continent. Between $4 and $5 million for each of those networks, and there'll be 14 networks. So we are talking at $70 million that's going to be available for this over the next 4 years. That would allow the recipients to design their own programs and projects. They will invest in the infrastructure, the equipment. They will get into recruitment of fellows and trainees, and they can upgrade their own institutional as best as they can. So we are not restricting this to research consumables. It is going into the broad strengthening of that environment. The focus remains the same thing that many of you have been working on. HIV, TB, malaria, NTDs, bacteriology, pandemics, viruses or related, and for the first time also getting into supporting networks that are very focused on something like cancer and neurosciences and climate change as well as part of this discussion. And the development of all diagnostics and all the other products that we want to get out of this. More work is required for NCDs. I think for non-communicable diseases, we know, for example, that we have this silent epidemic that is going on, but you have been addressing some of that. And I have mentioned climate as part of our area of interest. And some of you are co-PIs in deltas, and so many of you there are some of the leaders here who actually are very, very familiar with deltas. All have been products of deltas, or however you have moved across the various networks. The second area that is important is an opportunity for pandemic sciences collaborative, which we are in the making with our partnerships. Now, this will focus on everything that we want to see and to prepare ourselves for the next, hopefully never, pandemic. But genomic surveillance, vaccine R&D, clinical research, public health will be part of that call. And that means we have 2 areas of focus, 2 pillars that actually will be available. Now, first one is these epsilons that you're calling them, epidemic science leadership and innovation networks, hoping there could be between 4 and 7 such networks that will be funded, depending on how we end up with the available funding for it. Support a cohort of independent African leaders. That is the whole idea, so that we end up with world-class research enterprises that can also drive innovation, maximize the benefits that we have with experts who can then predict, they could be preparedness, it could be things that will support over the next 20 years. And then we promote research and solutions and the mitigations that need to come into the risk of epidemics, and enable also the creation, just like with Delta, just like with H3Africa, we are all trying to produce something that speaks to government to say, science actually produces high-level jobs, high-value jobs. So all of that is put into that. The other strand of this will be a knowledge exchange that will provide opportunities for exchange of people, ideas, enhance science policy engagement, and enhance this as an educational driver and a tool as a generator of income, like I've just mentioned. Now, the other opportunities that we are exploring and hoping that they will be available in due course of 2023. And again, they speak to some of the strengths from many of you. Drug discovery development, we are still struggling with building other centers in Africa that actually can do good drug discovery work beyond what Kelly Shibale and the team have built here. So we're looking for other centers that actually can emerge in future that are drug discovery and can position themselves and become eventually globally competitive such centers. We have a call for non-hormonal contraceptives, and we have a genetic infertility call that we hope we can go into. Clinical research remains our area. Now, just going into very briefly, we have this establishing of genomic centers of excellence that Nikki mentioned. And I am hoping that we will have an opportunity to build this collaboration that is truly Pan-African for what could come. And that means that we need all the key players that are going to be available to us. Now, beyond this, we are also interested in not just the human pathogen. The other opportunity we have is with non-human pathogens. There is a group of scientists called the African Biogenome Project. We want to see whether we can work with them to make sure that we end up with a call for people who work in non-human genomics work. And there's a paper if you want more information about that. Now, finally, I think for me is to the partnerships. There are all kinds of partnerships with governments, with Pan-African, with pharma, discussions here. And the heads of international research organizations is a group that sits together as funders. Don't look for a website. They don't have a website, but they are a very powerful group. And I think we want to say that we really want to make sure that we move forward together. So my concluding remark is the gratification that has happened over the last so many years, the global consensus, we still don't have global consensus on the next steps. The major benchmarks that you have designed yourself are there. There is a pivotal question around what approach would offer the greatest yield in what we want to do. That could be another African Genome Initiative and longitudinal LCS. And then, of course, we want to make sure that we end up with infrastructure better. Early applications would require that we do all the things that you have talked this morning, and the barriers remain our problem. Genetic services, you need to push this to the government. EHR and point of care, that is easy if we can do it, if we can get possible less expensive point of care available for our value proposition for government, that would be great. And I like the idea also thinking about how we can build small biotechs and there's bigger biotechs that could come out of Africa, even as we wait and hope and engage with the big pharma for us. Cross-cutting issues have been addressed again. Pursue government. Yes, please, let's do that together. All of you are citizens, scientists, so we all do everything we can do together, and not just looking at one organization to do this. And then hoping the programs we will put out, we will be looking at attracting, rewarding nicely so that people are not coming and then going back to where they come from, and the retention measures that we want. Data governance, I think, was spoken, and there are examples of that. So, and that's my final slide. And I was, I didn't know that they were going to play this when I was sitting here over lunch as a background music, because I love this, all of us being dreamers together. For those of you who are not old enough to know who John Lennon is, I propose you go look for that song. It's beautiful, because this was in the 70s and 80s, but I know, imagine, and I'm paraphrasing here, imagine if all the genomic public goods are available to every clinic in Africa, but we are not dreamers, and we are not the only ones. And so hopefully we will get there. So thank you for the opportunity.
Thank you very much, Thomas, for coming and presenting the vision for the SFA. So we have a hard stop at the top of the hour, so I'm going to go to a very quick introduction. I'm very pleased to have via Zoom from the United States, Francis Collins, who's currently is the scientific advisor to the White House. He's an MD, PhD, who's served as the NIH director for the 3 past administrations. So I'm going to be very quick and turn the floor over to Francis. So thank you, Dr. Collins, for joining us today.
Well, thank you very much. It's a privilege to be able to join this session and to say a little bit about my admiration for what's been accomplished by the H3Africa Consortium. In fact, if I might propose that we could consider H3Africa could add another H at this moment after this amazing 10 years. Maybe we could now call this H4Africa, because the other H would be historic. This really has been a historic 10-year enterprise. Kind of like the idea of H4 Africa, because it says it is for Africa. And that was always the intention by African scientists focused on African scientific challenges. So I mostly want to just say thanks to all of you. I want to recognize the partnership that NIH was able to establish right at the beginning of this project with Wellcome. And Tom has already gone over some of the remarkable achievements that made H3Africa a successful enterprise over its 10-year tenure, and has produced a vibrant and productive scientific community. Maybe a few things to point to as lessons of why this, in fact, did achieve things that many skeptics really didn't expect to be possible. It was a network. It was a real consortium. The whole was greater than the sum of the parts. The awards for funding were made directly to African investigators, which was not the tradition in many instances before this, to ensure African leadership and autonomy. There was a focus on infrastructure, which has often been ignored in the past, building up the capacity for genomic analysis and IT analysis and training modalities. All of the things which, when you consider one-at-a-time projects, oftentimes didn't get much attention and didn't have any sustainability. There was collective attention to ethical issues, consent for data sharing, data generation, and data processing. And there was a possibility, because of the way in which H3Africa gathered momentum, that additional funding and new academic positions emerged. And of course, then, with emergent health challenges requiring genomic tools, the Ebola outbreak in 2015 and COVID in 2020 and continuing on. That's why I'm not with you, because I am just now, for the first time today, testing negative after now 16 days of dealing with COVID here at home. H3Africa did also provide the confidence for other bold African projects to get mounted based upon that track record. And the Data Science Initiative, DSI, is just but one example. And Tom Kariuki has mentioned multiple others. So you should all feel really good. You have accomplished amazing things. 30 countries, more than 500 members, 201 workshops, 51 projects, 690 publications, over 114,000 research participants. The development of H3A Bionet is a remarkable component empowering computational genomics. So yes, after an amazing decade, I think this is now H3Africa, historic. But what next? Certainly the preceding speakers in this session, Claudina and Tom, have asked that question and have suggested some possible answers. Sustainability is an ongoing issue, and I know it has probably been pretty frustrating to H3Africa participants that NIH has this 10-year limit on the support that we were able to supply through something called the Common Fund. But the Common Fund is NIH's venture capital space, and things have to turn over in order to be able to allow new projects to come on board. And so many years ago, it was decided that projects in the Common Fund needed to have that 10-year horizon and then needed to evolve into something else, be picked up by some other source of funding. Frankly, personally, I wish that was not the case in this instance, considering that H3Africa has been so successful. But it is an opportunity to think about how to take this to the next level. And this is where I've been particularly excited about the proposals that have been talked about in the course of ICHG and with a number of other satellite discussions, the possibility of Africa Genomics Centers of Excellence, a bold proposal building on the lessons of H3Africa, but proposing an extensive expansion of what might be feasible with the design presented at this meeting earlier by Nikki Mulder and Christian Happy, now very much attached to a detailed business plan, which includes a coordinating center, but it includes a number of centers of excellence and a hub and spokes kind of model, and would be hosted by the African CDC. At least that's the tentative plan. The challenge, of course, is to find the funding for this, because this plan is bold. It doesn't start immediately on day one. It starts with the coordinating center, but then ramps up over the course of a couple of years to bringing on board the centers of excellence, and seeks to obtain something like an $80 million annual budget by the time it is fully fleshed out and extended forward for at least a 10-year period. That is a great business plan with an exciting potential. There was a wonderful meeting yesterday with representatives of industry, led by Mark McCarthy of Roche Genentech, indicating there is very serious interest on the part of industry in being a supporter of this, and in a sensitive way that would basically provide infrastructure support, would not give any kind of special access to data, which of course is a very sensitive issue for industry investments in Africa. But it was a very good feeling that was developed during the course of that meeting about how this could be done in a practical way. And it does sound as if industry is quite interested in the plan and potentially willing to consider a significant contribution. But where else will the funds come from? That's where GenCo, as we're now calling it, needs to focus in the course of the next month or two, exploring possibilities such as the World Bank, exploring where would NIH come forward here? Is it possible to design a new kind of common fund project that could support some aspects of GenCo? Because I believe this is the natural evolutionary next step for trying to build the capacity for genomics across the continent. So much to watch there. And I can tell you that with Nikki and Christian's leadership, but much boosted by others like Charles Rotimi and Ambrose Wonkin, I'm certainly personally very engaged in trying to do everything I can to help this take shape, even though since I'm no longer the director, I don't have any real authority, but I have, I guess, some ability to get my phone calls answered. So we are determined to try to make this opportunity into something truly significant and exciting. So I just want to conclude, because I know we're at time, but I want to conclude by paying personal tribute to the audience of investigators from H3Africa. I had the privilege of serving as NIH director for 12 years in 3 administrations. And I can honestly say one of the most gratifying aspects of that experience was to see the wave of scientific innovation emerging on the continent of Africa, supported by H3Africa and other projects, but driven by a generation of young, entrepreneurial, and globally competitive scientists. We are in such a different place than we were in 2009 when H3Africa was first envisioned and then launched in 2011. I believe this will be historic. This project will serve as an enabling foundation for future precision medicine applications across the continent, linking genetic variation and differences in gene expression, the individual disease susceptibility, and the development of novel and early interventions against both common and rare diseases. This provides the foundation for an agenda for Africa by Africans. And I will always be grateful to all of you who came alongside in what many people thought was a pretty bold and potentially fraught endeavor and made it happen in a fashion that is now much the source of admiration across the world. But it's just a beginning. And if we're really serious about the kind of empowerment that Tom Kariuki was talking about, where genomics and its precision medicine applications are fully developed and fully available to people across Africa, we have much to do. But we're on a great trajectory. And I think the way in which H3Africa has initiated this effort now some 10 years ago provides a great deal of confidence of what can now happen going forward. So H3Africa, I thank you, and I now pronounce you H4Africa. You have been for Africa, and you have done remarkable and historic things. Thank you all very much for the chance to speak to you.
Thank you so much for those remarks, Dr. Collins. And it's great to hear how H4Africa has been a springboard for some of these new initiatives that we've got to hear about today, and we will be hearing about over the course of the next couple of days. So we're moving straight into almost a lightning round of people from the private sector. We're going to be talking about how mobilizing the private sector can enable science and health activities in Africa. So our first presenter is Craig Kinnear from the MRC, from the South African Medical Research Council Genomics platform. And so, Craig, are you here? Yes. Okay. Okay. Sure. No. Okay. So, okay, come on. Are you Bridget?
I am Bridget.
Okay. So
From Illumina. So
okay,
thank you, Craig, for swapping out. I've got to run to the airport. Good afternoon, everybody. My name is Bridget Mohali from Illumina. I'm incredibly proud this moment to be standing here beside you. You know, our partnership with H3Africa goes many years before I even had the dream of working for Illumina. So, I mean, we've been talking about the need to increase diversity to improve precision medicine. Who am I to emphasize this, right? But what I really want us to discuss now is how to move further. And what I mean is that there's been a lot of discussions, iterations, publications around the need to not only increase diversity, but how do we equalize it? And by equalizing at Illumina, we're talking about how do we improve access? Many times when we have conversations about access, I hear price. Price and price. But at Illumina, we're quite obsessed about the entire ecosystem. And what I mean is that how do we make it available in the first instance? You know, when I travel and I speak to many of you, there's always conversations about, Bridget, it cannot take me so many weeks or so many months to have my technology, your technology in my lab. So we've been having a lot of conversations, not only with Illumina internally, but also with Africa CDC, in that how do we really remove barriers to access? Because what is that technology when it cannot deliver what you plan to do? And when we talk about availability, we also need to talk about adaptability, okay? Because then again, you know, how do we make it adaptable? When is it going to inform public health? And when we talk about adaptability, we also have to talk about affordability. And it's not a conversations that we are scared to talk about. We are very open. I think you know that at Illumina, we've partnered with H3Africa for many decades, and we are immensely proud of what you guys have been able to achieve over the many years. I don't have to tell you what, I've heard a lot of conversation this morning on how you've partnered with Illumina around our Infinium platform. So the question is that, how do we move the needle? And you can say, if you look at this map, you can see that there isn't even a blue or a pink dot in Africa. And at Illumina, we are incredibly, on a daily basis, talking about how do we fast track that access of Illumina technology. There've been an enormous investment in terms of our supply chain and operations. I hope many of you have been able to come to the Illumina stand during ICHD to really interact with the newly launched NovaSeq X. The question is, you know, we are really looking into how do we expand. And through our collaboration with H3Africa, we have seen a continual access of Illumina technology throughout the continent. In 2018, we have the first eye scan in Cape Town. 2 years later, through your support, through your immense commitment to improving genomics in Africa, we are now sitting with multiple eye scans and multiple Illumina technology throughout the continent. And in order for us to further remove barriers to access, I am certain 3 years ago, you would never know anybody that is actually in the continent from Illumina. Yes, we have an array of channel partners, and 2-3 years ago, you probably knew 1 or 2 people from Illumina, and you probably had 2 interactions with that 2 channel partners. Now we are very proud that we have at least 11 people based in Africa serving you from a commercial and a sort of a commercial and after sales perspective. We also increased the number of channel partners that today you're interacting with in countries. The whole idea was to be able to give you quicker response and to be able to serve you better. So few take-home messages, and I apologize that I will not be part of the panel later on, but perhaps I stop talking, take 1 or 2 questions if you don't mind. So that I'll be able to, because Gerald said to me, oh, is that a trick now that you'll be running away to catch a plane? So I'm very happy to stand here, take some of the questions. I don't promise to answer all of it, but I'll be able to take 1 or 2 questions. So what are the take-home messages from my end? That, like I said, we are a bit obsessed in unlocking the power of diversity in genomic data. We have the technologies to support all your large-scale genomic sequencing projects. And yeah, we have different sequencing platforms that will be able to support your different projects that you'll be embarking on. And that we are committed to increased presence of Illumina in the continent. As you can see, we've launched the NovaSeq X to support all large-scale genomic sequencing opportunities. And I don't know if you're aware of Illumina's newly launched infectious disease portfolio. And thank you to COVID, we were able to enable your labs to be able to sequence a whole array of infectious disease. So let me pause here and try to take some of the questions. Sharon? I saw your hand first.
Thank you, Bridget. Just very simple question. There's been a lot of infrastructure building in Africa in the last few years, from 2018 to 2022. So from business perspective, you are trying to set something because people are going to pay for the genotype and all that. What is Illumina doing in terms of maybe training or education or support for African scientists, apart from trying to to make the machine available and genotyping at a cost?
So, I mean, I spoke about access, right? And as I spoke about being made available, adaptability. So part of that strategy along adaptability is to be able to support skills and I mean, people talk about brain drain, a number of scientists not able to retain and work in the continent. And part of us talking about adaptability is exactly that. How do we partner with, for instance, Africa CDC? You know very well about the PGI network. Part of the network is to be able to support and train young scientists so we are able to really expand on the critical mass. So there are a lot of initiatives that we are partnering with throughout the industry. Yeah, so the idea is to really be able to do a bit of tech transfer. In some of the centers that we partner with in the continent, we are doing exactly that. But there's also plans, right, which unfortunately I cannot talk to about on my own today, but you will be able to read through them. So there is a number of initiatives, and the most important one that you probably know about is our work with the PGI network.
Bridget, thank you very much for that presentation. Superb. My name is Raj Ramasa. I'm head of human genetics at the University of Cape Town. We run a large medical genetics service and also run a very strong research environment. So we've benefited very much from international funding, for example, H3Africa, and we could, through those technologies, access Illumina and the other technologies that are available to us. But the reality is that the costs that are involved with regards to genetic testing, something as simple as panels of genes, are so prohibitively expensive, whether it's from Illumina or Thermo Fisher or whatever. Literally, I don't think we're making any difference with regards, and I'm saying this with the greatest of respect, and I appreciate the work you do. But the reality is, without NIH funding and international funding, we are unable to use the technologies for genetic services. We had Invitae coming to South Africa 5 or 7 years ago, and literally offered absolutely inexpensive genetic testing, which is fantastic for us in the clinical genetics environment. Mm-hmm. But literally to the extent that it wiped out local developments with regards to molecular diagnostics, and now suddenly they're gone because it was not a sustainable business plan. So I hope the message is getting across that unless you seriously engage with not funding NIH funded grants, but literally locally affordable solutions, we're talking pie in the sky. Again, with the greatest of respect. Thank you.
I do appreciate your question. I mean, I guess we can, well, maybe another question. We can talk about how Illumina has been involved in reducing the price of sequencing, right? I mean, if you talk about the last 15 years, you've heard our CEO, Francis, talking about, our vision to really get to that $100 per genome, and we are getting closer to it. But locally, in terms of, the challenge, one of the challenges that we're still looking, there's still a number of samples leaving the continent, and it has really become an issue around scale. Yeah. I've been really inspired in the last week, hearing a lot about doing big genome sequencing. I believe this is the time to now start saying, how do we bring together the critical mass to be able to reduce the price even further?
There were a lot of hands that went, is this mic going on? There were a lot of hands a moment ago, but they've all come back down. I have one quick question, if I may. There was a lot of fanfare for the new NovaSeq last year in the US. When will be the first? I know they're still being delivered to a few select locations in North America and Europe. When will that new NovaSeq X with its new pricing be potentially available to African research?
I know that there's been some numerous requests for it, and our ALS is that The quicker we can register it through our regulatory, the better. For now, as we stand, we are looking into Q4, if we're looking into regulatory processes in the continent.
Okay, great. Thank you. All right. We can thank Bridget once more.
Thank you.
Our next speaker is Junaid Gameldian from Roche Genentech. I don't know. Pardon me. Sorry. Oh, oh, sorry, you swapped. Okay, now I got it. Okay. So this is Craig Kinnear from MRC Genomics platform. Sorry about that. So.
Thank you. Thank you for the opportunity to talk to you about the SAMRC's genomics platform. I won't take up too much of your time, but if you forget everything else that I said today, all I want you to remember is that we are here. We are available to do sequencing, and we want to partner with H3Africa. So any sequencing project that you have, please consider the SAMRC genomics platform as well. Okay, so our genomics platform was established as a partnership between the Beijing Genomics Institute and the SAMRC. And part of that partnership was to expand precision medicine in South Africa. But we also wanted to make sure that we can make sequencing as affordable as possible. We want to make sure that we can keep the samples collected in Africa, and also to improve the access to sequencing in South Africa. And so we partnered with the BGI, and this partnership, was signed into the agreement on the top of Table Mountain in 2018. And we had the grand opening of our platform on the 29th of July, 2019. So as part of this agreement, the BGI trained 4 of our scientists in China to work with the MGI workflows and the MGI instruments. And when we were ready to do our first sequencing, we also, a field application scientist from the BGI came to South Africa to help us do our first sequencing runs. The agreement also then allowed us to place a MGI sequencer in our laboratory, as well as a liquid handler to do our first sequencing runs. And in turn, we purchased all our kits and reagents through MGI for these instruments. The SAMRC then also had to build the infrastructure and provide the staff for training on these instruments. So we built these laboratories and we split them sort of into the DNA and the RNA workflows. And we had separate laboratories for quality control and for sequencing as well. So we've sequenced our first 6 human genomes. That was the 5th of July, 2019. It's lots of fanfare in the laboratory at that time. I think my daughter was also there. She took off the day from school to come and look at this happening. And we then detailed these first genomes in the publication in scientific reports, just to show that we are able to produce high quality data, as you can see from the image. But we also wanted to make sure that we are also able to produce this data consistently, which we do show. And we also show that we are able to produce good quality data, sometimes even better than the data that was produced in China on the BGI, on the MGI instruments as well. So this was important for us because we wanted to make sure that we always have this issue that the perception that what we're doing is not good enough. But this again shows how well we were able to do the sequencing. And this is sort of a common theme that we see in South Africa. We are excellent in producing good quality data. So those first 6 genomes that we sequenced in 2019, and then of course, towards the end of 2019, COVID struck China, and China shut down its borders. We were unable to get any instruments, any reagents, any consumables into the country, but we made a plan. And as you can see from 2019, year on year, we were able to increase our sequencing output quite significantly. So it's almost an exponential increase in our sequencing capacity over the last 3 years. In 2019, we offered whole genome sequencing for humans and animals, and we also offered some transcriptome sequencing. And as the years went by, we've added some more sequencing modalities to our repertoire that we can offer. And we are now in a position where we can sequence the genomes of multiple species. We also part of the Africa Biogenome project that was mentioned earlier, where we are sequencing some plant samples. We sequenced the genomes of animals. I think the most exciting project that we did was sequencing the genomes of wild dogs, looking at their genetic structure and their inbreeding coefficients, and just also to show how these animals move around in packs and how interbreeded those packs are. We do whole exome sequencing now. As part of our repertoire, we do RNA-seq for a variety of species. And then we've also added some whole genome bisulfite sequencing. And as we've increased the type of sequencing that we did, the number of samples increased, we also had to increase our staff. So where we started off initially with only 2 research technologists, I think our center, our platform has grown quite significantly, where we're sitting now with 6 technologists. And even though we're still quite small, we are expanding quite a bit. And then we also had to increase the number of sequences that we had. So initially we only had the 1 sequencer, but as the work came in and the more sequencing were being done, we needed to increase the number of sequences that we have. And then again, through our collaboration with BGI and the MGI, we were able to add 3 more sequences, actually 4 more sequences, 1 small DNB G400, and this is what we use to run our South African Medical Research Council's wastewater surveillance program, where we sequence SARS-CoV-2 from wastewater samples collected from across the country. And then we've also added another 3 G400s. So one of the extra ones that have been placed, and we are expecting the placement of the next 2 within the next few months. We've also started doing some long reach sequencing, and we've recently acquired an ONT GridION to do our long reach sequencing as well, and we hope to offer that as a sequence service as well. Then, of course, we also have the automation ability. So all the work that we have done now, up to now, has been manual, but as we've increased our output, we've added some more liquid handlers. So we now have 2 MSP 100 liquid handlers and 1 960 that can do 696 samples preparation at one time. But we've added another one as well just to keep up with demand. Our data processing, so all the data that we process via the Center for High Performance Computing. Data is transferred from our sequencer securely to a primary server at the SAMRC. We also have backed up offsite, and then again transferred securely to the Center for High Performance Computing for processing. We also have a big capacity development arm as part of the SAMRC. Part of our mandate is also to develop capacity, and we work with many institutions across the country, many tertiary institutions, to mentor postgraduate students. We also a key national training research resource center for training of NGS technicians. Like I said, we host postgraduate students and postdoctoral fellows, and we also host workshops for bioinformatics and sample preparation. So we believe that our platform will play a key role in the SAMRC and South Africa's response to the 2022 WHO guidelines. So we believe that we can promote genomics through advocacy, through the number of samples that actually go through our laboratory. We believe we can be a key driver in the NGS market. And so we have opportunities to engage with people like Illumina and ONT, MGI. So we also have we're also a key national training service and training facility, like I said, and we are embarking on some more programs where we want to broaden the access of sequencing to many of the other under-resourced institutions in our country, and this we were doing in collaboration with Diplomics and ONT. And then, of course, like I said, we also want to be collaborative. We already are part of the Diplomics network, and we see that we can be a good asset to H3 Africa as well. And with that, I want to say thank you.
Thank you, Craig Kenny. So I believe our next speaker is Junaid Gamildian. I'm not sure I'm pronouncing that properly, but and Junaid is at Roche Diagnostics Cape Town, and he's the head of bioinformatics, and has been a member there since 2019. So thank you for coming.
Thank you, and I'd like to thank the H3Africa Consortium as well as the organizers for the opportunity. Maybe just want to take a minute to mention my background. So I am fairly recently from academia. I do have some history with H3Africa. We had, as SANBI, the Bioinformatics Institute at UWC, we held a BioNet grant where I was co-PI with someone you probably know a lot better than me, Nikki Tiffin. Before that, at least straight out of my PhD, I spent some time in industry, and that was in the pre-genomic era. And I just wanted to maybe say to the young scientists here that you don't need perfect data to build something. We were quite successful back then. So think creatively about how you can build something while you trying to build your career, because that's how Africa becomes sustainable. So with that, let's see if I've got the right one. So here we go. I just want to talk a little bit about around this African proverb. It's used very commonly in motivational talks by big motivational speakers, but it's African, and there's quite a lot of wisdom in this. So about 3 years ago, Roche embarked on what we internally calling the One Roche African Genomics program. And we were really mandated with looking at a way to bolster African genomics in Africa, by building on top of what was already there. So we spent some time, quite a lot of time, mostly listening, asking questions, engaging with African scientists and institutes and institutions, as well as Pan-African bodies. And as part of that process, we've designed and invested in research projects with African scientists on the continent, multiple areas in 10 different countries. We started a scoping exercise about what the future data requirements and data management requirements will be on the continent. And that's really about, sort of starting with the end in mind. And we've engaged with existing consortia, H3Africa being one of them, but also African biotech. And then we've started developing relationships with Pan-African and global institutions like the WHO, Africa CDC, and the African Union. And then something that's very dear to us, when we engage with our leadership, they always advise us to look for opportunities to make a difference sooner rather than later, not when the data is there one day. And so something that we very carefully have been thinking about has been building programs, fellowship programs, and internship opportunities for African scientists. And we've actually come up with a concept as well of reverse internships. So where we could bring a Roche scientist or scientists into a specific lab or institution where multiple people can benefit from such an interaction. And then we've also been in discussions with funders like Wellcome and the NIH to see how we can multiply their efforts and amplify their efforts. There we go. So we are really interested in co-creating a way forward for genomics in Africa. And H3Africa has proved that partnership and collaboration and networking is the only way that it will actually work. Notwithstanding the size of what the effort will be, but it's just that with synergy, we go a lot further. So we wanted to know where we could best support through partnering and collaboration. And as I mentioned before, we had a lot of engagement, and we've really listened more than bring in any preconceived notions. And we came out what we thought was a good starting point, as represented on this slide, to the areas that we believe we should be contributing towards, so that we can catalyze genomics in Africa. And I'd really like to point out African leadership and talent support. The two actually go hand in hand. So we really believe that for something like this to be successful, and actually all the initiatives that have been mentioned at the previous conference and this one, there has to be leadership on the continent, and that must be at all levels, from governance all the way down to the people that actually lead the science. And obviously, to get there, there needs to be support for developing talent, and equally importantly, retaining talent. And so we believe that by engaging the community and listening to Africa, where we let Africa tell us what Africa needs, that is where we can produce the highest impact. And obviously the intention is to fill the data gap that currently exists on the continent. And yes, I may as well just address the question. All industries will benefit from this if the data is as open as possible. But at the same time, if the data is made open in a way that preferentially benefits Africans, whether it could be embargo periods or things like that, then in the end, everyone benefits. And I'd maybe just like to call out the possibility of industry and academic research collaborations, and not us only being funders, only being data producers, and coming on later when we have to translate findings to products. And I'll talk a little bit about that in the next slide. Oops, one back. So as one of the strategies for trying to bring this into reality, we've started talking with other industry partners about building an initiative for advancing genomic research in Africa. And we're calling that initiative PAGRA. It's intentionally meant to be pre-competitive collaboration. And it's really to facilitate engagement between industry, African scientists and institutions in order for genomic research to really take hold and expand in Africa, with the ultimate intention of being able to translate findings into genomic medicine. I'm not going to go into the slide too much, but it really details a few areas in which industry can make an impact to African science. And one thing that we are actually quite keen to contribute to, and actually to make accessible as quickly as possible, are those foundational genomic data sets that we will need to build new reference genomes, new reference SNP sets for variant filtering, for better imputation of GWAS data, etc. But beyond that, we also want to facilitate more clinical studies on the continent. We want to contribute to improving public health. We definitely want to contribute to improving infrastructure on the continent. And that's everything from the infrastructure required to go from sample to data, as well as the systems, computational and other, that will make the data shareable and equitably accessible to African scientists. And what I mean by equitably accessible, it's no use data being open if African scientists don't have the compute resources to be able to process said data. So that's just one of the examples. And I already mentioned training fellowships and internships. And then in that last column, that's quite aspirational, but we don't believe that industry should be the only entities that translate findings into eventually genomic medicines. And it can be a lot sooner, right? So it could be reagents, that's a product, diagnostics is another product, but also medicines, right? So we believe that by supporting what one could call a biotechnology incubator type ecosystem, that we could get those smaller companies that translate findings made in Africa to products right here in Africa. So as far as PAGRA goes, the reason that we started talks around PAGRA is that it gives us a better way of communicating via single point of contact to African institutions and with potential African collaborators. And with that improvements in efficiencies, alongside that, we also ensure that we don't duplicate efforts. We are aware of other partners or other industry partners that want to do things that are very similar to what we want to do and have the same philosophies about sharing, about building in Africa, doing capacity development as an example, that makes us believe that we should actually be working together. So some of the areas that we really want to ensure are result from these partnerships would be, number one, to accelerate research. And to do that, we need to build clinical data sets. And then we need to expand data access to Africa, as well as access to infrastructure to be able for Africans to make use of said data. And thereby we will be addressing unmet needs, but these are needs that Africa decides are important. And so really we will be aiming to partner to expand the impact that initiatives like H3Africa has already been able to make. And so to do that, we're making a commitment as Roche to invest in research and development in genomics on the continent, as well as genomic data science and bioinformatics and all the associated sciences that brings that to reality. And maybe just to very quickly go back to that proverb, we believe that working together, we can go far, as the proverb says, but we can go in the current era, we can go far, fast and faster, I would say. Thanks for the opportunity.
Thanks very much, Junaid. Very interesting. Our next speaker is Carrie-Anne Pillay, and her talk's title is An Introduction to the Simply Spectacular Team of Separations.
Thank you. Good afternoon, everyone. I think everyone's getting a bit tired now, so I'm going to be as brief as possible to go through the slides. So as mentioned, the topic is Simply Spectacular, an introduction to the separations team. So I'm just going to give you a high-level overview of separations, as well as highlight some of the agencies that the company represents. Okay, so the first question is, so I joined separations a couple of months ago, and I'm not sure how familiar everyone is with separations, but I think that most of you know the company quite well. But separations is Promo Lab trading as separations, and it was founded in 1989 by Paul Quasar. And today, both his sons are involved in the running of the day-to-day business. And in the beginning, the business was basically geared up to serve the scientists that were in the chromatography field. But since then, the company has expanded and moved into multiple divisions, including Chrom Techniques, Biotechniques, Water Techniques, Biopharma Techniques, Robotics, and the newest division, that is now diagnostics. Sorry. Then some of the key agencies that separations represent, and this is just a couple, just for the purpose of this particular meeting. But I think the biggest one is Hamilton, Illumina, Molecular Devices, MN, and Takara. And these portfolios are basically very much linked to automation and biobanking, and as well as next generation sequencing and microarrays. The additional supporting techniques is obviously the extractions, purification, qPCR, PCR, normalization, as well as aliquoting. So I want to take a little bit of time to talk about the Hamilton BIOS. So during the course of today, we heard a lot about biobanking. And the Hamilton BIOS is a -80 automated refrigeration system. Sorry, I'm suffering with this horrible indigestion that's making me cough. So as I was saying, the Hamilton BIOS system is an automated -80 storage, sort of comparable to chest freezers that we have. So this system basically allows for temperature stability of your samples, and it's flexible in terms of how much of storage space, of how much the system actually stores. So it's able to store anything from about 100,000 samples up to 10 million samples. And the key features of this is the -80 universal tube picker, which uses the Hamilton robotic system. The sorting, it allows for the sorting of samples, managing inventory, processing and delivering of the samples, controlling sample access, which means only certain individuals will have access to the samples that are put into the system itself. So it's not sort of anyone can access your precious samples. And also there's a full audit trail of the temperature logs for the samples that's in there. So the integrity of the samples is continuously monitored. So we are proud to have the collaboration with the Biomedical Research Institute repository institute in Stellenbosch, where the first system was actually installed. This was a purposeful built institute where it's basically meant for repositories of samples that serve both the academic and private customers. And it has the capacity to actually store 3.5 million samples, and it can be upgraded to a bigger system to 3.5 to 5 million more samples. The system itself obviously has the backup generators, backup UPSs, but also it allows for the nitrogen, have a nitrogen access as well in case the backup systems do not work. The unit is connected to a LIMS system, and in the LIMS system, you obviously can control and you log all of the activities that happen on with the samples itself. So with separations, the next one big agency that we represent is Illumina. And in 2020, the portfolio was acquired, and in 2023, separations was awarded the elite channel partner due to their good business that they brought in. And up to date, we have the install base of sequences for about 83 sequences within the country and sort of Southern Africa. And the install base of the eye scans is at the moment 3, but we're hoping to grow that number as well. And from separation side, we basically want to commit to providing exceptional service and sales support to our Illumina customers. With H3Africa, as Bridget mentioned, the array itself, the array is run on the iScan, and it allows for the processing of 8 samples at a time. And in a week, you're able to do about 1,700 samples on the iScan itself. CPGR and Illumina. So an iScan is housed at CPGR at the moment, that was donated by Illumina. And the system itself has been automated with the TCAN robotic instrument that's able to increase the amount of processing capacity of the instrument itself. Illumina is committed to collaborating with researchers within South Africa and other countries in Africa to bring samples through CPGR for processing. Researchers that we are working with at the moment include Prof. Michelle Ramsey, Prof. Wonkum, and Prof. Sigan that we've seen today already. And then also CPGR does have a sequencer, which is the NovaSeq 6000 that was installed in 2022. So as part of the collaboration between Separation, CPGR, and Illumina, I think that we basically want to work together to support the H3Africa samples to be processed via CPGR, and for Separations to provide this on-point sales and service support going forward. Thank you. I'm so sorry, I'm so out of breath. I think this baby is just giving me a problem to breathe. Thank you.
Thank you, Carrie Ann. Our next speaker is Lyndon Mitnall from the Regeneron, is Executive Director at Regeneron Genetics Center, which is part of Regeneron Pharmaceuticals. Thank you very much, Lyndon.
All right, thanks everyone. So first I'd like to thank the H3 Africa Consortium, say happy 10-year anniversary. And I guess I should say happy birthday to H4 Africa. So I'll tell you a little bit about the Regeneron Genetics Center and our commitment to diversity and genomics research. Regeneron Genetics Center, or the RGC, was established about 9 years ago. So H3 Africa is actually older than us. But we've turned into one of the largest sequencing operations in the world. And we are a fully integrated genetic center. We have a goal of making genetic data medically actionable and use that genetic information for both drug discovery and development. We set up a lot of collaborations around the world. We have over 130 collaborations in about 20 countries. And we provide this genetic resource to our academic collaborators. And that helps with a lot of opportunities for funding for them. And, we're 9 years old, so we did a lot of work in establishing the Genetic Center at Regeneron. And now we have a huge commitment towards using genetics to better understand biology and specifically in making more broader global relationships in understudied diverse populations. So I don't need to tell this audience about the lack of diversity in genetics research. But we do have to increase this pie so everyone has an equitable piece of the pie. And the RGC is trying to do this. We're working, as I mentioned, across the globe. In this simple map, the red shows the countries that we're working with already, active collaborations, and the yellow are those countries that we're talking to. And as you can see, with 2% of the genetic information coming from people of African ancestry, we are having a huge effort here in Africa to try and increase that piece of the pie. and speaking with, again, over 20 countries here in Africa. And not only to increase the diversity of the genetic data, but also, as many folks have said today, and consistent with the H3Africa Consortia, we aim to support community engagement, student education programs, research capacity and infrastructure building, as well as building those African ancestry-specific databases. I heard at the ICHG meeting last week when it kicked off that we're all Africans. We're all Africans, right? And we have a lot of Black Africans even in the US. So being a product of a historically Black college, Benedict College in Columbia, South Carolina, just to give a shout out, I have a heart towards, a lot of HBCUs in our country. And I think a lot of the things that we're talking about today here in Africa also, it has a lot of similarities to those schools in the United States, where there's not a lot of educational opportunities, not a lot of pipeline programs for Black Americans. So I like to view it as a African ancestry, all Africans, all in issue. And we have to address these concerns, and it will take a collective effort because there's so many things that we're going to have to address. And we touched on them today, and I know we're going to have a panel discussion to talk more about this. But community trust is very much needed in the United States, Black Americans, as well as here in Africa. We need to inspire and mentor our students early in their educational path to go into the STEM fields, to build on that STEM workforce, build institutional capacity, as we've been saying, with bioinformatics, research, and et cetera. We need to build these diverse cohorts. And at the same time, we have to deal with and address a lot of the issues around ethics, the policy and legal, diagnostics and clinical validation of a lot of variants of unknown significance, and also make sure that we provide access and affordable access to healthcare, because I'm hoping we are going to be successful. We will be successful in studying these diverse populations, and therefore we should have programs that give back to the community for community benefit sharing. So lots of things to address, and I think it's going to take a collective effort of relationships and strategic partnerships between academia, healthcare, tech, government, philanthropy, biotech, you name it. It's going to take all of us to address these issues and solve the problems. So we've done this before in some small way, and I mean we as the pharma industry. We have come together to solve some problems. These problems were smaller, but it does serve as an example of us coming together. These companies listed here all pooled resources together to get the UK Biobank sample sequence through the Exome Sequencing Life Sciences Consortium. Again, a consortium of companies coming together to solve a problem. And we made this data publicly available, where now researchers like you could get access to UK Biobank data. And by us coming together and just talking together in a room can spark other things to happen. So that's why starting and coming together and starting and talking will spur other opportunities. And we did that in this UK Biobank project. We spun off, or started, I should say, a totally separate initiative called the Proteomics Pharma Proteomics project, where we did proteomics on 62,000 samples from the UK Biobank. So, and now this group is grown. You can see it went from 8 companies on the left to about 13 companies on the right. And we can continue to do this and do it in a focused way here in Africa. So I do think these are examples of pre-competitive merging of resources from biopharma to support research opportunities for the general public. So we're trying to do that again. We're gonna, we've been talking, Regeneron and a lot of pharma partners, and many of those here, about a new initiative that is focused around our HBCU, Black Americans, as well as Africa. And so that's why we're here, because we do want to go far by going together. And we're starting discussions and getting close to, taking this concept to something real here, where we're going to be, partnering with communities in the United States, Black communities in the United States, as well as here in Africa, to empower and educate our communities about the value of genetics and how that affects your health. We're going to have programs designed where we'll inspire and mentor students through STEM programs. We're going to diversify the genomics database by performing large-scale African-specific genomic studies. And we're going to support that research excellence in these institutions here in Africa and in our HBCUs by investing into student and faculty research. And last, all of that should therefore expand the STEM workforce across all STEM disciplines, not only genetics, but it should definitely increase the amount of geneticists and genetic counselors and medical geneticists that we have here. So we're here today to hear from you on how this initiative will be able to help you and your organizations, and how we could be a part of or make this initiative a part of what you're doing. So with that, I'll say thank you. And my email address is here if you want to hear more about this.
Thank you, Lyndon. Our next speaker is from MGI, Haider Barbosa, who I believe in his portfolio includes Europe and Africa. Introduction to MGI and patented state-of-the-art DNB-seq technology.
Okay, thank you very much to the organizers for this kind invitation. We really appreciate it to be part of this meeting and in this opportunity also to share. As previously mentioned, it's important that the private sector is also involved in this, and I'm sure we can contribute to your mission. And we're very proud today to be here sharing a little bit about MGI and what MGI can contribute. Let me see. Okay. I'd like to start with this slide because it shares a little bit of a vision of MGI. We leading science innovation. We are a relatively new organization, but I hope with this, at the end of our presentation, you can also see how we're innovating. Sometimes solutions can come from the non-obvious players, and I think we do have a role to play in this field. MGI is just one part of a big group, BGI Genomics, or BGI Group, which integrates several different companies or research institutes. And we'd like to see that our customers see us not as just a technology provider, but as really a partner that can collaborate with you and help you to achieve your mission as well within your projects. Within the group, we have BGI Research, which is the largest private institute in China. We have the BGI College, Beijing University, where we train what we consider to be the future of the NGS leaders. We also collaborate with many universities around the world and in common programs because we do think this is one of the key strategics for the whole field and ecosystem to advance. We have Gigascience, we have the Institute of Forensics, Institute of Agriculture. We are building what we consider the hospital of the future with more than 1,000 beds, where all these multi-omics and personalized medicine is integrated from scratch. And this is all part of the BGI group, and MGI is the equipment manufacturing arm, but it's definitely one of the important companies to drive all our mission as well. BGI Group started more than 20 years ago. I think many of you are familiar with BGI. We started with Human Genome Project, and since then we've been the first leading what is now very common frequent population genomics. We started these projects in China, but since then we've also been focused on de novo sequencing of many plants, organisms across different species, and we're proud sequence, the panda, the rice. We're leading in the field of single cell. And more recently, if you guys following this area in spatial genomics, we really disrupting this field as well more recently. If you focus on MGI, we're going to be 7 years old. So we very new. It's normal that many of you still don't know us. We started by the acquisition of Genomics, a US company. And since then, we've been growing exponentially. Now more than 2,000 people worldwide. We have increased the number of patents. The data generated with our instruments is more than 1 terabytes of data. And of course, all the scientific and publications that result out of it. And this is partially the reason of our success, our investment in innovation. We're really keen to focus on products that we are confident for one simple reason. All the products we developed, we also use. So we really focus on develop technologies that fits our needs, of course, fits other needs as well. And this is the reason why we, for example, when we focus on the sequencing technology, we use this technology called DNA nanoball sequencing technology, which is slightly different what you usually know from the market. Other sequence technology, basically, when they do the amplification, they do a copy of fragments. And when they do this copy of fragments, they basically propagating the errors as you know, as PCR bias. But we use a different strategy. So the DNA fragment is circularized, and then we use the Phe29 polymerase enzyme, which is the most accurate polymerase enzyme in the market, to copy the original template over and over. So rather than do copies of copies, we actually focus on copying the original template over and over. Of course, with this, we create what we call the DNA nanoball and DNB that names our instruments. But we also didn't stop there. We innovated completely how the flow cells are redesigned. We designed the flow cells with these blue spots that captures one and just one DNA nanoball. This is very important because what the result of this is that the number of duplicates are extremely reduced, but also this common problem found in other technologies that is called index hopping. So at the end of the day, you get very high accurate data that you actually use for your projects and research collaborations. And of course, with all the mission and objective to bring to the market again highly sensitive, highly precise products that can ensure the quality. This is just an example of all the products we have here. It's a lot of products. I'm not going to, of course, focus on here, but I just want you to take two messages today. One is that MGI can offer you and help you supporting you end-to-end solutions from sample primary to transfer to the plate, from the extraction and all sort of sample extraction technologies that we have, for automation of library prep to the sequencing part. So end-to-end solutions on the automation part, but also on the sequencing part. So not only end-to-end solutions, but we have solutions for different throughput, so different needs. So traditionally customers in their workflows, they need to start with a platform, then they buy another one, then they buy another one. One key advantage, for example, a G400 platform you see there, is that they can integrate several platforms. So you can start small and scale with the same platform. So you not only save on capital investments, but also you save, of course, on the sequence costs, which for us, we're very proud to say we are the cheapest and always pushing the price down, and welcome you even to visit us in our booth outside, where we can share as well some of interesting promotions and placements we might have for different regions, and we'll be very keen to grow our partnership within the consortium. But as you also see there, we have platforms for really big population genomics projects. We have what we call T10. We have even T20 that was announced last year in US for actually deliver what was $100 genome. This is not the future, it's the present. We have customers, including in the world already using or having 5 of these instruments installed in one only facility. So it is a technology you can have today. But we also proud to develop open platforms. So what you are or the libraries prep, for example, that you currently use now can be transformed to our technology. As I said, it's a different technology, but we have what we call a conversion kit. So many of these players that you're very familiar, they of course tested our technology even before announcing public agreements with us and starting to develop their own products to our technology. But what makes us also very proud in this short period of time is the collaborations we've been established with key organizations, private or public, literally around the world. But in particular, more focused in the African continent, we've been collaborating with MRC, with Professor Craig. You had a chance to hear him, and it's one of our most important customers in this region, but other customers across the continent. As I said, we're new to the market, but we are, very happy to already have proud collaborations with reference institutes that, of course, tested and bring these solutions also to the market. And this is my final slide because the way we understand our business is, as I mentioned early on, is we want our partners our customers not to see us as the technology provider, but really someone that can support them along the way. I mentioned the NGS or the BGI College, but more pragmatically is the support we can give it to you on day to day. And in South Africa alone, we have more than 5 people on technical support. There's 7 people based in South Africa, and we have more across the continent as well. But because we really understand this is how we can grow together as well. Our success depends heavily on your success, so we'll be really driven to you achieve your mission and your objectives, because if you do it, we also do it as an organization. So that's all from my side. Thank you very much.
Thank you very much, Helder. So we now have a couple of fairly short presentations. The first presentation is from Acclaim Moila, Inkaba Biotechnical Industries, Local Genomic Services Matter. Thank you, Acclaim.
Hi, everyone. I'm going to be very brief. Yeah. There you go. Okay. Good. So basically, my name is Acclaim Moila from Inkaba Biotech. I'm the application specialist as well as technical support for PEG in sub-Saharan Africa. Okay, so basically our headquarters and laboratories are in Pretoria. Basically, this is our whole team, past and present, but you can see that our footprint really stretches across sub-Saharan Africa with offices that have laboratory and fully functional laboratory in Nigeria, as well as planning to do so within Kenya, Nairobi. As we call ourselves Africa's genomics company, really. So what we really aim for basically is to provide the state-of-the-art sequencing services across the region. And this currently, as I've mentioned, we have fully functional labs in South Africa where we do Sanger sequencing as well as next generation sequencing, both short and long read sequencing technologies. And we have just recently opened a lab in Nigeria. And of course, we're planning to do so in East Africa, in Kenya. The idea of all of this is to facilitate the capacity building really in the specific areas and provide ease of access to the genomic services. As you probably know, there are plenty of platforms across the continent that some of them probably not used to fully used to full capacity. So we really see it as a need that samples that are in Africa should remain in Africa, and we should be able to as service providers, help researchers as well as other projects to really make use of this these platforms across the continent. So currently on our fleet, so we have basically a wide company that provide a wide range of genomic services, starting with we're able to synthesize primers as well as probes. We have the finger department, as I've mentioned, and we also have the targeted SNP genotyping solutions, as well as targeted methylation solution using the mass array multi-TOF system. And in terms of our NGS, we have the short reads platform, which is the NextSeq 500. And then we also have the PacBio 2E, which is our workhorse at this moment. And we use that platform to generate a highly accurate long read. Most of our projects are really run on this platform. And this is just a snippet of how we generate these hi-fi long reads. So basically you have your molecule, which is a genomic DNA, you cut it into like 25 kb long, you add the adapters, you bind and need the sequencing primer as well as the DNA polymerase. This is done outside the platform. And then once you have your bound complex, you then load it into the smart cell. And we generate this hi-fi long reads by sequencing the same molecules over and over again. And because the random are so errors, we are able to flush out all the errors when you do the hi-fi processing. And as we have seen, the method of there has been long reads, basically just reiterate the point that in all genome sequencing, we're probably going to need long read as a long read data. It's the backbone of data. And this is where I can explain more into that. Of course, our future plans, like in the next 6 months or so, we are planning to increase our throughput and add the newly released PacBio Revio. So this is just to show you that at the very moment, we are able to sequence the human genome at 30 times coverage, but that will take you about 3 to 4 smart cells. So adding the Revio to our fleet, that will reduce down to only single smart cell, because a single smart cell is able to produce up to 30 times coverage. So that should really reduce the amount of processing time as well as the cost of sequencing. And then we're also planning to add the newly released PacBio short reads to our fleet to increase the throughput and also to cater for other clinical research applications. With that, thank you.
Thank you, Claim. So slight shift in topic. Alice Matimba is going to be talking about Welcome Connecting Science, collaborative partnerships to scale up genomics training. Welcome, Alice. Thank you.
Hello, everyone. So I'm really pleased to be here and being part of the H3Africa meeting today. I just wanted to say that I am one of the recipients of the H3Africa grants, and I did also benefit a lot from my personal development. And part of the work that I'm going to present today is a way that I have also been giving back to the community together with the team at Welcome Connecting Science. So I'm presenting here on behalf of the team, learning and training team at Welcome Connecting Science. And basically, Welcome Connecting Science sits alongside the Wellcome Sanger Institute at the Wellcome Genome campus just south of Cambridge in Hingston. And we develop training for audiences, research audiences, healthcare professionals around the world, in the UK and also globally. We do run some training on the Genome campus, and so I think some of you have been there. And we also run training across different regions in Africa, Asia, and Latin America. And most of our training is really focused on genomics and laboratory-based and bioinformatics, but also covering some of the topical themes in genomics, including genomic surveillance, data sequence analysis. And also we've been working a lot to try and continue to develop that capacity through Train the Trainer, for example, and also other professional skills development. So, a key part of our program is really to train scientists that are based in Africa, Asia, and Latin America. As you can see here, since we've come back in person since November, we've already been to Uruguay. We've had a team that's just come back from Thailand. And we also collaborated with the Africa Society of Human Genetics meeting in Morocco, and also had a very successful course that ran in Santiago in Chile. So we're really excited to be partnering and working with scientists at these institutions in these regions to train some of the scientists, regional scientists. So I'm here to talk about the work that we've done with H3Africa, and it's part of the H3 Bionet with colleagues Nikki, Mulder, and Verena, and other people that have contributed to this course. So this is one of the most highly demanded courses. And I remember that the first time that we ran this course at University of in collaboration with Sydney Brenner at University of Wits, we had about 150 applications for only 20 to 30 spaces. So we decided that we could actually make this more accessible to people using the H3 Bionet remote classrooms model. So by doing that, we have actually been able to reach more countries, more people. And as you can see, in 2019, when we ran this course, we only reached about 30 people, but over the last 2 years, working with H3 Bionet, we've been able to reach more people. So we've really want to work with partners to scale up the training to make it more accessible to as many people as possible. One of the things that I've heard throughout several discussions here is that some people find that they want to get trained, but they don't have access, or it's too expensive, or they have to pay high registration fees. So, our training is free to attend, but we also want to collaborate with other regional partners or partners that are also funding training, so that we can actually go a long way and make sure that all these materials that are being developed are actually accessible in the best possible way. So here, this is actually my last slide. I just want to invite you to our stand. If you want to know more about our program, we do have a booth upstairs, and hope to see some of you afterwards. And also do come to me if you have any ideas on how we can continue to deliver training to African scientists, and also thinking about how we can have a lot more impact by partnering up on these really important development of skills for African scientists. Okay, thank you.
Thank you, Alice. Our next presenter is Cobus Hurst. There you are, who's the HRI's Director for Population Science. He also is working with us on the International 100,000 Plus Cohort Consortium as well. Welcome. And so you're going to talk about the African Population Cohort Consortium.
That's correct. Thanks to the organizers for giving me the opportunity to talk about APCC and what we're busy with and what we can learn from H3Africa. So our aims is to harnessing the genetic, socioeconomic, cultural, and environmental diversity of Africa for scientific discovery. We've heard a lot about genetic diversity, but that's not the only diversity in Africa, of course. And also, sad to say, Africa is also diverse in terms of access to care, and universal health coverage remains an ideal that is not yet realized within Africa. So we have huge inequities in the continent, and we need to do more, of course, to address that. We believe that we can unlock the potential of population cohorts in Africa through interoperability, data harmonization, and so forth, so that the unique value proposition, in fact, of these longitudinal population cohorts is that you have direct access to individuals living in communities on the ground, so to speak. And in these longitudinal population cohorts, some of them have been followed up for decades, giving us an unprecedented view over how changes have happened over long periods of time. We want to use this investment in these existing cohorts to build research capacity, to identify and fill gaps by establishing new scientific collaborations across cohorts or new cohorts where needed, and also across the whole spectrum from basic and translational science through clinical science, intervention research, and implementation research in these communities on the continent. So in this process, we want to engage a broad spectrum of African scientists, policymakers, community contributions, and other stakeholders to co-create the APCC. We started last year through funding from Wellcome Trust, Bill and Melinda Gates Foundation, and UCRI MRC. We're now in the first phase of our collaboration to establish the APC. So this is not yet the APCC, that's the work that precedes the establishment of the APCC, by doing scoping reviews and stakeholder identification. And this opportunity, of course, is part of that process to engage with the broader scientific community in Africa to help us to co-create the APCC. By May this year, we'll go into an extensive phase of engagement with the broader scientific community for them to help us to formulate what the APCC should look like and what its objectives and aims ought to be. We will go through a phase of workstream-based collaboration to eventually produce the output, which would be a blueprint for the APCC and an investment plan to get funders to assist us with the final establishment of the APCC. This is the different work streams that we are working with, and it gives you an idea of the different components of the APCC that we are thinking about, from stakeholder and community engagement, existing cohorts, and I'll talk a little bit more about that to engage them, our research vision, objectives, data and methodology, omics that Michelle Ramsey, that you know from H3Africa, is the leader of, governance and ethics, and capacity strengthening. We will also work closely with the deltas that you've heard of from Tom earlier today. So far, we are still early in this process. We've identified 87 cohorts in Africa, and there are clearly still a lot of gaps, especially in North Africa, where there are still cohorts that we need to identify and incorporate. About 50 of them are health and demographic surveillance sites. These are sites that look at complete populations in a defined geographic area, and then following them up over time, birth cohorts and other types of cohorts as well in 23 different countries. These are the preliminary criteria we've used to identify those cohorts. The final membership criteria for APCC is something that we will jointly develop with the broader scientific community, but we're looking at larger cohorts of 10,000 and more, or 2,000 in the case of birth cohorts, should be population-based. That means that the members of the cohort should have an identified place of residence, full representation of a target population, longitudinal. It could be a facility or a clinic-based cohort, provided that there's a known catchment population for that facility. We're looking for longitudinal, longer-term cohorts, at least 5 years, or intending to do so. It could be open or closed cohorts, panel studies, but not repeat cross-sectional studies. There should be clear inclusion and exclusion criteria, and we're looking at current cohorts. Of course, the APCC will also be looking at where the gaps are and where new cohorts should be established. What can we learn from H3Africa? A lot, of course. I'm incredibly impressed with the continent-wide collaborations that you've established. And this is, of course, what we are trying to do with APCC as well. Your governance structures, including data and biospecimen access. I'm a member of the H3Africa DBAC. Research capacity building, data standardization, identification of cohorts, but in short, everything we can, starting with you, the participating scientists in H3Africa, to really invite you to engage with us during this process, for you to co-create with us what the APCC should look like, so that we can unlock the potential of this incredible resource that we have in Africa, namely longitudinal population cohorts. Thank you very much.
Thank you, Kobus. Again, we'll have a couple of short presentations back to some potential industry collaborations and partners. Rochella Ramden? No. Sorry. Cora Vasher. Oxford Nanopore, the complete picture of the human genome.
Hi, everybody. So I'm Cora Vacher. I'm the Market Development Manager globally for Oxford Nanopore for human genetic. I'm going to give you a short presentation to show you that achieving the complete picture of the African genome is absolutely possible with long read, because yes, size does matter. And so the goal of Nanopore is to provide the analysis of anything by anyone, anywhere. And I think here, what's really important is the anywhere, because the model can really be decentralized and brought to the sample and the research as well. We're really reshaping biological research by providing technological solutions that now are meeting needs that have been so far unmet. We really, we have global operation. We support a customer on all continents, including Antarctica, and we're also supporting customer in space. Through continuous disruption and innovation, we're really improving the development of new markets and new application. And we're actually really starting to see that in applied markets, so more in agriculture, but also very much in clinical, where it's actually very easy to translate a biological research test into a clinical test that says with research use only instrument at the moment, but on the same platform, the same unique, single accessible platform. So we're really empowering a new genomic era. And I wanted to give two examples of where we're really driving new insight in new biological discovery. So the dark genome is basically the part of the DNA that is not currently easily resolved with high throughput short read. But that can be much better resolved by long read. And the group from Mark Ebert and his team has really put a publication to look deeply into the functionality of the dark genome in coding region. So nothing super complicated. But they use long read to look at dark region, and they identified 96 genes that are currently dark genes, so not easily identified with short read. And they've associated them with over 300 human disease. And I'm not talking super rare, super syndromic disease. I'm talking Alzheimer, ALS, autism spectrum disorder. So really, those dark genes are really hidden in plain sight and could be easily resolved using long read. Iwan Escher has done a nice review where he shows that 34% of all variant disease causing mutation are larger than a SNP, basically. And those large DNA changes like inversion and deletion tend to have a much more deleterious effect. But again, because of the difficulty in sequencing structural variant, there's lots of allele and there's lots of genomic region that are most likely to be pathogenic and are not currently being analyzed. So there's another 2 examples I wanted to give, actually 3. We're really working on some of the national program, and people start to think, yeah, but nanopore is just if you have a few samples. Actually, we're working with G42, which is a national program in the UAE, and they've just done 90,000 sample whole genome sequence with us on their population. They're achieving amazing production scale data. The 2 projects that I actually wanted to talk about are the one from Ewan Ashley at Stanford, because last year they broke the World Guinness record of ultra-fast sequencing. They did a whole genome in less than 8 hours for a clinical purpose, which is in itself quite interesting. You can tell people that's the problem on the same day. But they also identified 12% more disease-causing variant using that pipeline than their standard of care. So basically it was faster, and it was also much better for the diagnostic. The other one, I think, is because there's a lot of missing information on methylation, and we haven't heard a lot about methylation apart from Leon during the meeting, is that using nanopore, you do get your methylation and hydroxymethylation data with the rest of your data. So you get your canonical basis and your non-canonical basis is one run. Again, people can say it's not really scalable, but that's not true. The group at CARD, which is the Center for Alzheimer and Related Dementia at NIH, have just published their protocol and workflow for a comprehensive view of haplotype result variation and methylation on with the goal of doing thousands of samples. At the moment, they've got 3,000 brain samples. They're really planning to increase that. And so, you know, until recently, we haven't had a complete picture of the human genome. And the NIH identified that quite a while ago, saying, you know, by using the last 15 years of Sanger and 15 years of Sanger, and then after that, 15 years of short read, we still ended up with about 8% of the genome missing. So the telomere to telomere consortium was created, and last year, almost a year ago, they've published their first complete telomere to telomere genome. But I think what's really important is they're moving forward. So Karen Miga, who's there at LGBT, she presented her new data, and she's one of the co-chair of the T2T, but also she's really moving towards the Human Pangenome Project. And I know Neil has discussed it, and I think Nikki also said there's a lot of interest in looking into that. But Karen has shown that using nanopore only, using duplex reads, ultra long reads, and PORSI, they could do exceptional quality telomere to telomere whole genome from the sample that they were having. And they're really starting to broaden the capacity. And I think that's very much where we can come with you into our own, because we can really provide you with solutions that are scalable. But if you want to do decentralized approach, if you want to have the sequences where the instruments are, where the PIs are, because it increases education, because it increases adoption, because it makes it a lot more accessible, we can absolutely work with you on this one. Thank you very much.
Thank you, Cora. You're welcome. Last 2 presentations. One's coming from, I believe Eric's here. So Eric Vanderwald from Watchmaker Genomics. Thank you, Eric.
Thanks. It looks like I landed one of the best slots of the day. I do promise this is going to be pretty much an elevator pitch. I was only given 2 or 3 minutes, so bear with me. I'll just give you an overview of who we are. Right, so over the last few days, I've heard a lot of discussion about sharing data, controlling access to data, analyzing data. But we haven't talked that much about what goes into generating genomic data, and that's exactly what Watchmaker Genomics and I'm very focused on. So we produce precision tools for life science, as the intro slide says. And our vision is to enable the generation of data that is as accurate a representation of the information in the sample as possible. Maybe I'm not sure how much you guys will think about this, but when you go from sample to data, there are a lot of complex steps, and often artifacts are introduced, information is lost in that process. So if you think about it, all biological information is read, written, and maintained by enzymes, and we think that's at the core. And so if you can improve the enzymes, then you can improve the quality of the data. And so we've been setting out to build this full stack genomics tools capability, so a fully vertically integrated genomics tools company. That means that we manufacture our own enzymes. We're capable of manufacturing them at large scale and high quality, and specifically for genomics applications. So we think a lot of the industry that generates, provides DNA and RNA modifying enzymes are still, based in the PCR era, and they're not always looking at enzyme quality from the perspective of genomics. So we think some of the most exciting innovations in life science lie at this intersection between computational biology, biology, and engineering. We're bringing all of those technologies together to focus on developing optimized solutions for your applications. So you can see we have the high stringency enzyme production, advanced protein engineering. We use a combination of rational design, computational design, and directed evolution to develop engineered enzymes, and then computational biology to develop these solutions for the applications. And because we're fully vertically integrated, we have improved supply chain consistency, so we're not dependent on third parties for the ingredients in our products, and we can also provide very competitive pricing and custom solutions for both OEM and business-to-business type opportunities. So the other reason why I'm here today is to make sure that everybody who's in South Africa and Africa realizes that we're on your doorstep. We were founded in Boulder, Colorado, so we are an international company, but we have a research and development facility here in Cape Town, and we are busy building out international distribution and manufacturing here in Cape Town as well, which obviously also has implications for supply chain and cost in the future. In addition to those two R&D and manufacturing facilities, we also have sales teams based in the UK servicing Europe and also a team in China. Just a very quick timeline. We're pretty young still, so we were started in the early 2019 in Boulder. We started our Cape Town company early in 2020, and you can see we've not been sitting around. We've entered into a number of partnerships with new arising technologies in the genomics space. So Molecular Loop, Element Biosciences, Twist, Singular Genomics, and a whole host of others who are not publicly announced yet. On that same timeline, you can see we've launched a number of products, mostly in the NGS space, but we also have a large portfolio of single enzymes that are available to anybody who's trying to develop their own applications. So if you are part of a technology development group, either at a university or in the private sector, and you're looking for some either wild type or engineered enzymes, please get in touch. And then just a quick summary of some of the products. So you can see we've got products in the DNA library prep space, RNA-seq, NGS amplification, and we also have a uracil tolerant amplification reagent for methyl-seq. We've got a couple of products in the synthetic biology space. And then, as I said, a whole portfolio of single proteins and enzymes, both wild type and engineered. And so, if you know of anybody who's maybe still a student or finishing up their studies and looking for career prospects, please send them our way. We're looking for talented young people in South Africa. If you're developing an exciting new application or technology in the genomics or any omics space, please feel free to get in touch. We'd love to collaborate with you or provide you with some of the tools that you might need to get to where you're going. And lastly, if you're planning any large sequencing projects and you're looking for reliable, cost-effective supply of reagents, and we have, as I said, redundant manufacturing in 2 continents. Please get in touch. Thanks very much.
Our very last presentation before we bring people up for discussion here will be from Cindy Lawley, who's from Olink, and she is the Director of Population Studies. Thank you, Cindy.
So I am delighted to be here. I really want to thank Philip and Michelle for fitting me into the schedule. And Philip, I give you permission to grab my hand. If you see Lyndon doze off, please. Definitely don't want to bore Lyndon. So I'm at a company called Olink. It's a proteomics company, but what I'm going to talk about is how we're empowering the use of genetic data and insights from genetic data, like many of the projects that are being talked about here today. So I hope this will be of interest to you. I certainly don't need to tell this crowd about the enabling of insights in that being able to collect DNA and even RNA information cheaper and easier has allowed. And I spent 14 years at Illumina, spending a lot of my energy on consortia, where I founded their consortia program. Some of the most fun I had in that space was building some of these diversity chips. It's been really rewarding to see Illumina continued engagement in this. But RNA, as a reflection of real-time biology, has some challenges. High abundant proteins, we'll just talk about plasma for convenience, but abundant proteins in plasma have been well served by mass spec approaches, and we've got LDL and HDL tests to show for that. But what we don't do very well in the proteomics space until I'd like to argue till now is a broad look at the proteome, including the low abundant proteins. And so that's where I think Olink really fits in nicely. So proteins make up 95% of the clinical trial candidates are targeting proteins. Over-the-counter medications are most often targeting proteins. And some would say that proteins are the business molecules of the cell. So being able to get some insight into proteins, I think, has been very important. I'm going to guess that, yes, awesome. So high abundant proteins are here. So I'm showing this graph. It's just a sorted graph of proteins in the first launch of Olink's library. High abundant proteins are up in the upper left. And as you can see, there's a really high concentration down here in the lower right. The scale is off to your left, so from above milligram per mil to below picogram per mil. And what I think Olink did well was really focus on this low abundant plasma proteome, because mass spec was already looking in this high abundant area. With mass spec, as I understand it, you really have to push a lot of sample through to see these low abundant proteins, and there's some complications in being able to interpret those data. So being able to do this at scale is very helpful. So what Olink can do today is 3,000 proteins against 384 samples in one run of a NovaSeq. So we're NGS compatible. We're also compatible with the NextSeq by Illumina. And we've announced collaborations with many of the other sequencing companies that are driving down costs in that sequencing space. I think this is the secret to why Olink has over 1,100 publications that are peer-reviewed from customers just since 2016. So that's the exciting company that I joined. We are at the end of the day, so I'm not going to spend any time talking about how it works, but you certainly feel free to reach out to me to talk about it. I'm going to talk about how people are using it. And I'm going to talk just very briefly about 2 biobanks that are using this. And Lyndon actually mentioned one of them, the UK Biobank, in a project called the Pharma Proteomics Project. Here's the same slide that Lyndon showed. So on the heels of the Exome Sequencing Consortium, the group or a subset of that group got together and talked about doing proteomics at scale on a large number of samples from the UK Biobank. It turned out that 10 partners came together and signed the agreement to do that project with the UK Biobank, got access to those samples, and they evaluated proteomics technologies, decided on Olink, and we had 3 additional partners join that were already Olink customers. So that was fun to see customers join and add value to that consortium, bringing it up to 13 total members today. There'll be over 60,000 samples worth of proteomic data that will be put into the public domain. The first tranche of those data should come out at the end of Q1. So end of March, we think. Of course, there could be delays, but this is really up to the UK Biobank. And it's just exciting to imagine what's possible with these data. So the next couple of slides, I'm going to talk about a powerful precursor to this UK Biobank project. So I'm going to skip over a couple of slides and talk about the SCALLOP project. SCALLOP is a consortium of cohorts that's led by someone who's got a co-affiliation at Karolinska Institute and Pfizer. What SCALLOP did was bring together 30,000 samples and look at 90 of the proteins on Olink's platform. Just with 90 proteins, this team, and this publication is in Nature Metabolism, Volkerson et al., this team identified 450 pQTLs, so just correlations between protein levels and areas of the genome. They then were able to narrow those 450 pQTLs using clinical, proteomic, and genomic data with Mendelian randomization, co-localization verification, to 25 that appear causal. So Pfizer has publicly announced they've started at least 2 programs from the data from SCALLOP. The blue circle is meant to indicate 14 of those 25 that appear causal are recapitulating known therapeutic targets. So some of those are in that blue circle. The green circle is meant to indicate 11 that are new novel targets that were very exciting for this team to find and represent new possibilities. And the purple is really exciting to me because those represent repurposing opportunities. 18 of those were identified just in 90 proteins for either repurposing existing drugs in new indications or potential for a better understanding of target-mediated safety in these therapies that we already have in the market. So that's the bulk of what I wanted to mention. I had a poster here. I'm happy to send that poster to anyone who's interested. I do want to say that the future for genomics in Africa is looking extremely bright. Proteins are representing a multi-omic component that might add value at some point. We will be here whenever you're ready. So I just want you to know about it. And the first service provider that is offering our qPCR readout, which offers 92 protein plex assays, is Decipher, that is within the Diplomics network. So super excited that ribbon cutting ceremony will happen soon, but they're already offering services. So this was the first time I actually got to meet them. And that's my spiel. I hope that some of you will think about us, and we will certainly keep expanding our library. Our goal is to expand beyond 3,000 proteins by the end of this year. Thank you so much.
That was brilliant, Cindy. Okay, so you may think we're near the end of the day, but we're not. We are now inviting people up to the stage here. Craig, Junaid, Lyndon, Alice, Cobus, Carrie Ann, and Helder. And this is our opportunity to ask questions of our speakers. We already have quite a number of these questions, and I'm co-leading this question period on increasing genetic diversity or increasing diversity in genomics research with Segun Fatumo and Tanash. Nash, are you here? Okay, great. Fantastic. So we'll just get everybody up here. We have enough chairs. Yeah, I'm gonna stand. So I'm gonna take the first question, which was printed out, was already asked, and I think it's a very pertinent question. And then Segun, you already have some questions here anyway, so that seems natural that you can ask those questions. And so I think this first question is a great way to start things off, which is, are we ready for the precision medicine era in Africa? So would anybody like to jump at that question? And if not, I can start asking somebody directly. Fantastic. Thank you.
Yeah, I'd say that we are ready to start in earnest. And there are ways to make use of what has been done elsewhere to use methods to make inference from African data, to enrich African data with non-African data. So I guess that's the computational biologist, data scientist type in me that says, yes, we are ready to do some things, but we need to start the generation of much bigger data sets.
So I had a follow-up question to that. So if we are ready, which sounds like we hopefully are, is there a specific place that we should be starting in the context of precision medicine? So we can't do everything in precision medicine right away. Is there perhaps a disease that we should be focusing on? Or should we be, maybe I'm being wrong here, maybe we should be thinking a little bit bigger. So if you want to continue, that's fine.
Yeah, so I'd say reference data sets are very important as a first step, because that helps build the foundation. So everything from reagents, like right now, if you think you could order a clinical exome for an African individual and get perfect data out of that, it's very unlikely. And that's purely because it's been designed against a genome and reference variant set that's not relevant to Africans. So I think as a first step, we should be building that reference data set. And that will lead to a series of products already, in my opinion, or improvements in products.
So references for sure. Are there proof of principles that we might want to be thinking about?
Well, maybe I can come in here, and I think this is something that Prof. Ramasa had mentioned a little bit earlier when we were talking about the costing of the sequencing and mentioning that Invitae had been, had panels for clinical genetic services. So I think when one wants to look at precision medicine, I think one could sort of go for the low-hanging fruit and look at these Mendelian disorders, and specifically here where the gap now that Invitae had left is a good opportunity for us now to start looking at these diseases. For example, I know my laboratory manager is Dr. Nadia Carstens, and she's quite passionate about developmental disorders in Africa. So having those Mendelian type disorders that one could start off with would be, I guess that would be my sort of inroad into the precision medicine in Africa, sort of the low-hanging fruit.
Great. Thank you, Craig.
Thank you. So I was listening carefully to your presentation, and some words that really come out is that all of you are showing interest in increasing diversity in Africa population, which is exactly what we want to do also in Africa population. So we have the same agenda to increase diversity in Africa population. So you may recall that so currently in the US, there is All of Us project that is now moving to get 1 million people sequence in the US. In the UK currently, we have a new project called Future Health that is aiming for 5 million people. If you are aiming to increase diversity, I don't believe that we should be talking about 50,000 sample, 100,000 sample, because by the time we are done with 50,000 sample, 100,000 sample, you already have 5 million. We are still going to be saying the same thing, that we are underrepresented. So my question is that how much of engagement are you really prepared for in Africa?
How many samples?
Ambrose published a paper. You are aware about that, and Ambrose was suggesting 3 million. I'm not saying that we start with 3 million.
Yeah.
But if we look at the current ecosystem and we do like 100,000, 50,000, it means that in the next 10 years we'll still be trying to catch up, because other population they're not waiting. And this other population is the same company. It's all of you that are supporting them.
Yeah.
To increase diversity, their own diversity.
Absolutely.
So how much of engagement are you prepared for?
Yeah, so, and, that's a great question, and it's something that we're gonna have to deal with and work on. I don't think the initial sample number that, we're talking about here in Africa is the end. I think it's the start. I do think we're gonna have to ramp up faster than what we did in, say, the UK Biobank. I think the UK Biobank was a preview of our future health, for example. So they started with 500,000. Now they're going to 5 million, if you will, in the UK. So I think we have started some efforts here with H3Africa, with some of the other initiatives we heard about at these meetings. That's a great start. And I think now what we have to do is to build these relationships with companies like us and start. But it's only a start. It's not the end. And we're going to have to ramp up fast to catch up so that it's not, you're always playing catch up, And secondly, I think, we may need 5 million European samples sequence, but we may not need 5 million African samples sequence because of the genetic diversity. So I think there's going to be a lot more discoveries with lower numbers. And I think we saw some of that data at these meetings regarding that. So it's not just a numbers thing, it's more of a scientific, answering scientific questions.
I think Cobus.
Yeah, I think that is part of what we want to achieve with APCC to, and our sort of initial estimates, there are 3 to 4 million people in these longitudinal cohorts in Africa already well documented, but not, very few of them sequenced, of course, and to create the environment in which scientists have access to these populations to increase the pool of data that is available.
Thank you so much for your responses and also the discussions that we are having currently. I think in as much as we increase diversity, we know that we might find some discoveries along the way, and bearing in mind the history that has happened in the past where there's been exploitation by companies. Have you started to think about how these benefits are going to be shared in the future? And if so, maybe can you clarify how you anticipate that this can actually be an equitable or a fair game when you are planning to partner with communities and even with academia?
Yeah, and I think one of the sponsors of these meetings we've had this week is a good model of dealing with profit sharing and community benefit. It's not something that I think we've solved, and we're going to have that conversation as pharma companies coming together in these types of partnerships. So I don't have an answer, but I do know that we have to find a solution, especially with partners like Africa and African institutions. It's different than those 130 collaborations that I noted that, for example, the RGC has. Each one of those collaborations are different. They have different, contractual deliverables and obligations. And I think when we are thinking of coming into Africa and into our HBCUs, for example, in the United States, we have to have those discussions and come to a win-win for everyone. I can tell you we've had discussions about profit sharing and community benefit. I can tell you we have not solved that problem, though, and we have to have that conversation. There are ideas I have, but there's no solution on the table. We believe in capacity building. We provide in-kind sequencing, for example. We would like to provide resources for follow-up so that communities that are participating do get the healthcare needs, coming back to precision medicine, actionable variants and clinical validation. So we can talk about, those things are more short-term, scholarship, training for communities, building STEM programs. But, the longer commitment of, profit sharing on intellectual property, that's something I think we have to have a conversation on.
So I'm going to be butchering this question admittedly, but or putting my tweak on this question that came from the audience. is there a role, so we're all industry representatives with the exception of a couple here. Is there a role for partnership with government in helping enable genomics in Africa, getting more genomes sequenced? Is there a role for direct to consumer? I'm kind of combining a couple of questions here now, in supporting that. And is there a place for your organizations to either partner with government or partner with the individual? So.
Maybe I can say something here on behalf of MGI. We're certainly very interested on these partnerships. We done it in the past. I think one outcome, positive outcome, let's say maybe with the pandemic, if we might say so, is how only way maybe we can fight something is putting resources together. And there are different stakeholders here, but we definitely leverage a lot on the support of local governments to advance. And I think these projects that we were discussing here across these days, it's very relevant for the population. And you mentioned a question, what diseases to focus, this is all very specific. We can go with each country might have different interests because, it's very, very specific. So I think a project like this ones, when we truly talk about population genomics, there's several factors involved. And one of them is, I think it's engagement of the key stakeholders like the government, that can, put the structure together so we can even collaborate across countries. I think pan-genome in Africa means collaborating with different structures, different regions. And you mentioned direct-to-consumer. This is a very interesting one as well, because maybe we're not just right there, but we at the same time, we live with a more consistent about our lifestyle, and genetics for sure can have a very important component where you can help you to take decisions on your day-to-day life. So more and more, there will be more scientific evidence where this is supported by science, and direct to consumers should have the choice to do a genetic test and ask for genetic consultant to take their own decisions that affects themselves, because it is very easy for us to engage with any risk factor that leads us to a disease. But sometimes it's difficult, even we live in a time where it's difficult to do a test and that help us to take decisions for a healthier lifestyle. So I do think there will be more engagement of direct-to-consumer, but I'm not so sure if this is ready just yet.
And that would require training of a different sort, a clinical training, and deciding how and who want to have that.
Absolutely.
Sorry, Clovis, go ahead. Yeah.
Yeah, I want to talk about a partnership with government of research institutions. And I want to use as an example the South African Population Research Infrastructure Network that Claudina mentioned in her talk, where these are either university-based or independent research institutions that are a network of longitudinal population cohorts. And government is investing in the core funding of those cohorts, because they see that as a national research infrastructure opportunity to promote population-based science in general. And that is also an idea that the APCC want to advocate for in our engagement with other countries in Africa, to see investment into these longitudinal population cohorts as a way to increase capacity.
I'd like to come back to that, but go ahead.
Okay, to the industry again, just to ask about if you can explain what has changed, because it's like this is the first time that we are having industry come to us. So there must be that there is something that's actually changed, that's actually motivated you to come to us. The pharmaceutical company are very collaborative a lot with people in the West, in the UK, and in the US. So what is the thing that has motivated you to change? So bear in mind, I would like you to talk a bit more about if you already have a model that really work from previous partnership in the West, maybe with UK Biobank, in terms of mutual respect and as well of in terms of trust.
Yeah, I think the UK Biobank really helped a lot in terms of showing that we can do this collectively and getting, groups to come together to pool resources so that one company doesn't have to do everything. I think there's always been an interest in companies. Why now? I think, I would turn the question around and say, why not now? But, we have to start. And I don't think there's any alternative motive here. I think we all understand the science, that the opportunity for science here and helping communities and patients. I do think technology has caught up with sequencing and getting cheaper sequences. I think a lot of that, just the timing has helped us to do this. I also think that there's been a lot of social justice things happening around the world that's raised a lot of people's eyebrows and got us to say we got to do something and we got to do it now. And last, I would say there's a lot of folks in these companies who care. We got the Mark McCarthy's, we got the Lindens, we got the Allens. We have people in the companies who have been pushing things like this. It's just, now they're listening because I think there's examples like the UK Biobank. There's examples of need through social justice efforts that we see around the world. And it's easier to do now at a large scale with technology being available.
I think just a follow-up question in that vein. I think there's an earlier question that has been asked that in as much as technology is improving, the cost still is a hindrance, particularly in Africa. And as we see the funding legs also dwindling, with H3Africa about to end, how will this partnership be? Is it, what kind of mode are you going to use? Are you going to aggregate as companies? And how are you going to intersect with the academia and communities? Can you be more detailed in that arena, such that we're not, because everyone, I think, wants something practical to hear if you have a practical vision.
Yeah,
I'd like to leave us with excited, but maybe not so sure how it's going to kick off.
Well, I think just the fact that we're having this conversation here, and as you said, industry it's got a panel up here of technology companies, pharma companies, I think that right there is a very good step in the right direction. And we just need to continue that dialogue to build it the way that's best for you. And I would say how we're going to do it. I think we have to do it collectively, as I said in my presentation. I do think it takes government. I think it's going to take philanthropic groups. I think it's going to take technology groups. And we're all going to have to bring our tools that we have to the table. And some of those tools are funding. Some of those tools are technologies that we have. Some of those tools are relationships that you could shape policy and things like that. So, we need you guys to also help us to get this, more broadly out to these other companies and so forth that folks would join.
Can I just add as well? I think prices is definitely an important topic because, very expensive might leave many projects in the drawer. But I think cost reduction might be driven by two ways. One is increasing demand, and another is innovation. It's really allow other companies to have a chance to bring their products to the market, do their investments in innovation to bring the cost down. So I think it's important in any setup in any region that there is always new technologies coming to the market and keep a very rational way of seeing these technologies coming to the market, but not close the door to this technology, because these small or large organizations, they might come with technology that can bring the price down. And I speak on behalf of the MGI. Sometimes they don't know us, but we can offer the same quality, much more affordable solutions that are currently in the market. But we still not as known maybe as the main players, but we do have a solution that's cheaper. And invite all of you to talk to us. We will be very happy to collaborate with you in many different ways.
Great. I just have one comment and one question before we turn things over to Barry and Emer and Simone. And you guys will all stay up here. Great, Craig, that you mentioned the work you're doing in, I think, in agricultural space as well. And, as we think about climate change and impacts there, World Health Organization has a program to think about everything more holistically, not just in terms of human health, but also of the health of the planet and the ecosystem as well. So I was glad to hear that that's still an activity in your organization. Cobus, you mentioned longitudinal cohorts, and so you made a point of emphasizing that. And so my first question is, how longitudinal? What do you mean by longitudinal in this context? Do we mean longitudinal biologics, or just surveys or electronic health records? And how important is that from, say, a biofarm kind of perspective or a tech perspective? And is it important? Like when I think of industry and biofarm, I usually think, oh, I want to cure a disease. How many samples do you have with, say, type 2 diabetes, and how many cases could I pull and do that? But from a longitudinal perspective, how valuable is a longitudinal cohort as well? So, and I think that's a great asset. A lot of the cohorts that Cindy showed up there are going to have pretty short lifespans because they don't have consent for longitudinal analyses or studies. So that was long-winded, but go ahead.
Yeah, so with longitudinal, I mean a defined cohort of individuals, ideally a whole population, so from birth to death, that you maintain a record and contact with them over time, so that the observations that you have are not just events, but states. Secondly, the benefit of that is, the same sort of reason why COVID is not a pandemic, but a syndemic. So disease is a complicated interaction between various factors. Obviously, we're in a omics conference. Genomics is one part of it, but also the environment, socioeconomic conditions, political conditions. And I think that the complex problems that the world and Africa specifically are struggling with to achieve SDGs are all multifactorial. And these longitudinal cohorts offer us the laboratory, the human laboratory, population laboratory, but more than that, also the planetary health laboratories, where we can understand disease and the complex factors and get the next generation of solutions to health problems.
Great.
Yeah, so I think in a future of data science, when things are a lot better than they are now, and they're already pretty good, we'll be able to probably identify from samples from longitudinal cohorts, we'll probably be able to identify multi-analyte markers that are prognostic or predictive of future health issues. And so it's precision health now to give the right, let's say, the right treatment to the right person at the right time, based on, let's say, some biomarker. I think sometime in the future, there will be a bigger focus on prevention. So I believe that that's where those cohorts are extremely valuable.
Fully agree. I think we have to step down and let our colleagues come, and thank you very much.
Okay, so in this next section, we're going to broadly cover the topic of capacity building. And we're gonna follow a few themes, moving from genomics and population genetics through the types of technology infrastructure and the human capacity building. And then there'll be some wildcard questions will come up as well. I'm going to start actually just transitioning between the last topic and this topic, addressing a comment that was made. Lyndon, I think it was you. Don't kill me. Something along the lines of the importance of an ability for H3Africa to ramp up in terms of numbers quickly, and that 5 million is the number for our future health. Maybe we don't need that many in Africa. I would make a counter argument. Maybe we actually need more. UK is one country of 60 million people. Maybe we need maybe a lot more. But my question is, from your perspective, from your lens, what do you perceived as the biggest barriers for ramping up?
First of all, 5 million was just a concept. I'm saying the concept of trying to catch up to the European number is not really what we should be focused on.
Yeah.
It should be identifying the most variants that we can in this population. The largest barrier, I think, is getting support to get behind the initiative. I think we see here in Africa with H3Africa, with a lot of these other initiatives ongoing here, there's a lot of energy. There's a lot of folks here that are willing and participants. I've been talking to a lot of groups, and they have community engagement, programs, and folks are ready to participate. So I think it's getting funding to do it. That's the barrier.
Anyone else want to jump in there? I mean, maybe a little bit about the role of industry in helping to generate that funding.
Maybe just a quick note on the technology point of view. In terms of scaling up, I think there are definitely solutions for it. I think some of you already saw last week at LGBT, we are announcing a new sequencer that can do up to, if I'm not mistaken, 70,000 whole genomes a year. So we do have factories and lab automation system that can do 5,000 lab preps a day. So in terms of capacity, technology-wise, to get the data, there are solutions for that. Of course, upstream or even downstream of this, there might be some other channels which I'm not even as familiar as others maybe here, but it's some part of the workflow. There is definitely solutions for it.
Yeah, I think one of the other barriers that we don't talk about often enough is actually the amount of skilled personnel to actually generate the data. And this is something that we do try to address at the SAMRC by building capacity for things like sample preparation. We spend a lot of time generating data, and we spend a lot of our effort in training people to analyze the data, but we do also need to focus quite a bit on those who actually have to generate the data. That I find is, in my experience, quite a bit of an issue to ramping up.
Any other thoughts?
Yeah, so I think really we are addressing one of the big barriers. So willingness to engage and talk to each other, even industry competitors, if one call it that. So identifying areas of potential duplication and preventing that will increase representation. So just by doing that, we end up being able to produce more. So and engaging around GENCO as one example, initiatives like that will start removing political barriers as well between industry, academia, industry, and nations, as an example.
Yeah, I just want to follow up on that great point, Junaid, because with GENCO, you got some government, folks to come in and bring together competing, if you will, academic organizations. I mean, these organizations are academic organizations that compete for grants against each other, right? We need that same sort of energy around different pharma groups, other technology groups, other, philanthropic groups, someone to come in and do what, Francis and the NIH and others did with GenCo, but to do it in these private sector areas, to bring folks together and say, we got to do this, even though we're competitors. And to Janae's point, we need to all come together and do this.
Fantastic. Okay, so sort of then building off of that and thinking a little bit more about capacity, we heard a lot about GenCo and the 8, potentially up to 10 centers of excellence, but thinking also about how we bring others to the table and offer opportunities to others too. How do you think about the benefit to other researchers working in the field of genomics in Africa? And can we reach out to them to become part of H3Africa or GenCo or these types of partnerships and initiatives that are, placing a central goal at the center of a mission?
So I think I'd like to jump onto Craig's idea of rare diseases, right? So, I mean, it's probably a good way to build success stories for genomics in Africa. And success breeds success. And cost is potentially lower. You need smaller cohorts. And you may only identify a target that becomes a diagnostic that's useful to a region in one country, but it's still a success. And that will attract more people continent-wide to genomics and omics in general.
I think we're going to need more. There's a lot of need around the world, including in Africa. We're going to need more than just GenCo. We're gonna need more H3Africa type of efforts. I think you guys had a wonderful model, and that should continue. And I think we need to build other initiatives, like I said, because there's going to be GenCo doing certain things. I think there's other initiatives that can fill the gaps in those other areas that of need, because there is a lot of need.
And so do you think the idea is to create kind of entities like GenCo, but also sort of infrastructure that fosters alliance and working together?
Yes. Yes, I think there's going to be, if I can say GenCo and some mini GenCos, but GenCo can't be it all, is what I'm saying. I think, and H3Africa can't be it all. I think, or H4Africa. We have to do more. We have to have more focused strategic partnerships that, address the needs of the people.
Great. And I'm going to ask one more question before I pass it over to Barry. But so what is the profile of scientists to be trained for the different topics of genomics? And do you think that capacity building should be something that we don't just do every 10 years, that we do periodically and redress every, some periodicity of cycle?
Yeah, so I have a background in this. I was an academic until very recently, and we need continuous capacity building, not even periodically. It should be all the time. The field changes all the time. We need fresh new ways of thinking. Yesterday's bioinformatics, for example, doesn't necessarily work today, and sometimes the older generation gets stuck in a way of thinking. So we need to bring in new talent, new training, individuals that can bring their creativity, and Africans are extremely creative. We need to introduce those individuals to the sciences that we need them to, that the omics community needs, and do that continuously, every year, all the time.
And just to follow up, I totally agree. And the continuous progress it's evident for everyone. And sometimes it was mentioned maybe there is a biomarker for one specific country, and maybe none of the industry here can offer a solution. So there's also opportunities for local companies to develop solutions that fits their own market, or bring new products to the market. So I do think it's a big challenge because of that, because it's a multi-stakeholder project where everyone needs really to collaborate to achieve the end goal. And as mentioned, the technology evolves very, very fast, very, very fast. In 2 years' time, I'm sure what we're talking about standard is going to be a different one. And this is important, I think, to keep as I mentioned earlier, keep the doors open, always look for, solutions that fit your needs. And at the same time delivers the quality, for example, that you are expected to have. But this has to be ongoing work, in my opinion.
So just to follow up that question, we spoke a lot about training and, staying ahead of the curve with regard to new technology, new approaches. But another thing that's a barrier to, having evolving cycles of capacity building is that the time it takes to stand up new infrastructure. That is often quite lengthy. And is there anything that you are doing on your side to provide solutions to make infrastructure more lightweight, easier, and more rapidly to get up and going?
Sorry, maybe I can just comment on this a little bit. And I take your point that the infrastructure, to put that infrastructure in place for sequencing, it does take a long time. So what we've done at the MRC is, and this is in partnership with Diplomics and ONT, is to use available technology, the small Monian sequences, for example, to take those sequences where you don't need this big infrastructure, but take it to our under-resourced institutions and use those instruments for training, and actually just to give these these institutions access to genomic sequencing to next generation sequencing where they haven't had it before. So you don't need the big infrastructure investment initially, but you're actually giving them access to technology that is available already, and it's something that is easy to use. So that's why I see using those types of technologies can actually broaden our access to sequencing across Africa.
That's it. Any other comments?
Yeah, so I think I just want to add, sort of having that connection between the industry or the technology providers with the people that are offering training. So obviously there are quite a lot of groups that are offering training, and to make sure that those connections are kept up to date, so that we keep up to date with the skills that are needed for the new technologies that are coming in. And so far, it's something that obviously is just come to my attention, that this is not, since we are training a lot of people, but our efforts to actually keep up with what's happening with the technologies is actually reduced. So it'll be good to have like a combined effort with what's happening with technologies as well as what's happening with the training, and so that there is a continuous feeding into each other of what are the skills that are actually needed and what is actually possible and practical, because some of the technologies are new and they're good, but is it practical to train people in skills that they're probably not going to use for the next, 5 years. So yeah, so just keeping in touch with what's actually happening on the ground, and all the new technology is something that we probably need to collaborate more on. And I'm just going to have to excuse myself because I have to go, but it's been lovely to present and to meet everyone today. Thank you. Thank you.
I'm going to turn this over to Simone to ask some questions.
So my questions are going to be largely around sort of human capacity, I would say. So one of the biggest problems that Africa currently has actually is it's a double-edged sword. It's a good thing, but it's also a tough thing, is that we're producing a lot more PhDs than we ever did, right? But there's very little space for them to go anywhere. There's a limitation on how many academic jobs they can actually get. You know, there's very small pool. And so I wondered if there was space within, you know, industry, or industry had considered the idea of a professional PhD-ship. So that is, you get your PhD, but you maybe do it in collaboration with industry and academia, and it kind of prepares you for a role in industry over simply academics. And I wondered what your opinion on that would be.
Yeah, so this is something that Rosha has been thinking about very seriously at the postdoctoral level. And we are working with funding organizations to try to make that happen. And we're also seriously considering the concept of mid-career scientist positions, as well as research chairs, as ways to retain talent, but to retain talent that can train the next generation of talent. And we're also really quite seriously thinking about how do we build the next generation of leaders so that there's continuity. So yes, it is something that we're seriously thinking about. We don't have concrete solutions, maybe for the postdoc one pretty soon, but we're thinking about all those different levels.
It's good to hear.
Just a quick note as well. There's a lot of PhDs, but even in industry like us, we need even lower degree people to make this happen. And of course, PhDs is a high level specialized person in one particular field. And we're talking about quite advanced technologies most of the time, but I think there's a lot of opportunities. And if there's one industry that is growing, is the NGS and proteomics and genomics. I think it's an industry that's here to stay. I'm sure there will be always a lot of opportunity on university, from university students to come to industry for sure.
Yeah, and I would like to add that I think that's a great idea. We should definitely, talk about that and develop, programs around that. I know Regeneron would be very interested in it. However, I would like to create an environment here in Africa at institutions where you have more positions for them to stay. And I think we can do that by, again, creating research resources that they can study and they can write grant funding and get funding and stay here. I would love to recruit, your wonderful, the wonderful scientists here in Africa, but I think we should think about setting up a situation where they can stay here and educate, the next generation of African students.
Right, and how much of that is actually, providing the environment for that to happen. So it's also about, creating branches of R&D in this country, in African countries, because there are companies that are doing it, but it's very small, and again, not a lot of opportunity. So I think, especially the big guys, big players, even, obviously small guys as well, it really is going to become increasingly important over the next little while to really increase the capacity. And I guess this all ties into, oh, sorry, you want to go? Sorry, you put your hand up.
Yes, I did. I just want to say that, of course, I'm talking about population cohorts in Africa. And my experience in previous iterations of something like the APCC, the in-depth network, have taught me that how these longitudinal cohorts can be places where young researchers can gain experience and provide them with the context and opportunity and access to data to develop their own careers and become competitive and obtain grants over a whole spectrum of different disciplines, ranging from social science to the basic sciences. And so the diversity and the range that can be provided by these longitudinal population cohorts are a great source of capacity building for science.
Yeah. And then I guess that, kind of all ties into a specific question that was asked by the audience, which is what strategies can be considered to retain genomic scientists, and I think you alluded to it, in Africa, working on African data in Africa. You know, the incentive is if they, I mean, I guess we've covered some of this, is, you know, the incentive to go overseas for industry positions is strong because there's not a lot in this country. I mean, have the companies thought about how they can retain what they've achieved staying in the place that they were trying to achieve it?
Yeah, I work for Roche Diagnostics Cape Town, and that's actually a very good example of where we currently have 300 a staff of component of 300 Africans producing at a very high quality and high level reagents that the entire world uses. So it's a good reason for them to stay. It's cutting edge. So we need to build that bioeconomy type ecosystem. But I think we need to be open. The future is going to look different. We need to be open to things like allowing our students to take advantage of and graduates to take advantage of the gig economy. So we should allow them to consult while staying here, while doing their academic work, as an example. The alternative is they will leave. I think the next generation is going to be very different, and we need to be creative around how do we keep them. And then entrepreneurship, you know, we don't need to build a huge company, but we should foster entrepreneurship. We should provide formal training programs around that. And there are going to be findings that are actionable, that are not necessarily interesting to big biopharma, but very important and very needed in Africa. So there needs to be some training about how, around translation and productization and what it takes to get something approved for clinical use. Once you put those things in place, we will stimulate a bioeconomy. And they may not stay in academia, but at least they stay in Africa and contribute.
Maybe I could chime in now. We had a discussion with some of the emerging leaders last night, and a couple, I thought, interesting questions arose. One was, what are the skills that you as leaders and producers in industry are looking for in people who are currently in training, with a view not just for what you're doing now, but they have careers ahead of them. What is the kind of training that you're looking for that will carry them through to be productive collaborators with you over the next 20 years?
Maybe I can jump in. As I was saying earlier, I think there's within the company or industry, there's multiple kind of jobs, one more technical, others less technical, other more marketing, other logistics, operational, even strategic ones. But I will share just an example. All this might be, totally different. Recently hired a person that was, selling yogurts from that to Danone, and now he joined us as a, selling our platforms to the market, and it's been very successful. And more than anything, what drive me to hire this person is his energy. He really wanted to understand our focus on the customer, and I know we are here just to support the customer and all of that. And he was really driven by that. And that alone, sometimes he needed to have the technical background, but that we can also teach. And for some positions, we are here also to coach and grow with them as well. But it is very diverse, and more than anything, sometimes it's attitude. And none of these companies that are outside the continent might not honestly, they won't be successful if they don't work with the local structure, the local community, with the local people. I'm sitting in Spain in my office, and I don't know exactly what is happening here. That's why I have a team dedicated here, and I leverage a lot on what they can, what they know and learn also here. And so it's really important for our own success to have the people from local that actually know. Of course, there's always a different paths, but I keep saying, even for management roles and all of this, we're not taught at school. Many of the things that we do in our daily lives, we're not taught at school. We learn maths and all of that, it's all very important, but then you have to lead people, and then you have to lead conflicts, then you have to do a budget, then you have to finance. And most of us didn't taught this. It's the real life that comes, and you have to do it. So I think more than anything is to be curious, learn, constant learning process. We've been talking about this. We need to constantly adapt and learn and focus on what we are here to. What is the mission? And at the end, for an organization like us, it's the customer. So if the person understands this concept, I think they can succeed in an organization, in my view.
Yeah,
please.
So for me, I think it's the passion of that person. If you are sitting in an environment where you want to move into diagnostics, you want to help the researchers, we need the young people to be passionate about wanting to do what they're doing. It's not just a job. It's not just something that's, you're doing, and that passion's got to come from within. You've got to want to run that extra mile. You're going to want to help get the best product. And the commercial side is also important. No one is taught about budgeting. No one is taught about, profits and all of those things, but you've got to learn it, to move into the commercial industry. You've got to learn that, and you've got to be passionate about the research that you're going to be selling, and then the products that you're going to be selling. So I think that's key as well.
So it's interesting, you've both focused on the quality of an individual's ability to learn how to learn. But when you often hire people, you have all these forms to fill out of what their skills are. And one of the questions last night is, what are the skills not now needed to do sequencing and analysis, but what's going to be the result of that 10 years from now that they should be able to bring to your companies? That's assuming they're all motivated and bright and willing to be able to learn.
So I'm going to take the question, I think, a little different. What I tell people to be successful in whatever role you're in, I think you need three things. You need the technical skill set, you need the relational skill set, and you need an influential skill set. And each of those bubbles, I call them my bubbles, my three bubbles, technical, relational, influential, each one of those bubbles change as your career progresses. You're going to come into a role, they're going to hire you probably because of your technical skill set that you know. You know how to do NGS, you know how to do this, you know how to do that, they're going to hire you. Then as you grow in your career, you're going to have to become more relational leader and build those relations, and your technical bubble is going to grow a little, but your relational bubble is going to grow a lot more as you progress. 5-10 years in, now your relational and influential bubbles are much bigger than your technical bubble, because now you're influencing departments or teams or even the company. So you got to know where you are in your career. You got to know what you want. You got to be very passionate in what you do. You got to know the technical skill sets to get in the door, but to survive, you're going to have to build those relational and influential skill sets.
Yeah, so I think that an agility mindset, embracing the knowledge that the only constant will be change is very important. I'm probably preaching to the choir for the bioinformaticists in the room, but dedication to lifelong learning and the ability to play more minor roles on certain projects. if a person can accept that occasionally they will have to work that way, then they'll be better team players and they end up learning a lot more, but they end up contributing as an aggregate a whole lot more. And that's how I run my team. I reward collaboration. So it's partly our job as well to guide whoever we employ through that path to set them up for success.
I'm somewhat surprised and both pleased with the answer of focus on individual skills and development and capacity to interact and provide leadership within groups, not just for themselves personally. But I come from a university where students have learned how to do AI GTP, and they can have machines with artificial intelligence write their papers for them. I was surprised that I didn't hear your sense that all of a sudden you're going to have vast amounts of data that I'm not sure individuals are going to be able to cope with. Where does the role of artificial intelligence come into the genomics world? I've just been reading grants where physicians' notes are no longer relevant. A machine will translate them from verbal contacts into reports that then analyze them for what the nature of the disease is, without a doctor having to do anything except check it in the end. So I'd be curious to know how you see, if you think I'm right, that AI is going to be a major new tool that in every medical profession is going to be important. It didn't seem to be important in the way your businesses are going.
So there are many areas to apply. I've done quite a bit of work on knowledge engineering for the purposes of AI, and I don't feel that we're ready yet to remove the human from that process.
Sure.
And AI is a tool, absolutely. So if we think of something that we do, protein engineering, Google came up, DeepMind came up with a way of determining the fold, at least the theoretical fold of every known protein. So that's a good use of AI to build templates that one could engineer things against. 100% AI from problem to solution, not quite sold yet, but it will play a key role in speeding things up in the future.
Let me ask then a final question, which is, databases do not prevent or cure diseases per se. So we study genomes to be able to improve health of populations in Africa, particularly for this program, but globally. What would be the skills required for the translation of what is being accumulated through these genomic studies over the next 5 to 10 years to translate them, particularly for common variants that will be identified that provide increasing risks for disease? What are the skills that you would be looking for that if there were greater pharma and vaccine companies in Africa, that they would want for trainees to be prepared for?
Well, I'm not expert in AI whatsoever, but I read an interesting quote the other day saying AI will steal only the jobs that don't use AI. So I think we need to, the point I was making earlier, this is evolving very fast. We need to follow how this is evolving, but AI might give you the path, but then even at that stage, we need to execute. The several biomarkers is identified. Then how do you translate this to a clinical outcome? So we need to build a kit. We need to have an infrastructure to make it. I think there will be actually amazing opportunities overnight for many things that we didn't know 1 year ago, and now we know. but then we have to implement these things and execute it. And at the same time, AI might replace many of activities that we do now, but I think we all humans, we all appreciate, for example, after pandemic, we actually here, we could have been a Teams meeting, but it's not the same thing because it's important, this relationship between us, talk and discuss and go for a dinner afterwards. It's important we as humans. So I think this will never be, or if it will be one day replaced, maybe it's a sad day because we also need as a human. So many activities that are highly dependent on this, I think they still be prosperity activities. Of course, other activities will be easily replaced, but as I was saying, maybe these other jobs will replace. We need to adapt and use the AI tools maybe and embrace them and not ignore them because it's almost unstoppable right now. They will be here.
Any other further comments?
Yeah, so I think that
on the translation relationship between what we're doing now and how to get tools to intervene in disease.
yeah, so I think AI type tools, so maybe large knowledge models rather than large language models, as a way to get to the point where a human, again, trained enough to be comfortable with computation, but strong in biology, makes the final decision about whether what the machine found is truly relevant and whether any translational lead that was generated makes biological sense. So I still believe we need strong biology training with enough comfort in computation.
Human judgment and intervention has to be the less.
That's my opinion.
Please.
Yeah, I think I'm sitting far enough from the audience to be able to dodge any water bottles thrown my way. But of course, omics is not the only science that has something to say about human health and well-being. And so to implement the wonderful innovations that are developed through your science, we need a whole host of other scientific disciplines before we will be able to make a difference to population health. Anything from social science to understand human behavior and what they will accept, what they will follow, what they will do to make an impact on their own health, to health economics and implementation research to look at the practical ways of implementing those solutions and getting the access and the universal healthcare access to populations in order to benefit from these technologies.
Wonderful answers. Thank you very, very much. Thank you, Simone. And thank you, Ina.
Okay, well, thank you, Barry and Simone. I am sticking around for the next topic as Charmaine is feeling a little under the weather, and I'd like to invite, I think it is Yasser and Gerald up to the stage. Hi, how are you? And what we're going to try and do is maybe finish this up at around 6 so that we can give you all a break. I'm sure everybody would love a break before the breakout interest group sessions happen at 6:30. You all might like a break soon too. Okay. So the next topic is sustainability, and here we're going to focus on a number of themes, including economic impact, commercialization, and intellectual property. Folks, I think I'll maybe start with economic impact and then pass over to you, and you can decide which one you would like to go with. Okay. And I think this is a really nice segue, given your comments about the context of genomic information broadly in society and public health and medicine and in economics and politics. And we were actually having a discussion about this at lunch. You know, there was an economic report issued by the American Society of Human Genetics a couple of years ago, maybe just last year, that actually was an audit of the value of genomic research and genomic technology to the United States in the preceding 10 years. And it was an enormous value, very indirect applications way outside of academic sectors through many, many other sectors. And, anecdotally, the economic value was as much as agriculture and fisheries combined in the US. So how do we think about those types of value statements that we can make about genomic research, genomic technology, also here in Africa, and how do we frame and analyze the economic impact that will happen over the next 10, 20 years? Easy one to start with. You can take a beat before someone jumps in. Who's going to be brave, Lyndon? Doug? Just it.
I mean, there's a lot of talk about data being the new gold.
Uh-huh.
And I would say that genomic data is probably the gold of the gold.
Uh-huh.
And the potential for making discoveries to just improve human life in general will pay dividends in the future. I don't think it's easy to quantify, but it's easy to dream about. And we should rather sell it that way than in concrete numbers.
Yeah. But sometimes concrete diamonds are the pieces you need to put in front of particularly non-academic researchers, right?
Well, there will be economic, savings to healthcare, right? Because if you use genetic information to prevent disease, that's going to have a direct impact on healthcare. So what it is a lot. I don't know what the number is, but it should be a lot. And to Junaid's point, the human capital that you gain there is priceless. So so I think, especially in Africa, where there's a lot of variants to be identified in what they do and how they play a role in disease, I think that can really have a high impact on people's lives in preventing disease, which then prevents the healthcare burden, which then, also opens up other economic opportunities like, clinical validation labs and so forth, So
fantastic.
The ripple effect.
The ripple effect.
Yeah, maybe also just to add not only prevention, but also diagnostics. We spent lots of money diagnosing and bringing patients to the end of a diagnostic odyssey without an answer. And this is where we can see that genomic medicine can shine, where we are able to get to that diagnostic point a lot quicker for a lot less money. Yeah.
Fantastic.
Yeah, so I'm thinking of like your pharmacogenomics and your precision medicine, your drug treatments. It's all about unlocking your genome and knowing how your body is going to respond to the different drugs, and that in itself has economic value. So for me, I would think that pharmacogenomics is sort of the key sort of applications to genomics in that space.
Just to add a small thing here, and I agree with my colleagues here. I mean, healthcare is huge. It's huge, and the value can be created in many different stages, service, patient, and all of that. But we all live, depending on many other industries where genomics has a tremendous impact. I was just thinking, for example, agriculture. We all, like, for example, I'm Portuguese, but I live in Spain. The eating from the certified pork, it's really something even ingrained in the culture. It value much more. So there's a lot of big industries just on certifying that the produce is really authentic. So I think the opportunities will, it's been mentioned, genomics is gold, but opportunities is not just in one point, it's across the value chain of any industry. There'll be more clinical trials, there'll be the need for developing more kits or whatever, there'll be more, logistics, operational, that everything is interconnected. And I think the challenge is, okay, this is moving fast. This will represent a lot of savings, but where would it be, the demand or that will drive this opportunity? But I'm totally confident this will be one. it was mentioned as well. I think one of the major challenge where the industry goes, if we were more pragmatic, or if we have audience here most focused on science and all of that, is we now talking about genomics, but we have our link also presented here. Our proteomics might change this, but then the metabolome, but then the patient data and all of that. And I think AI might solve part of this integration, but there'll be really a huge opportunity on technical people with a technological background to make sense of this, just on this scientific part of this, supply chain of all the industry. So there is for sure a lot, and the easy way we can identify this is looking what is the bottleneck. But what there is definitely, and we discussed here some bottlenecks. Why we do not more advanced? Why we not? What is a bottleneck, and how can we solve it? Maybe this is an opportunity right there. Maybe a company can be created based in here that can solve this bottleneck. It's looking where why we're not progressing as fast, and maybe change it in such a way that can run fast. Another way of looking at this is why, for example, we are late adopters of something, why we are maybe the last to do adopt to something. And there was, again, something there that was blocking this, and identifying this, just the bottlenecks that will have us around us might create all these opportunities. And most of these opportunities will be driven by local people, by people that identify it here and make it happen. They understand it, and it might create value through executing here.
So it sounds to me, and correct me if you think I'm not right in this, but it sounds to me that what you're describing goes beyond the type of scientific endeavors. It really goes into a large part of implementation science, testing what works, what doesn't work, and also to some degree entrepreneurship, whether it's startup or within sectors, because you want to be imagining how you could use technology in ways that maybe we couldn't anticipate in 10 years' time, right?
Mm-hmm. Absolutely. I think it's I think entrepreneurial spirit, and I'm maybe a year in South Africa. It's incredible how much driven the private sector is at the moment here. Mm-hmm. I discussed with some companies, private companies, very interesting project, and it's just not one or two, very few, and the entrepreneurial spirit is here. And I go in Europe, in Italy and Spain, at least from my own experience, I haven't had yet these discussions, and either is already in place. And I think this is very, very interesting. And I'm sure it's happening in South Africa or happens elsewhere there as well. But I think most of this is, we are here. I mean, many companies in the industry might solve many problems, but I think the idea here is also to look at what can we solve? What is the things we can look around, and maybe the solution is not yet there, but because maybe we're not focused on it. And you definitely have the bottleneck, you definitely have the problem, and maybe the solution is just here.
Mm-hmm. Fantastic. Anybody else here?
Unfortunately, I have to run.
Do you want the last word?
So thanks again for the opportunity. It was really engaging. Thank you.
Thank you very much for your insights. Okay, so I want to make sure we have time for Yasser and Gerald to be able to ask questions too.
Okay, so thank you very much. Maybe we can move to the commercialization section. So here we have a question on public-private partnerships. So the question is, for this partnership are important to advance genomic medicine in Africa. So, however, these playing fields are not even there. So how can we develop partnerships that fairly balance the contributions and benefits of the partners? PPP, so the partnership between public and private sector. So it's really important, but how to really, what are your guidance and advices to really make more collaborations and partners between the public and private sectors?
Well, I'll take a stab and say that's why we're here. I think that's why I know that's why I'm here, is to hear what how to do this. I think Regeneron is saying, look, we're here, we hear you, we see that there's a need of, precision medicine, genomic medicine is coming down the pike. There's a lack of genomic diversity of the African ancestry, and there's opportunities here and folks that are willing to work with us. So we have to work that out. We have to talk about how best to make that fair, how to make it equitable, how to make it fast as possible. So we're not playing catch up, but a part of the field that's moving. We don't have all the answers. We want to understand some of those bottlenecks and understand what the needs are so that we can help.
Okay.
Sorry. I think a lot of the public departments are governed by a lot of legislations, and we need to align with that so that we can help as best as we can. And I think that is the challenge for us. And once we get over that, then we'll be able to work together to strengthen the partnerships, for both public and private.
Okay, so personally, I want to paint the picture from the real world where I work, and then you can try to respond accordingly. So the same way you see H3Africa, so I work where there is a program to do the mapping of the NGS capacities across the continent. After mapping, we do resource mobilization, equip where we need platforms, train, sustain, then offer technical support. So I think along that path, we have done a good job. So here we have 2 industry partners who are providing platforms for NGS. But along the way, they don't give you all the information you need. They say, okay, you buy it at this much and then deploy. 1 year down the road, they say, okay, the software license is $13,000 per year. And what you have to know is in the academic setting, there's so much people are willing to learn, and they can even do this analysis off the APIs and able to give you results. But in national public health facilities, people just want a button, press the button, get the results, move on. So the situation here we are is when it comes to sustainability, we have deployed these platforms. In fact, for one of these suppliers has given us almost 31 platforms distributed across the continent. So these labs reach out and say, okay, the license is up. We don't have the money to pay for the annual license. What do we do? I say, okay, if you don't respond, then we'll put it aside and go back to what you used to do. So that's the situation I'm painting for you, and then see how you can try to engage with the public facilities, and always willing to give as much information as we need to get the right decision across, because we didn't see this coming. And they will tell you we can't pay a license of 13,000 per year for an instrument that we use once a month.
Maybe I will give a little bit away of the industry also works sometimes. First of all, I think this is a price transparency at the beginning, and this can be solved at the beginning. But more than anything, this is sometimes a cost that has to incur, because it's something that we need to have. But my point is, how can we work together to sort of compensate this additional cost, for example? And in an industry like us, where, for example, my organization is developing products at a very high speed. We have, new extraction kits almost every day. We have new library prep, new applications. We have new partners working with us and bringing new solutions to our platforms. We're also looking for, working with our customers, for example, sometimes to validate the product. And this is as value to us as an organization. So we can find ways. Okay, you give something back, you give something back to us. And for you, it's very easy to just run that machine and give all your feedback to us that, it works fine. Sometimes it's simple as this, but this organization like us has a lot of value, in particular us that we're innovating very fast. You have a lot of products. But I'm not saying, there's definitely different ways of getting the same result with different business models sometimes. And here is also mentioned here, Genomics is gold. If it's gold, you guys have the power. Genomics is here. So the people is here, the data is here, right? So it's also a way of selling the industry, what fits you? What fits your purpose? You mentioned the budget doesn't work as you expect. And sometimes, the budgets in different countries is totally different. But I'm not saying we are super mega flexible organizations and sometimes change is difficult. But I also think we only work together if you understand your needs but you also need to understand us as an organization, as an industry, which have a lot of people, salaries and invest in innovation. We invest a lot on this, on bringing these solutions to the market. So we need to make it sustainable as an organization, but there is different ways we can always find a solution. So sometimes, at least for organizations like ours, we are flexible. So we just want to understand how can we collaborate, and maybe a solution will be made for that case.
Okay, is there any other burning question that you wanted to ask? You're good. Okay, well then with that, I think we're going to end this discussion. I would like to gratefully thank all our panelists for all their thoughts. This has been extremely educational, informative, and lots to think about. And thank you also for your candor. It's much appreciated. I also just want to say that we will at 6:30 have the interest group sessions from 6:30 to 7:30. And then at 7:30, hope to see most of you on the roof deck for cocktails and reception and some unwinding. Okay, thanks everybody.
Sorry, just one quick announcement. If anybody has picked up a lost mobile cell phone, please hand it in at reception. Also, the rooftop terrace where the networking exchange will be, there will be some finger foods available, and that is on level 2. And adjacent to that is where the breakout rooms will be for further discussion on these topics. And dinner.
Where's the camera box and a tripod box? Okay. We're gonna pick up now. Then we're gonna put the but lecture thing. Okay, no problem. Yeah, there's a bit without that one time that you catching me or what. Yeah, no. When you're busy, you don't really talk. But I mean, you gotta feel for the shit. Yeah, I was gonna say stop being boring. I'm like, okay, I can actually. No, I didn't say don't stop being boring. Yeah. It's a bit. The other thing was gonna ask, so on that audio was the whole time background music on the recording? The whole time, yeah. So we can't really it's probably going into the cam. Yeah, it's coming from them. They're sending us channel. So for the whole day, basically. I don't know, man. Not all day. Not all day. I don't know. I thought it was someone doing yeah, because a lady sent a message to me to say like some of the camera guys are listening to me. So that's why I thought it was someone in that room. But I mean, if they've been sending music to that camera the whole day, then the audio kind of died. Anyway, yeah, we're just gonna pack it. Yeah, camera and tripod.
So, where should we pack down the?
You can maybe just bring the camera and the tripod here. Then we're gonna carry it upstairs anyway. Yeah, just leave it in there for now. Steve can sort it out. Yeah, because we're gonna pick up now and go upstairs. Because tomorrow we're carrying on the second floor. Okay. So is that why you're picking up? Yeah.
So what it was that it stopped before me because if you don't want to shoot over to them to slot me, so I was like take out tape so there was
like
late.
Yeah.
And
when did you listen to the audio?
What audio?
Audio coming in to you from the desk.
I didn't.
Just put the cards back. Put the cards back. Then you just pick the camera up. No, it wasn't there, so I'll just bring the camera up here then. Yeah, the tripod. And then from there we're gonna go upstairs and I don't know guys here tomorrow. Yeah, I'm here tomorrow. Yeah, and Carla, I think you will probably be here tomorrow. Yeah, you're gonna meet us on the roof terrace. I'll get you guys wherever you are. Yeah, I don't know exactly. But I'll just ask them. Cool. I'll just ask them. Okay, cool. And that,